Hello and welcome to this 8th class of the Protein Engineering course based on artificial intelligence. And on this occasion, we're going to review the entire world of AlphaFoldology. In particular, this is the 8th class of a total of 10 that make up the course, which is completely free and is available on the YouTube channel so you can delve deeper into each of the specific topics. And before starting the class, the classic reminder is that all the literature, images, quotes, hosted for the preparation of this course are found in the notes section of each of the slides. So for more information about the course, please check its official website. Likewise, check out its repository where you will be able to find tools, databases, as well as courses on AlphaFold, structural biology, among others, and you can go into more detail. To be able to download the slides and access the information, or to be able to adapt them to a course you are developing, you can do it from the Google Drive platforms or from that node. If you like it, you can leave a question here in the comments section of the YouTube video, or you can send it to me via any of these social networks, where you will be able to find me as the user at Myanguar. And finally, if you like to financially reward this course, because it was useful on a professional level, you can make a financial complaint via PayPal in the following link. So let's see what topics we're going to cover in this session. Clearly, they are the AlphaFold algorithms, AlphaFold 2 and 3. However, we are not going to explore the architecture of AlphaFold 1 in such detail, because it did not have a great impact in real terms on biology. And as a first topic, we are precisely going to explore the great impact that AlphaFold has had on science and, well, on biology in general, with respect to how we currently proceed now in carrying out our experiments. So let's review what the impact of AlphaFold has been on science, or more particularly on biology. And here we start first with 3 opinions from great experts in the area. The first of them could be André Lupas, one of the most recognized protein evolutionary biologists, who, in an interview regarding the impacts that AlphaFold could have, really recognized that it will change everything. Something that, yes, really happened, really changed the way of proceeding, of acting, of thinking, even of several colleagues. As a second opinion, we have here Shimbo Yu, who was actually previously working on this type of structure prediction algorithms using deep learning, particularly. At that time, convolutional neural networks were quite useful or quite interesting, so several teams were trying to obtain their algorithms based on this type of architecture. And Shimbo was precisely one of the main teams that led the predictions ranking. And even he now recognizes that biologists can now have some certainty in structural predictions, something that was not currently the case due to the poor qualities that were presented. Even one of the most renowned proteinologists in the world of biology, Janet Thornton, one of the creators of what is this structural classification system called CAD, Janet Thornton, even says that it is too difficult for these types of moments like those generated by AlphaFold to occur again. This great revolution that made many of us turn to look at these types of algorithms and start using them in our work to see, well, what new ideas could we extract from them? Something that, although other competitors have obviously tried to adapt their own algorithms, none of them are going to generate as big a break as AlphaFold did at that time. And this has precisely generated this idea of Alphafoldmania, which really exploded in terms of the creativity of scientists and how they precisely adapt this algorithm to their work. Such that even when we look again at the number of citations that each of the AlphaFold articles have generated, it is quite noticeable that they have had a great impact. Currently alone, in what would be December 2025, the AlphaFold 1 article has approximately 4,000 citations. The article that was key in this world of Alphafoldmania, which is AlphaFold2, already has around 44,000 citations, which is quite a lot in terms of the terms that can be cited in a scientific article in general, and what would be the publication of AlphaFold3 even since it is almost reaching more or less a quarter. That is, it has approximately 10,000 citations, which is also enough for a scientific article, and here we see precisely how this evolution was, which seems to be almost linear in what it was since the launch of AlphaFold. You can see it there from what was July 2021. Subsequently, some relevant milestones within the world of alphafoldology, which would be, for example, the launch of the AlphaFold version to be able to make predictions of protein complexes, until finally, what would be the release of the AlphaFold database with approximately 200 billion protein structures, which is obviously quite a lot, 200 million, 200,000 no, 200 million. And we effectively see that there has been a great option either at the level of preprints or at the level of published articles, where more and more teams around the world have begun to use the AlphaFold algorithm. And when we actually do a review of how many teams have used AlphaFold, we come to a number of approximately 3,300,000 users, which is quite a lot in terms of users of this database created by AlphaFold. And when we review what topics these scientists are working on, we obviously find what is the application of artificial intelligence for scientific discovery, and then actually, what are the applications within structural biology, such as molecular dynamics analysis, structural level analysis, enzymology analysis, and even what would be more applied cases, such as bacterial physiology or even research. Well, cancer treatments, as we can really see, the applications are extremely varied and well, to review a little more about it, I personally recommend these two resources. The first is that preprint by Google DeepMind, which is precisely a recovery of several of the great applications that have been seen at least within the first 3 years after the release of the model. And then this other report on artificial intelligence in science, which is much more detailed with approximately 100 pages, where you can review in quite detail how AlphaFold has been applied in science and what its results have been. This DeepMind has really done, let's say it, a great marketing and propaganda job, compiling what would be the most impressive stories of AlphaFold applications, in such a way that we can see that they extend to areas it is already being done, such as the treatment against what is resistance against antibiotics, for what would be the engineering of agroecological systems, to investigate the evolution of proteins themselves, the design of enzymes that can degrade plastics, to be able to solve issues related to heart diseases, or even to be able, normally working with these agroecological systems. See how we can benefit the entire term of beekeeping. So, as we have already seen, there are even more than 3 million researchers in more than 190 countries who have used this algorithm to carry out their work, and it has really opened many, many doors to research thanks to the release of the AlphaFold2 model. We evidently see this reflected now in what have been many of the covers of several quite recognized magazines, such as Nature, the classic one, Science, or even Protein, where what we see is that many of the covers now have as their protagonist precisely proteins, or even the algorithms themselves. Like, for example, when they launched what would be the AlphaFold3 system, as a new system to be able to predict biomolecular structures, that is, interactions between ligands with proteins, among other things, the construction of what would be the nuclear core of our nucleus as eukaryotes, which is an extremely large macromolecular complex, and even the progress in other algorithms inspired by AlphaFold, such as the great language models, which also already have quite extraordinary generative capabilities, among other topics that, well, they are evidently reflected in these quite beautiful covers of multiple magazines. And that evidently led us, around 2024, to what was the Nobel Prize given to these three great researchers, for whom I think several of us have great intellectual admiration, to David Baker precisely for all his work related to what would be the design of proteins, and to Demis Hassabis, as well as John Jumper, for the direction of the team that set up all the AlphaFold training. But in addition to these three characters, it is quite clear that 2004 was also the year of what deep learning was, because even Geoffrey Hinton was also one of the winners in the world of deep learning of the Nobel Prize, and where several turned to see precisely the great capabilities that this type of algorithms used in deep neural networks have to be able to really apply them in their particular scientific areas, and therefore contrast with what was previously known, as well as perhaps find new patterns that could provide us with more information in our particular research. This clearly did not happen just because of these three people, but it happened through the work of many, many people. For example, at the top we see what would be part of the team that integrates DeepMind for the specific development of AlphaFold, which is approximately even more than 40 people. And at the bottom we see several of the doctoral students, including postdocs, who have spent time in David Becker's laboratory and who, thanks to their creativity, their work, their time, their money, obviously, because they were able to make many of the works that have emerged from their laboratories a reality. And obviously, this is not only a product of the work of these communities, but of the global community of scientists that has deposited in public and free databases the information that they have, obviously, analyzed over several years. Right. In the protein databank, right. Which, well, we can really appreciate them for what they are, which are, well, public goods to which, well, other quite relevant databases are also related, such as, well, the UniProt, which has approximately 250 million protein sequences, which come mainly, well, from metagenomics experiments of several people who at some point on this earth, well, went to sample some puddle, well, somewhere in the mangroves among others to be able to sequence and upload their sequences precisely to this database, so that other scientists could analyze it and precisely be able to access their data, as well as thanks to other quite famous algorithms such as MMSX, such as Amber, even Hammer, which, well, the AlphaFold team integrated it into their framework to be able precisely to make AlphaFold a success, and even, for example, having a vague approximation of how much it would cost just to replicate all the experiments that led to the consolidation of the PDB as a database. Approximately $20 billion are estimated to be able to reproduce the structures, and this being quite concise in the estimates, that is to say, this database costs billions of dollars, and that currently many companies and many conglomerates that have economic interests behind them can access free of charge to this type of resources, truly achieved thanks to the work of thousands of scientists, as well as all the personnel who are in charge of maintaining precisely the infrastructure of the research centers where scientists work. And obviously, what is not only the protein structures as a public good, as a root of the success of AlphaFold, but also its adaptation to a form that was quite easy for the public that does not have knowledge of bioinformatics, quite detailed programming. So another quite relevant tool that helped its popularization or democratization within the world of biology also comes into play here, which would be AlphaFold. A platform that does is use the cloud that Google provides us, called Google Colab, which can make inference using the GPUs that Google lends us for free. And it really is quite easy to install, quite easy to use, and it is also quite good and efficient. In order to learn a little more about how AlphaFold works, you can review either of these two publications, which is the original, where the entire infrastructure behind AlphaFold is precisely presented, as well as a protocol that is sufficiently detailed to be able to make predictions of either monomers, multimers, or make predictions a little more en masse when they want to predict a few hundred or thousands of proteins, using this type of adaptation of what was the original AlphaFold algorithm. But in this case, the authors make it slightly more efficient because precisely to improve the structure predictions are made in terms of speed, as well as in terms of the number of homologs that can be recovered by configuring a fairly well-curated database that integrates various sequences from several environments in particular. Even when we turn to see what the impact of AlphaFold has been on the democratization of structure prediction, we see that there is only an approximation by its three main authors, such as Milod Miranda, Merdita, Martin Steininger, or Sergei Ovchinnikov, who even tell us that at this moment, approximately in February 2023, they had approximately 3,000 runs using the AlphaFold algorithm, and even what this MMSX service is to be able to generate multiple sequence alignments, since there were already around 5 million services, jobs, whatever you want to call it, to be able to generate this type of information and use it for structural prediction. So we see that obviously also what was the public investment in this type of projects that are for the benefit of the entire community, since they have evidently contributed a lot to what is the success of AlphaFold, well now as in its applications within biology and in structural biology. And this is quite relevant, since if we wanted to have a personal computer to be able to run this type of algorithms, actually it is quite expensive. An approximation could be about 100,000 pesos, approximately $5,000, and well, this is generating a computer such as a relatively common desktop computer. But in the case that we were looking to apply AlphaFold already in terms of what scientific computing would be, obviously it requires the purchase of servers, as well as GPUs of a more scientific nature, and not so much of the domestic range as NVIDIA's RTX could be, since in this case, the prices of a server or a GPU already skyrocket to approximately $10,000, $16,000. So what is this use of the cloud that Google Colab provides is really quite valuable for most of us, especially in Latin America, where we do not have the financial resources to be able to buy this type of infrastructure. Just to be able to have AlphaFold running on a computer. So it is suggested that we should have approximately 2 TB of storage as desirable SSD type storage. I think this is even too small because sequence databases, such as Uniprot among others, are quite heavy. So I think it would even be better to have at least 10 TB because obviously, not only are you going to search the database, but you're going to generate a lot of information about this computer, so you don't want to find yourself short on storage. Approximately 750 GB of RAM is even suggested, which is quite a lot, and obviously if we use RAM of this type, more of a scientific type, which corrects all the errors that may occur due to bit changes, it is usually much more expensive. Or even also in terms of the CPU, well, it is clear that we need a machine that can coordinate quite well what would be the use of billions of sequences. So a minimum CPU of about 16 cores is recommended, and the appropriate thing would be to have a CPU more aimed at what would be the high performance segment with about 32 or 64 cores, so that it does not create a bottleneck when computing the structures or when searching for homologs. And then actually, what is the use of GPUs to be able to load these types of models and make the inference, since it is suggested to buy what is the range with the highest performance of the NVIDIA domestic range, to understand what the 90 series would be, such as the 3090, the 4090 or currently the 5090 from NVIDIA, which alone cost approximately $2,500, the equivalent of about 50,000 Mexican pesos. So as you can see, really only the GPU constitutes approximately half of what the computer would be. So in case you do not have the financial resources in favor, it is already clear that the option to choose would be cloud computing offered by Google Colab for free. And now let's address what was the world of structure prediction seen as a competition, where in particular this favorable academic environment was created to be able to develop this type of algorithms, which is known as CASP for its abbreviations, or critical assessment of structure prediction, which is a competition where scientists from all over the world propose to improve the predictions of protein structures with each year, right? With algorithmic, scientific, biological inventions, to be able to integrate all this type of information in the algorithms and be able to make structure predictions from the sequence. And if we look at how this type of competition evolved, we can see that in a cartoonish way, it really went from a really much more artisanal configuration of inputs during the first steps of this type of competition. Even algorithms that are practically completely designed by the computer, that is, the representations that are used to make predictions now already do what transformers or, in general, deep neural networks are, which as we already reviewed. This type of algorithms is characterized because they are capable of obtaining these representations rich in information, and we really do not have to do this entire feature engineering process to design a good predictive framework, right? In this sense, the first CASP competition was proposed by John Mouth approximately in 1994, and the last competition, for which we are even close to knowing the results of the next one, would be CASB17. The last one occurred in 2024, so soon we will be finding out who the winner of the next competition will now be. And obviously, during all these years of research, particularly in this competition, there were many advances by multiple scientific communities that made AlphaFold a success as well. In particular, we're not going to cover the entire development of the CASB. As you can see, there are some improvements in terms of searching for homologs using remote homology algorithms at the sequence level, whether with CBLASD or HAMER. Then what was consensus engineering? And then using what would be more of an option, such as using these shared fragments between multiple superfolds or non-homologous proteins. But precisely by having these structural signatures, we can relate and further investigate what their possible evolutionary relationships would be, and therefore improve our predictions. Even the modern era, let's call it that, where a large part of the community turned to see what would be the information that we can obtain from the contacts of proteins, using precisely algorithms based on deep learning, which is precisely what was part of this success related to AlphaPod. And currently, we are already in this era where what is known as multimodality is incorporated, where we can incorporate structural biology experiments of multiple kinds to be able to, well, precisely to know or characterize a specific biological object. However, it must be considered that the problem that this CAP competition sought to solve would really be more related to structure prediction and not to the problem of protein folding. Because protein folding is a problem that is much larger and more complex in this sense, which, as we can review here, we could really say that it is made up of three major pillars in general. The first would be precisely the one that AlphaFold sought to solve, which is the prediction of the structure from the sequence using deep neural networks. The second pillar would be the quantification of the atomic forces that now constitute this new prediction, such as hydrogen bonds in the van der Waals forces, salt bridges, among multiple other molecular interactions that we previously reviewed. And the third pillar would be the characterization of these energy landscapes to precisely know the folding pattern that is adopted over time. So first we must recognize this state of conformational assemblies of the proteins, and subsequently evaluate how they are distributed in energetic terms, and how contacts are established over time to see how precisely their folding pattern is formed until reaching what would be the harmful structure of each of these foldings. So as you can see, actually, the problem of structure prediction is completely different from the problem of protein folding. Something that is usually quite poorly communicated, even when one can review documentaries made by DeepMind itself. We find that much more frequently than we would like, they actually refer to this problem as the problem of protein folding, something that has not been resolved, as would be quite clear, and that perhaps what we could say has been resolved would be what structure prediction is, perhaps mainly in terms of monomers, and which is now diversifying to what would be macromolecular complexes, as well as biomolecular complexes with non-protein entities in that sense. And well, starting from the idea precisely that we can predict the structure of proteins, we must recognize the relevance of the seminal works that suggested that this was possible. And everything can be traced back to what would be this seminal article by Christian Amundsen, who actually performed a relatively simple but quite powerful experiment in this sense, where what we can observe is that what he did was take a protein, particularly a ribonucleose, which is maintained in its native state thanks to the presence of disulfide bonds, as well as the interatomic forces that we previously reviewed. And then he added two denaturing agents for the protein's strength, such as urea, which in this case can break non-covalent interactions, such as the hydrogen bonds or the hydrophobic forces contained in what is the core of the protein. And the second agent was 2-mercaptotenol or mercaptotenol, which what it does is reduce precisely these disulfide bridges and therefore destroy them in that sense. So what he observed in vitro is that by adding these two denaturing agents, precisely now we obtain the denatured form of the protein, but that by removing them, then the protein is able to adopt its folding pattern as it originally was, which was quite interesting in terms of carrying out this experiment in vitro, since it then suggested that what the structure of the protein is contained in informational terms in its sequence. This is an observation that John Mott even highlights in one of the interviews, so that it is quite evident that the structure is then encoded, or we can approximate that it is encoded in what would be its chain of amino acids, which implies that one can really use computational methods that start precisely from the sequences of the proteins to be able to make this structural prediction. And that is why, due to this observation, we began to delve deeper into the type of structural prediction algorithms. Well, in order to have this advance in theoretical terms, and perhaps even extend the hundreds of thousands of dollars that the crystallization of a protein can cost, as well as the time necessary, since, as we previously reviewed, which is to find the specific conditions for which your protein can be crystallized and obviously obtain a pure crystal, since they are quite complicated in general, then a doctorate can even relate to only the crystallization of a single protein, which is why it can be quite complex to find these types of solvent condition to create this type of crystals. We must also recognize what would be the first works on the use of neural networks to predict the structure of proteins, which in this case were mainly aimed at what would be the prediction of the secondary substructure, patterns of alpha, helices or folded betas or turn regions, where we find two main works, both proposed in 1988 at the University of Jeff Hawkins, as well as Harvard or Camp, among others, where what we found is that really the systems that could be found at that time had a performance of approximately 63% success in being able to predict this type of secondary structures, which was not really useful at that time to be able to do exploratory or even characterization analyses, and which was something that even the authors themselves recognized at that time, calling particular attention to what would be the great limitation that the databases presented at that time. As we can see, in both cases the same authors suggest that what it means to have a greater number of sequences, since it could improve the predictive capabilities of this type of algorithms based on deep neural networks. Something that at that time the sequences were not counted as 200 million, as is currently common, but rather was much more limited because massive sequencing had not yet been democratized through projects such as metagenomics in this sense. Another great work was done in 1993 by two great proteinologists, such as Bookhard Rost and Chris Sander, who also adopted this idea of being able to predict the secondary structure of proteins from their sequence. However, in this case, they now integrated not only information derived from the sequence, but now the evolutionary information derived from multiple sequence alignments. An idea that was even taken up by the AlphaFold algorithm, and that is even the basis of success, precisely as we're going to review in following slides, and that in this case the secondary structure prediction reached approximately 70% accuracy, using the evolutionary information derived from alignments, which could even be relatively comparable, at least at that time, in the type of characterization that one could do using circular dichroism experiments, and that could really save both time and money by using this type of algorithms. And maybe also, very important is the idea raised in this same article, where it is important to include within this type of predictions a type of confidence index that helps us determine which prediction has what quality of confidence in which specific residue. An idea that was also adopted by the AlphaBot algorithm as well as many other predecessors, or even BLAST when comparing protein sequences, also gives you a confidence index, which is quite relevant in bioinformatics terms because when one proposes one's algorithms, usually uncertainty or confidence in the predictions is not usually a characteristic to be considered during the development of this type of algorithms. And yet, it turns out that what is this degree of uncertainty or confidence that we can associate with each prediction is of great value to biologists in allowing them to consider whether it would be useful to incorporate these types of predictions instead of proceeding with a more experimental framework, which is really important as part of the success of what was the AlphaFold algorithm. And in this sense, we must also recognize the great work that was carried out by the Butcher-Dross laboratory, which quite consistently continued to promote the idea that this type of algorithms based on deep learning could be very useful to characterize the three-dimensional structure of proteins. The Buchartrost or Roslov laboratory has actually been doing this type of work for about 2 decades with its first adaptations, as we already reviewed, such as the prediction of secondary structure, based on the incorporation of other properties, such as the percentage of the surface of the proteins exposed to the solvent, even in the incorporation of evolutionary formation derived from amino acids, or even in more recent terms, the first language models applied to proteins not using transformers architectures, but using architectures, let's call them that, predecessors such as Elmo, which we also already reviewed in the transformers class, and this consistency also led to the idea of being able to apply this type of deep learning algorithms, not being stuck in time as a curiosity, but rather demonstrating that it could be a really useful application of these algorithms. Here you can review what would be a compilation of the first works, at least until 2023, of how this type of networks were used to predict properties related to the structure of proteins, and that in the following slides we are going to investigate in detail what these types of algorithms can now do in terms of structural predictions. The great contribution of multiple works is also to have main ideas that were taken up by AlphaFold to be included within its framework, and obviously, to be able to exploit the information contained in this type of ideas. The first is precisely that we can see the structure of proteins as a kind of distance map. Here what we have is a comparison of different residuals on the x-axis and on the y-axis, and then we can do this idea of measuring how much the distance is in Armstrongs. Of a residue, for example, yellow in relation to blue, in relation to green, in relation to purple, and then have a kind of distribution of distances for each of the residues. And as we can observe this, it could be seen as a matrix of distances, which considers at what range of distances in Armstrong each residue is separated from each other. Something that if we want to make it a little more discrete, we can set a cutoff point, a threshold. In this case, usually one of the most used thresholds is 8 amps away to consider that 2 amino acids are in contact. And then we can say that if we apply this to discretize it, then there would now be contacts between what would be the yellow residue with the red residue, or the blue residue with the blue residue, or the purple residue with the blue residue. And then we can make these distributions of each of the residual in terms of distances, now seen as a more dichotomous or binary object in that sense. The other great idea was precisely the incorporation of evolutionary information derived from multiple sequence elements, where we found this idea of correlation between residues, such that if one residue is mutating and correlates with the other, it is possibly done because they have some structural relationship in precisely the three-dimensional structure of the protein. These ideas were evidently explored by multiple other groups. Perhaps we could trace the use of these two types of information to what would be publications from approximately 1997, which in this case were also developed by other laboratories such as the laboratory of Sergei Yevshenikov, Mohammed Al-Qureshi, Deborah Marks, Marcin Weitz, who were consistently investigating how to adapt this type of evolutionary information, as well as distributions of distances of the structures to be able to make predictions. Even with relatively more recent publications, but preceding AlphaFold, such as the use of metagenomic information, that is, millions of sequences obtained from multiple environments to be able to exploit evolutionary information, and thus be able to make increasingly better structural predictions. It is quite relevant to recognize what the contribution of this type of information is, and then you have to look at what they mean in particular, which they can mean. Note in more detail in this review that I highly recommend it, but in summary, we obtain these two alignments. As you can see, here are some alignments where we can find a first column of amino acids, which are super conserved in this sense, either because they are quite relevant in what would be the analgan site of a protein, or because they are related to what would be the hydrophobic core of the protein, something that we now recognize is relatively important, because it is thanks to this hydrophobic core that the protein can adopt its native conformation. Then we see that in the vicinity of what would be the active site, a certain variability of residues can be found, such as here some arginines or valine, that they would effectively be modifying the behavior of each of the homologs within this superfamily to be able to actually attach to each of those substrates with which it is related. Or we can even find residues that, although they are installed in the sequence, are relatively contiguous in the structure, and therefore perhaps they would be establishing some type of molecular interaction to be able to stabilize the protein, which in this case we can observe that they would be correlated in such a way that they always complement each other in this sense. Even this complementation can not only be found between residues of the same protein, but we can also find it between residues of different proteins, as could be now the second orange alignment, where we find that there is also now a correlation between the type of residues that are here, now indicated here with this other blue color scheme, where we see that now, precisely when making a protein complex, perhaps these two residues are establishing an interaction that leads to stabilizing precisely this complex, right? So then we can find precisely this type of correlations, that if one atom mutates, then the other necessarily has to mutate into a corresponding equivalent in order to maintain either the properties related to the function or the structure of the macromolecular complex, which is obviously rich in information to really improve the prediction. And in this sense, well, a great variety of methods had previously been explored, some obviously suitable, taking into account what would be the type of interactions between residues between proteins or within a protein in blue, where we can see that a derivative was also made of the information that we can find from metabolic pathways. Interactions between complexes, protein-protein interactions, using the information of the active sites of allosteric networks, among multiple other sources of information that were incorporated into this type of algorithms, and that precisely attempted to exploit the information derived from multiple sequence alignments in order to improve structural predictions. That is, all the evolutionary information obtained from an alignment really represents quite rich information with which even current algorithms can be used. They base their predictive capabilities at structural levels, which we will review in detail in subsequent slides. We must also recognize the main events and programs that led precisely to the success of the prediction of protein structures. As we can see in this timeline, that we can really find quite a few milestones, as we can see in each of the boxes, but perhaps just highlight the most important ones, such as the creation of the PDB, where we can deposit protein structures and access them easily and for free above all. The creation of the CASP competition, which brought together and motivated many scientists to increasingly improve their predictions. The creation of what would be UNIPROT, where now multiple scientists can deposit their metagenomic projects, their thousands or billions of sequences, so that everyone can access them. And finally reaching the milestone of AlphaFold as an algorithm that has a success in being able to solve the problem of protein structural prediction. And as we see, there are obviously many predecessor algorithms, many databases that were also part of the success in this sense, and that this could be more related to the world of biology, but we must also recognize other milestones within the world of deep learning or machine learning in this sense, such as the emergence of convolutional neural networks since approximately 1990. What was the AlexNet moment, where the vast majority of the artificial intelligence community turned to see these types of deep learning algorithms as a possible great tool to address multiple problems, which was the transformer architecture, which really is the core of virtually any successful algorithm today. Or even what would be your application to structural predictions that integrate a more specific geometry, that is, a variant known as the SC3 transformer, which has much more refined geometric information to be able to make predictions related precisely to three-dimensional entities, exactly what was the first version of AlphaFold, which in this sense used precisely convolutional neural networks, and that evidently thanks to the adoption of the transformer the predictions in this sense could be improved even further. So, as you can see, really reaching the moment represented by AlphaFold is based on the great contributions of multiple other works in multiple laboratories, whether in private industry as well as in academia, which led to the consolidation of this type of algorithms as a great tool today. And in that sense, we can ask ourselves, what was DeepMind doing at that time? And in particular, DeepMind is quite recognized for its research in the world of reinforcement learning, where their first steps were precisely the idea of being able to surpass human capabilities in multiple games, such as the game of Go, as we previously reviewed, or even in some other games such as StarCraft, a game that is played the same on PC, among multiple other agents that they even trained using that type of algorithms, but that in general they share three great characteristics. They have a wide range of possible answers where you can search. They also had a fairly clear optimization metric, and evidently, there were also large amounts of data with which to train the AlphaGo, AlphaStar, or AlphaZero algorithms to be able to solve this type of game, and that by taking advantage of this gamification, the algorithms could now predict precisely how to win a specific type of game. And the same idea of adopting reinforcement learning algorithms. In the first instance, an attempt was made to apply what would be a game developed by the Protein Design Institute, an institute where David Becker is director. And in this case, the central idea, at least in its first steps, was to try to teach an agent who could precisely play this game called Fold It, where an entire platform was open to the community so that, through gamification, well, they could precisely help the scientific community so that cures could be carried out at a manual level by a human and precisely detect which residues could be in contact with a protein, which would be more relevant in others when investigating the alignments between multiple other options that can be made in this game. And well, by teaching several games precisely, the idea was to teach this type of reinforcement learning algorithms that could learn to play this game. However, little by little they adapted the idea of using convolutional neural networks and eventually transformers, and this idea of gamification based on Fold It was somewhat discarded in order to teach people how to play protein folding. And then we have to review precisely how these types of AlphaFold1, AlphaFold2 and AlphaFold3 algorithms work, which we are going to review below. So in this case, well, let's address how it works and very briefly what the AlphaPod1 algorithm is. In this case, one of the great contributions, at least at this time, was to think of proteins now not only as three-dimensional entities, but also to represent them in a two-dimensional way with these distance maps, something that they called histograms in particular. And so now we can use precisely this type of convolutional neural network algorithms to adopt three-dimensional information, but now convert it into two-dimensional form with this type of maps. As such, what AlphaFold1's algorithm was is published in these two articles, one in Nature and the other in Proteins, where they already describe in detail how their specific algorithm works. Here we're going to review them in a fairly brief way, and here only draw attention that within the authors in the Nature publication. It is highlighted here in one in blue, which was John Jumper, who at that time was practically newly hired, and that later this character would have a great contribution in terms of his biological contribution to inject into this type of algorithms, because he precisely has a doctorate in terms of physical chemistry and protein folding, and that by being director of this project, well, he was able to precisely coordinate his team to be able to integrate better biological, evolutionary, and physical chemical bases into this type of algorithms. In AlphaFold1, the first thing we did was consider precisely the distance relationships that exist between each pair of residues, as we can see here in this diagram, where a particular residue is placed in the center. And then these yellow lines would represent each of the distances that residue has with respect to others in Armstrong's terms. We can think of proteins now as relationships or distributions of distances in this sense. And this is precisely what is represented with this type of histograms, which in this case were renamed histograms, where what is done is to group these ranges of distances in the form of 64 categories or bins, which consider ranges from less than 2 amps of distance to more than 22 amps of distance. So now we have precisely this type of two-dimensional histograms, where practically what is counted are distributions of distances between pairs of residues, which, as I recommend, are known as histograms at least within the AlphaFold jargon. And we can then represent the three-dimensional structure of the protein in the form of two-dimensional maps, as you can see here. The idea then is that we can adopt a convolutional neural network, and that, starting from this type of images, which really represent a three-dimensional entity, we can precisely predict the atomic coordinates of this type of protein. AlphaFold1 as such is represented by three large networks, one which is precisely a convolutional neural network, which is responsible for predicting this type of histograms, as well as the torsion angles, these IV angles that we already reviewed pi and psi, which also help us precisely to predict the atomic coordinates of each of the protein atoms. A second network that does is evaluate the reliability that this folding could have using the global distance test. And a third network that has a more generative behavior, which we can even see is a quite complex architecture in that sense, because it is an autoencoder that uses convolution operations. But it is also a recurrent network of the LCTM type, which in this case having a generative behavior precisely also helps to be able to precisely show different fragments of a protein and then assemble the structure in its entirety. For more details on each of the architectures, you can obviously consult the specific articles. However, I would not recommend it so much, because the success really arose from the implementation of transformers in what was AlphaGo 2. So perhaps this would remain a little more anecdotal, so we are going to review it very briefly in this sense. As you can see here, the framework is this. We obtain the sequences, and then we look for their homologs to be able to construct a multiple alignment of sequences from which we can derive a quantity of statistical information related to what would be the correlation between residues, as we reviewed previously. Later, we pass it to a convolutional neural network, and what this neural network is going to do is predict the distance between each of the atoms and also their ihedral angles that allow us to parameterize the predictions that are then later used by the gradient descent algorithm to precisely consider other potentials that in this case are scoring functions that help us to precisely evaluate the quality of the predictions, and these potentials can be in terms of the distances of each of the pairs of residues, the torsion angles, as well as whether they are colliding precisely in the three-dimensional structure, because thanks to what is the gradient descent, we can parameterize these three facets in an integral way and find a general minimum in which a protein fragment can be found that may be associated with what is the native structure of the protein. As such, the AlphaFold architecture, or at least the main one, which is the convolutional neural network, is precisely based on this histogram information, as well as information on between pairs of residues derived from the alignment, as we can see in this case, and then they are traversed with these kernels, as we previously reviewed in the deep learning class, and then they are grouped until they generate feature maps, which eventually arrive at a rich representation that eventually will end in the prediction precisely of these histograms, which, as we already mentioned, represent what is the two-dimensional form of the three-dimensional structure of proteins. And thanks to the implementation of convolutional neural networks, it was seen that deep learning could really be a great tool to predict the structure of proteins, as we can see in this other diagram, where what we observe are two metrics of structural similarity, the DEMS score and the RMSD. In red, the RMCD, and as we can see here throughout the training, it begins with a fairly large RMCD of approximately 40 Armstrongs, which is then minimized through this idea of potentials until reaching an RMCD of approximately 5 Armstrongs at the end of the training of this convolutional neural network. And something relatively the same. The same thing happens with the TM score, which in this case starts from a value of 0.2, remembering that a value of 0.5 is indicative of a fairly similar fold. And in this case, we see how this TM score is precisely improving until it reaches approximately 0.7, indicating that actually the fold that we are trying to predict with respect to the real fold with which the data is trained would really be adopting the same three-dimensional pattern in this sense. So in this sense, it was demonstrated that the use of convolutional neural networks was quite useful in predicting the three-dimensional structure of proteins. And well, when we look at it in terms of comparison, some authors suggested that, well, if this trend continues, at least within 5 years, with even more research, we could reach global distance test values of approximately 85% in the predictions, which in general terms could correspond to a solution to the protein structure prediction problem. So the global distance test, as we already cover it in the class of protein structures, helps us to consider that a structure is also predicted, being values greater than 80, some that we could consider to really reflect good positioning of side chains, and values greater than 70, which would represent a fairly general folding, but well predicted in this sense. And what we observe here is precisely the performance that has occurred throughout the multiple CASB competitions, in this case being the last CASB in the particular figure, CASB13, in which AlphaFold participated. And then we see precisely on the yes-axis this percentage of GDT with respect to the difficulty of each of the structures to be predicted, such as easy structures, moderately complicated structures, and others that are much more complicated. And what we see is that evidently the proteins that are easy to predict, because perhaps they are numbers with many homologs in the databases, have a GDT greater than 80%, suggesting that their predictions have good quality even at the level of side chains. However, when we move on to what the models are at a medium level or even at a difficult level, we see that this GDT falls below 80%. But in this case, thanks to AlphaFold1 and the use of convolutional neural networks, we see a great improvement from the other contenders, well, in the other CASB competitions, which really did not use this type of convolutional neural networks as massively as DeepMind did, because thanks to DeepMind it also has a great computing capacity, since Google is precisely supporting them, and this computing capacity is not so common in laboratories, at least within the academy. So this was also a great contribution, at least as a research team, but now coming from within the business sector. And precisely what we observe is that in this new CASB competition, AlphaFold1 manages to exceed this 60% limit, where we can consider that in general terms they are predictions that are somewhat acceptable, but not really good, but rather, they remain within this acceptable range. And then when we look at what happened when AlphaFold2 came out, well, we see quite drastic changes in this type of diagrams. It is evidently the same type of diagram. On the y-axis, it is what the percentage of G of t is, and again, on the x-axis, it is the difficulty of each of the structural predictions being easy, medium, and difficult. And what we see is that now, thanks to the implementation of the transformer architecture, AlphaFold is now within the range of 80 G of t, which could be considered as predictions that are now already good, and that even in some cases are already very, very, very good, even at the level of what would be the positioning of its side chains, which obviously represents a great success when compared to the other CASB competitions, even when we compare to what would be the same CASB 14 where AlphaFold competed. But in this case, if we remove precisely the contribution of the DeepMind team represented by this green line, we see a reduction of precisely this GDT of approximately 65%. And that it was actually the predictions coming from this AlphaFold2 team that raised the GDT from approximately 65 to above 80. That is to say, the AlphaFold algorithm was actually the one that represented the great moment of success within the CASB 14 competition. And this, because what we can now see more clearly is precisely the evolution of each of the CASB over time with respect to the GDT measurement. And what we observe is that in CASB 13, AlphaFold1 had a CASB in general terms approximately 60, and that with AlphaFold2 in 2020, a CASB was reached that was already very close to 90, and that even if we discard some much more difficult macromolecular complexes, this GDT metric can be raised precisely to 92.4, indicating that they are actually already very good quality predictions. Even when we compare what these AlphaFold2 predictions represent, regarding the results that we can obtain through experiments such as X-ray crystallography, we found that the AlphaVolt predictions would be comparable precisely to this type of structures, reaching a resolution of approximately 1.1 to 1.5 Armstrongs. Or in other words, now we can save all these years of work and money of work. We can save these years and money used to be able to crystallize a structure. Three-dimensional we already found predictions of approximately 2 values of RMCD, as suggested precisely in this article, which was really quite, let's call it groundbreaking, let's call it catastrophic in this sense, because it forced us to change the thinking that we had about how we can precisely implement these types of predictions, even though it is nature for them to be predictions. In our structural characterization of multiproteins, because before we only had quite low reliabilities, and that thanks to this type of algorithms based on transformers, we now have structures that are equivalent in terms of their qualities with respect to experimental methods, and this has evidently come to bother several scientists in the community, mainly in the crystallography community, who defend that it is evidently not comparable or replaceable in this sense. With which we must also agree in that sense, because obviously there are different information that can be obtained by both types of experiments, and that it will not always be possible to resolve very good quality structures in both methods. So well, now we have precisely algorithms that help us save all these years and dollars, so that we can now have a prediction of what the three-dimensional structure of proteins would be. And due to its importance, now we move on to explore precisely how the AlphaFold2 algorithm works, as well as the necessary context to understand this type of algorithms, where perhaps the main contribution in this case made by the AlphaFold2 version is that proteins are now being represented as a series of residues that are evolutionarily aligned, that in particular when we talk about residues, we're actually talking about tokens, because what is really used as a central idea within this algorithm is a transformer, and that precisely by being able to compare between pairs of tokens, or what is the same between pairs of residues of a protein, we can gain more contextual information about how these types of three-dimensional structures are structured. So in order to understand precisely how AlphaFold works, we have to look at the context in which all this phenomenon was precisely occurring. Strictly speaking, as we already reviewed, the transformer architecture was suggested since 2017, and what was AlphaFold2 appeared in 2020, and it was quite sensible to expect the type of algorithms that were behind this success precisely. It lies in what the transformer architecture is, which, as you already know, is made up of what is an encoder module and another decoder module, which what they do is share information to create semantic representations that are very rich in information and thus be able to make their predictions, and that now you also know that there are transformers that are of the type only encoder or only decoder, each one aimed at a special characteristic. Those that are only encoder specialize in being able to regenerate rich representations of information. Encoders are more generative in nature. And when we turn to look precisely at the year in which AlphaFold2 was launched, we find in this map, or in this tree of life of language models based on transformers, that one of the great milestones in natural language processing had already occurred, which was the launch of the GPT-3 architecture, which in this case also made many researchers look again. This type of algorithms, at least in their generative capabilities, to be able to adapt them to what their applications are within science, which really was kind of a dream to see that this would be reflected in the success of AlphaFold, as we already know. And later the launch of GPT-3. As you can see, there really was an enormous diversification of language models of a generative nature, having language models that were specific to what the scientific world is, such as Galactica. Language models that allowed us to characterize these scaling laws, such as the Chinchilla language model, among other language models that are evidently the basis of the current revolution within artificial intelligence, along with what would be the diffusion models. So the idea is that we can adapt these language models to now model proteins, because they share certain similarities between both informational entities. For example, when we compare the text, well, we can see that there was a certain structure, such as the classic subject-verb-predicate based on words. And then we form sentences that, then with a more enriched context, we can declare if they can be associated with a specific feeling, a theme, or even if they are typical of a characteristic language. And this turns out that we can transfer it to what would be the world of proteins, in such a way that we can also find local properties such as those that could be precisely sites related to post-translational modifications that may occur, the presence or not of regions of secondary structures, whether alpha helix, beta-folded or turn regions, diesel-Fure bonds, atomic interactions, among multiple other properties that can be related to what is the world of protein sequences, and that thanks to their context, we can now precisely point out global properties that could be related to what is their location within the cell, whether or not they correspond to an organism within the taxonomic hierarchy, or whether or not it could be associated with a stable folding that manages to adopt its native conformation within the cell. The idea is then that we can take this alphabet of 20 amino acids that we use to represent protein sequences, and then apply these tokenization algorithms. In this case, one of the most common applications within the first language models that were developed, it was precisely tokenizing at the level of waste. However, there is evidently more diversification of the type of language models that can be applied to the world of biology, so that we then find language models that tokenize at the level of nucleotides, at the level of codons, even at the level of chambers or at the level of domains, where really each of these units can be seen as proto-linguistic units, given that they precisely aim to represent what this macromolecular entity is within the cell. However, as you also already know, tokenization is one of the most key steps in the training of any transformers, and that can also precisely condition the type of results as well as the type of information they can capture. Therefore, it is really sensible to adopt this 20 residue scheme to be able to precisely model protein sequences. But there will also be language models that now aim to model not only protein sequences, but also protein structures. However, we also have to precisely design some tokenization scheme that allows us to represent proteins as discrete units, which is really a research topic that is quite difficult to address in this sense. However, we can notice that there are some similarities at the level of proteins, as well as at the level of languages, so that we find these same ideas where we can classify the great diversity of languages that we have into what would be approximately 140 linguistic families that in principle would have precisely diverged within ancestral languages, such as European Protein, later Latin, among multiple other languages that emerged throughout the evolution of humanity, and that they precisely share these similarities in syntax with respect to what proteins would be. For example, when we look at what would be the primary structure of proteins, we obviously have a chain of amino acids whose comparable equivalent within language would be precisely the idea of alphabets. When we compare the secondary structure of proteins, we can now think that the equivalent in language would be precisely the idea of words similar to what secondary structures are. Subsequently, we can arrange this set of words in such a way that they now contain a syntax and then provide a meaning, a context to a complete phrase, thus precisely forming increasingly longer phrases and sentences, which would be equivalent to what the tertiary and even quaternary structure is, when they are already gaining much more context within complete paragraphs and key ideas that we seek to share. And this is also reflected even at the level of how letters are used within the language. For example, there will be effects that are quite terrible when we look at the change of a single word at the level of what said phrase seeks to communicate. For example, in this case it is said that your manuscript has been accepted for publication, and by changing it, in this case in the English word now to a T, which would be no, well, your manuscript is now being rejected for publication. Just by changing a W to a T, I completely change the meaning, which would be equivalent in terms of proteins, some mutation, perhaps some disulfide bond, which, well, when it is broken, then the entire structure of the protein now no longer adopts its native three-dimensional structure. We also find cases of recombination of fragments, such as the case of circular permutation in proteins, where the connectivities found between gyres can be changed, but precisely the same pattern of secondary structures is maintained. As we could see in the phrase, I am happy when it rains, and when it rains I am happy. They really look for or share the same information in this sense, and in the case of proteins, simply by rearranging what would be the loops or turn regions. It can be precisely rearranged, but share the same meaning, which in this case would be the three-dimensional pattern of the protein. However, we must also consider that what is a folded structure does not always really translate into a structure that has a utility, a function within the cell. As we could observe, what is the behavior of abelloid fibers, which we already reviewed previously, which precisely they form superstable aggregates that become cytotoxic in the cellular system. In a similar way, then we can find that a correct grammar does not necessarily mean some idea of a meaning, or something logical in terms of the ideas that we seek to share in our language, such as this word that would be colorless. Grenadillas sleep hurtly. Well, they really are correct words, but they don't really share any particular meaning in such a way that it is also similar to how an amyloid fever would behave within the protein sense. Well, it actually even becomes harmful in this sense, so we also see that there is a certain relationship in the form of how these two informational entities evolve. For example, we can see that in what is the evolution of language, we have words that have these signatures or these morphemes that seem to be shared between multiple words that can be used during different contexts or whose meaning can be completely different. In this case, we have several words such as isomorph, halomorph, mesomorph, metamorph, morpheme, morphology, and all of them, as you can see, share the word morph, which would be equivalent to what the behavior of protein structures are like that come to share these structural signatures, such as the P-loop motif, which is shared in multiple protein folds in multiple superfamilies, and that fulfills a particular function within each of these cases. Also, the way in which proteins evolve, as we already discussed, can precisely start from the idea of these segments that are shared across multiple floats. For example, we could see here how one of these structural motifs, fragments, or signatures, whatever you want to call it, is evolving, which in this case is the beta hammerhead motif. In this case, we can speak of that their homologs, specifically the orthologs, such as ribosomal proteins, precisely share this ability of this motif to be able to bind to what are RNA molecules. We can find that this motif is found in different contexts, that is, in different proteins, but its function is always maintained, which is precisely the union of this type of nucleotides. However, we also found that throughout evolution, this type of homology can precisely change in such a way that it is now a paralog due to duplication events and genetic divergence of both its function and its sequence. Where we now find that what this motif is, hammerhead, is no longer associated with its original function, which would be binding to nucleotides, but now we can find it with functions related to what is binding to cofactors, or even functions that are more of a structural nature, as they could be in these other proteins that we see on the right and left side. Well, evolution in languages also occurs in a similar way. And to do this, we must address an idea that is quite simple to understand, but quite powerful, regarding how there are also some words that become more conserved compared to others. For example, the idea of mother is a fairly shared idea throughout all societies, throughout evolution, because it is quite clear that we are all born from a mother. However, there will be other words that may be more characteristic or unique within all varieties of the language. For example, the word mother. Pairformer, because it really is a word that is within what would be the world of Spanish and more specifically within Mexican culture, and even more specifically within what would be the topic of tongue twisters. So we could expect that there were no homologs of the word Pairformer, but there were homologs of the word mother, which we see precisely reflected here in these lists where we find different languages. For example, Romantic languages, Germanic or Slavic languages, where, for example, we have Spanish, where we use the idea of mom or mother, and we express it through the words mother, mom. Or in the case of Italian, they use words quite similar to Spanish, such as mother or mom. In English, we use mother or mom. And what is the Germanic mutter mama? And what is Ukrainian mati mom? Or even within the same language such as Spanish, we have synonyms to refer to this same idea that something is a mother. In this case, we have mother, mom, mamita, mommy, mamacita, parent, boss, which even under specific contexts, even under periods, both of space and time, because precisely the meaning that each of these words can adopt can be completely different. So we notice that what is the evolution of words or languages in general shares certain similarities with what is the biological evolution of proteins. That in this case, now we see it in more detail with what would be the semi-mythical root. In this case, that is QWN, an idea that is abstract concepts used by linguists who intend to share the idea of Cain, in this case here represented by this scheme on the left. And what we see is that precisely this Semitic root of Cain or rod is precisely being used by different languages throughout time and space, such as some very ancient languages, until we reach what would be the ones that we most commonly recognize, such as the use of Latin or Greek, and that again there is this use of what would be orthology, in such a way that we find precisely the use of these words as cana in Latin or Greek, then cana in French, or can in English, and in this case they are orthologous because they intend to share the idea of a specific cane. However, we can also find these cases where precisely a different meaning is adopted, and then we could consider them paralogs in such a way that the concept of cane becomes a concept that is either much more rigid with these words or a slightly more flexible context with these other sets of words. So we see that there are different similarities in the evolution of both languages and protein sequences. And here we can clearly see some mechanisms when we compare precisely how these two informational entities evolve. For example, we see some common points in purple, such as divergence phenomena in which we cannot trace their last common ancestor, hybridization phenomena. Even evolution is already made of this type of ideas, from what would be the horizontal compartment between multiple languages. However, we also have other mechanisms that seem specific to each of these informational systems, which would make it quite clear what natural selection is in the type of biological systems, or what learning is within linguistic systems. However, it is very important to recognize that proteins are not a language, which you can investigate in more detail in this particular review that I strongly suggest, where it really becomes quite clear that proteins are much more than just a sequence of amino acids. That we have adopted a unisystem of 20 letters to be able to represent macromolecular entities within cells, since it is a utility that we can adopt precisely to understand them better. But perhaps they do not necessarily reflect the totality of what the macromolecule represents within the cell, because we could also have adopted an alphabet at a structural level, which perhaps is not completely clear how we could define, but which when seen as composed of amino acid, seems quite sensible to adopt precisely this type of representations of protein. However, there are different points that really distinguish that proteins are not a language, because language precisely has a purpose and an end. In the particular case, for example, we have that communication is a key aspect because, for example, we cannot speak, write, or read in terms of proteins. We as humans, perhaps the molecular machines that we have inside our cells, well, yes, right. There are also ideas of limits. In this case, when we look at proteins, there are no really clear ideas equivalent to what words would be, etc. What would be their meanings, or even the ideas of being able to have as separators, or what would be punctuation symbols in that sense. We also have that the lengths are really very variable, as we could see with the examples of the nipple that we reviewed previously, which can be a protein of up to 35,000 amino acids, and while there really is no word, at least, as far as I know, that has up to 35,000 words. Perhaps Nahuatl, due to its syntax, can have quite long words. However, not at the level of thousands, as we can find within the world of proteins. At the information level, we also find some statistical properties that are also quite different, such as the size of the vocabulary or the entropy of the symbols that are used to represent both information systems. Obviously, they're completely different understandings. The world of linguistics lies within what is the semantic world, while the world of proteins It really reflects a physical reality that we see as macromolecules that we can crystallize among multiple other aspects. Also, what evolution is precisely is completely different in both worlds because precisely in one mechanisms operate, that in the others do not, and that even the levels, or rather the level of compression between the noise and the signal that are present in these systems, it can be completely different because what language is, since we precisely use words that are intended to communicate ideas, which is relatively clear in this sense. However, when we can analyze what type of information an image intends to share, it turns out that it is now much more complicated to decipher what an image could represent, because if we change the patterns present in each of its pixels, they could mean completely different things. And when we take it to the world of biology, particularly proteins or even DNA sequences, this becomes even more complicated for us in terms of humans. In that sense, we must recognize precisely that proteins are not a natural language, but we can adopt natural language algorithms to be able to precisely model protein sequences due to the similarities that we previously addressed. So the idea is precisely to adopt what the transformers architecture is, in particular in this case, adopting what the BERT-type architectures are, to be able to train this type of protein language models, which have been seen to precisely learn statistical patterns that could be related to biological concepts. So we move on to describe how these types of protein language models work, which is relatively simple. In this case, it is clear that we start from what is a biological database, with millions of protein sequences that we have to process, clean, remove duplicates, partial sequences, among others, to have proteins, which, when seen with a BERT-type architecture, proceed to be masked, as we previously reviewed, and then the model will have to calculate all these query key and values matrices in order to compute the tension. And obviously, when calculating the voltage, the BERT-type architecture is used, which will make predictions of which residues would be most likely to fall into these masked regions. And then, when these predictions are made, we will see that here there are some proteins, such as this one that was correctly predicted, which in this case complies with these same residues that are in the original sequence, which would be GAMA. In this case, it is also GAMA. But in this case, in the second protein, what its sequence would be was poorly predicted. In this case, it is wrong, and in this case, it is BIBA. So because of this, we can now compute a loss function to precisely tell the algorithm where to direct its gradient to precisely correct all the weights of the model and make increasingly better predictions. The idea is that precisely from this type of training, the transformer learns vector representations rich in information of the proteins derived from its sequences, which we know precisely as embeddings, which can be derived from each of the layers of what is the architecture of the transformer, represented here as a simple perceptron, which here we see the first layer, the second layer, or the third layer, which in this case, let's say, we can obtain precisely the embeddings of each of the residues of our sequence of proteins. Speaking precisely of embedding entities for each of the residues, which we can carry out a multitude of operations, in this case simply taking an average, and then go from these representations of embeddings for each of the residues that make up our protein sequence, to going through a single embedding that represents the complete protein, through, as I told you, this idea of taking a kind of average. And now the idea is that precisely these are high-dimensional vectors or embeddings, in which each of the dimensions precisely captures statistical patterns that can be precisely related to biological concepts, such as secondary structure, folding area, or there will even be some dimensions that may have overlapping concepts such as taxonomy, size, and some dimensions that we may not have any idea what they correspond to, but that by capturing precisely what the semantic context of protein sequences is, well, we can use in a posteriori analysis, such as transfer learning approaches, where we adopt these embeddings and now use other prediction or regression algorithms to be able to make classifications of, for example, whether a protein can bind or not to X ligand, if it corresponds to some site within the cell, among other aspects that we can predict. And then we come to the comparison of this type of approaches, based solely on data with respect to classical approaches based on very well-structured biological bases, as we can see precisely in the y-axis, or that have a biological base that is very well thought out and very well structured by its developers, who were really the rock on which we built the entire bioinformatics base with which we have operated. However, thanks to algorithms based on deep learning, we now have approaches that no longer necessarily start from these very well-planned biological bases. We simply show them the data, and it is the algorithms that learn these types of biological bases, or at least their statistical similarities, which even allow us to model the same aspects that we achieve by implementing the biological bases of our algorithms. In that sense, what protein language models are, since they almost do not include any biological basis, other than indicating what they are an alphabet of 20 proteinogenic amino acids. So we can consider that these would be mainly driven by data, and not so much by biological bases. However, we also have other types of algorithms based on deep learning or mainly based on data, such as autoencoders, trained only with one type of sequences from a protein family, which in this case would be seen here, because they do not have as much information as the large protein language models that cover all proteins, not just one family, could be. Or we can also contain models that in this case already inject a greater amount of biological bases, such as the AlphaFold algorithm that we are going to review in detail. And in this case, there's also something known as multimodal learning, which precisely aims to incorporate information from multiple experiments that aim to describe some biological entity, and by being able to combine them through different strategies based on neural networks, to be able to achieve the implementation of algorithms that are completely based on data, but that also have a strong injection of biological bases that can be distilled by incorporating multiple modalities. Specifically, what was the research into protein language models, since it is already quite a few years old? Since approximately 2017, we can trace the first ancestors within this theoretical framework, where we found first works where what they did was investigate the use of what is coevolutionary information to be able to predict structures. Subsequently, we moved on to what would be the training of language models that were quite small in this sense, compared to the ones we currently have using either LSTM networks, autoencoders, or even language models not based on neural networks that only adjusted to a specific protein family, until the first language models appeared at a global level. That what they did is adjust a neural network for all the protein families that we had in the databases, and that proved to be precisely quite interesting results, to be able to capture multiple biological patterns that could be related to the proteins, and thus improve the predictions using these transfer learning approaches. This is like recognizing the works that were ancestors to these two other key works, in my opinion, which were part water precisely for what is the use of protein language models in biological research. We have two works that in this case appeared in relatively similar time windows. One by a meta-team led by a scientist named Alexander Rives, and another led by a collaboration between Google, NVIDIA, and Buckar Drost's lab, who also each trained different language models based on transformers. In the particular case of the Goal team, they trained different transformer architectures, talking about the number of layers they can have, as well as variability in the training set that was used, along with other language models that were not based on the transformer architecture, such as Ingram models or LSTM architectures. And what we observe is that here there is a metric that, in summary, what it shares with us is that the residues in an amino acid sequence are also being predicted, being values equal to 25, equivalent to the number of the alphabet that they used in particular, because they considered about 5 more amino acids within this alphabet of 20 amino acids. Then using this metric and signaling with a number 25 means that it is not being predicted well, and if it is signaling values similar to 1, it would be like the oracle in this sense, which is saying that if the amino acids are being predicted quite well, and then here we observe with this metric that when seeing variability within the number of layers, as well as the number of data used to train the transformer, The best combination that was found was to train a 32-layer transformer with approximately 670 million parameters and with a database derived from the uniprot characteristic, which had a specific metric of 8.46, which I insist, this reflects the fact that the amino acids are also being predicted. And so they called this particular transformer architecture ESM1, because evidently this was the ESM1 family in total, and because this was the modified variant that turned out best, they called it SM1B. In the case of this other publication, which presents an entire family of protein language models based on transformer, they trained different architectures such as the XL variant, the XLNet variant, the BERT variant, Albert, Electra, T5, and then T5 even taken to much larger scales to see what type of transformer architecture could be best adapted to be able to model what protein sequences are, in such a way that a large amount of computing was also involved to be able to train this type of algorithms. And because this really represented a large energy and economic expenditure, it was precisely that this collaboration with NVIDIA and with Google was established to be able to access these enormous computing clusters. And in this sense, the Meta team even further developed the idea by taking what these protein language models are now to language models of protein alignments, with what would be the development of this algorithm known as MSA transformer, where practically what they do is apply what would be this idea of attention, but compute it not only between pairs of residues, but in the alignment compute attention at the level of rows and at the level of columns. Obviously, when computing the tension at the column level, what you are attending to is the information that exists shared across several species, or in this case, several proteins, which in the event that they were very well conserved residues, such as these alanines or these phenylalanines, we could infer that in the alignment perhaps this amino acid fulfills some quite key function, either at a functional level or at a structural level. And in the case of row-level computation, it would be equivalent to computing the tension between pairs of residues of a specific protein sequence. Therefore, it models, let's say, more specifically the behavior for a particular protein, but also considering, then, what its counterparts within the alignment would be when computing the tension at the column level. This idea was very key to what would become the development of AlphaFold, in fact. The idea here is that precisely when we turn to C, in the patterns of attention derived from these types of protein language models, we find that what attention really does is reflect or capture structural patterns that are related to binding sites or even active sites and even residues, that, although they are distal in the sequence in the structure, are in contact, as we could see in this scheme, where we can color the ingredient from blue to red in terms of start red, end blue. This would be amino acid 1. This would be amino acid 150, so to speak. And we found that although they are distal in the sequence, it turns out that they receive more attention to each other because in the three-dimensional structure they are in contact. Then we can say that some patterns of attention, through these statistical relationships that were learned, present certain similarities with what would be the contacts established at the level of the protein structure. Or even here we also see cases where multiple residues turn to look at a single particular residue that turns out to be associated with what would be the binding site of this particular protein. Or we can even precisely map these tension patterns. That could even be related to what would be the idea of complete folding, which in this case we could see in green. The tension pattern that occurs from residue to residue contiguously is reflected, which fairly consistently reflects what the folding pattern of this specific protein would be. And because of these types of observations, it was found that these types of protein language models could actually serve as unsupervised learning algorithms that precisely learned structural patterns of the protein. And this was something that was demonstrated from the analysis of the tension maps of this type of algorithm. Again, we find here a basic scheme of how they work. We find the sequences they mask, and then they are predicted. The matrices chi and chi values are calculated. And then with the chi and the chis, as you know, we compute these tension maps. And then we can use these tension maps. We make them symmetrical because, as you remember, the tension is not symmetrical. And so now we use these attention maps with a logistic regression to predict the contacts that there may be between proteins. so that now we can couple what would be a fine-tuning of this type of language models, so that the predictive capacity of these types of contacts is maximized, and that only by using the attention maps can we reach structural information that reflects quite well what would be the three-dimensional pattern of the protein. An observation that was precisely developed later to be able to apply this type of language models and further refine what is the prediction of three-dimensional structures from only sequences. Then we see this diagram again, which is precisely the success that the transformer's architecture had in multiple areas of research, which in this case we already saw that it had great success in what is computer science, what is language, processing, reinforcement learning. And that, for example, now we can say that they have also had success in protein science with those types of examples that we addressed previously. And so it was that we arrived precisely at the AlphaFold announcement, that in this case we really woke up, particularly on November 30, with this statement issued by the CASB, which in this case, and in a very British way when reading it, it is precisely noted that they share this idea that a solution using artificial intelligence algorithms has been found, and that has a coherency that is quite comparable to what can be obtained through X-ray crystallography experiments, which even they suggested, as an entity of the national organization of this contest, that it represented great potential to revolutionize the life sciences, biology in general, and that we could consider that at least this problem of predicting structures at the monomer level could be solved using this type of algorithms, but really, we only woke up with this statement from both the CASB and a small blog from DeepMind, where they announced that the protein structure prediction problem that already had approximately 50 years of research behind it had been solved, and we were left for a long time without knowing more details about it in such a way that we even reached approximately July 2021. 9 months since this statement, where they finally released what was both the publication and the model on GitHub to be accessed by multiple researchers throughout the Earth, and it still took about 8 more months for the intellectual property of what the AlphaFox models are to be configured so that they would then be considered open source. And then the scientific community could work in a much freer way regarding how to adopt this type of algorithms for multiple purposes in biological research. So it was that then the only thing that was shared within this type of communication was this idea, which presented a model where what was done was to share information between pairs of residues to generate representations rich information from which the structure is generated. So the people who saw this information at the time, it really wasn't very clear to us how this algorithm worked, but we could intuit that they used precisely the transformer architecture due to the success that we saw in its applications in other areas outside of biology, and that precisely because of this type of schemes, which are quite similar to attention maps. It suggested to us that precisely what they did was train a transformer, just a very simple transformer, that could learn at the level of alignments, as was the MSA transformer developed by Meta, and that then we could share what coevolutionary information is by comparing column information or row information, and then enriching this type of information with the type of information derived from the histograms, as well as predictions of the confidence of each of the residues, to arrive at what would be a structure of the protein. However, we were quite wrong about how the algorithm worked, and that really it was just pure speculation in such a way that many of us were just like this meme, since even what the architecture was, because it was launched in a period of pandemic, so many of us were in our pajamas trying to understand how AlphaFold could have achieved this milestone of having solved a problem of more than 50 years of research, which, I insist, was simply launched with a small statement from the CASP organization, from DeepMind, and with a small presentation that did not really provide fine details of how its algorithm worked. And we did not know that until the Nature article was launched on July 12, 2021, and then here we see precisely the entire community behind the AlphaFold team, and in particular what would be the leaders of this project, which would be John Jumper and Demis Hassabis as director of DeepMind in general. And in this case, we must recognize precisely the great importance of this team being interdisciplinary in nature. When we turn to see what their training would be in terms of the doctorate, in the area of the doctorate in which they specialized. And what we see here in this graph is precisely that the number of doctors who are here, which are 35, they have different cultural backgrounds at the doctoral level, right? By quickly asking two agents such as Gemini or Grok to investigate what the doctorate of each of these people is, we can see that in general there are backgrounds within what computer sciences are, backgrounds within the world of biology, physics, chemistry, mathematics, and others who may not have a doctorate but who are quite agile and skilled to be able to work precisely within this research laboratory. So it is very important to recognize that the success of this type of project lies precisely in interdisciplinarity, something that is precisely sought after. And in this publication precisely, it is already presented in detail in a total of 12 pages in its shortened version and 62 supplementary pages in the supplementary material part, a total of 32 algorithms of how the AlphaFolder architecture works precisely, which, as you can imagine, was much more complex than a simple language model trained at the level of protein alignments. In order to learn more about how AlphaFold works, if you want to go deeper than what can be explained here, you can review these resources, such as this practical guide to AlphaFold offered by the European Molecular Biology Laboratory, which is quite easy and intuitive for those who do not have a background in bioinformatics, since it speaks in a fairly understandable way, without so much technical jargon that we usually implement in our languages as bioinformaticians. I also recommend this other series of 3 seminars led by a researcher named Nassim Bawada, which addresses in a fairly technical way how each of the AlphaFold algorithms work, as well as a kind of summary. And I have also compiled a multitude of resources, which can be accessed precisely in the repository associated with this course. And the same on Twitter, because they can usually find information that they share, where it can also be useful to learn this type of algorithms from what would now be the AlphaFold family of models in general. And perhaps among all the courses I could recommend regarding AlphaFold, this would be the best candidate. It is called AlphaFold Decoded, a course created by Kieran Mandel, which is completely free and available on its YouTube channel, where practically what it does is give us an introduction to the world of machine and deep learning, introducing general ideas such as what tensors and transformer architecture are, and subsequently breaking down step by step the main algorithms that allow things such as the creation of the inputs for AlphaFold, how to process the sequences from an evolutionary or structural view, and finally carry out the prediction of the atomic coordinates. So perhaps after this course or this reading, it would be highly recommended if you could attend to this series of videos developed by Kilian, because, at least in my personal opinion, the information he gives us is quite detailed and it is really a very good resource to be able to learn from the algorithm behind AlphaFold. And for us to learn precisely about the AlphaFold algorithm, we must familiarize ourselves with what are the 2 main inputs that are used both in this architecture and in what AlphaFold3 is. So if you understand AlphaFold2, you will surely be able to recognize what AlphaFold3 is. So the first relevant representation is this one known as the MSA representation, or representation of the MSA in Spanish, which in its simplest form we can understand as precisely the collection of homologous sequences that are evidently evolutionarily related to what is our sequence that we intend to model structurally, and that thanks to this alignment of sequences we can compute these correlations between amino acids in such a way that if one amino acid mutates, perhaps the other is necessarily related to mutating in order to maintain patterns related to its structure, to its function, from which we can derive covariance information. This is the easiest way to share it, but in practical terms, really when we talk about the representation of the MSA, we're talking about a rank 3 tensor, which in particular the first dimension is composed of what would be the sequences that we are modeling. In this case, we are modeling a protein from a human, represented by an icon of a human. The second row would be a fish, third a rabbit, fourth a chicken. The second dimension of this tensor is precisely the one corresponding to what the columns would be, or what the residues are of each of the sequences. And then the third dimension, or depth in this case, we will be talking about precisely an embedding of 256 dimensions, which is precisely the dimensionality used to be able to generate this type of representations at the LMSA level. And here it is important to recognize that when we are talking about, for example, each of these cells, we are actually talking precisely about an embedding which would represent the position of the J residue in the I sequence, that is, conditioned on all the information belonging to what the MSA is. So you have to understand that this is a representation that AlphaFold aims to learn in a rich and high-dimensional way. To be able to represent the evolutionary information seen as a tensor, and then this evolutionary representation is learned during the training of many protein families, many sequences and structures, so that each of the properties of the amino acids is precisely related to what would be the uniqueness of the protein that we intend to model, and thus identify which residues would be key both at the level of the protein and of the entire alignment to be able to predict the structure. The second important representation that AlphaFold occupies is this one, which is the pair representation, which again in its simplest form can be understood as this interaction matrix by pairs of residues where there is a spatial relationship between each of the amino acids that constitute our protein. Again, this is the easiest way to understand what this three-dimensional representation would be. But really, when we're talking in practical terms, we're talking about a tensor again of rank 3, which has as dimensions the positions of the residue Y, the position of the residue J, and the dimensionality of the embedding in this case is 128 dimensions. In this case, it is a little simpler to understand what each of the cells means. Again, it is an embedding that represents the relationship between pairs of residues, such as the embedding related to the combination of the residue blue with itself, or the blue residue with what would be the red residue. Here the idea is that we can even explicitly inject information derived from the structures of the proteins. It is done in zip format or in PDB format, which are known as templates within the AlphaFold algorithm. And the most relevant thing is that we can precisely inject this type of information explicitly, such as the distances between beta carbons, the dihedral angles, among other characteristics that can also be derived. But it turns out that the algorithm does not necessarily require this explicit information derived from the structure, but to initialize what would be the AlphaFold framework. It is enough to simply consider what would be the relative positions of the sequence to be able to precisely create this tensor. So, we could even completely rule out the presence of these templates and initialize it only with what would be the positions of the sequence, and then again, as with MSA, what AlphaFold does is learn a representation rich in information and with high dimensionality that allows capturing geometric and spatial aspects of protein structures from this representation by pairs of residues. Also, another thing that is relevant to consider is that, as we know, this is a representation that, although it is in pairs, is not necessarily symmetrical. Here you can see different combinations of what would be the IJ residue relationship or the JI relationship, which are not necessarily the same because each residue shares a specific context, and that during training, although the same relationship is discussed, since it may give relatively similar embeddings in this high-dimensional space, but they will not be explicitly the same due to the particularities of the context of each of the residues. So now we are going to detail what the architecture of AlphaFold2 itself would be, and step by step, we will address each of the main modules within this architecture. Here, as you can see, we start from an input sequence that we want to model its atomic structure. And then what we have to do as a first step is to find homologs at the sequence level using a database search to compute a multiple alignment of sequences, and precisely have the MSA that has the diversity of the proteins. This is the first part, and then this alignment will have to be encoded to be taken to the embedding level, which is precisely this representation of the MSA that will eventually be used by another series of neural networks. As a second step, what we have is precisely what the pairing of the residues is, where it is very simple to understand. Really, the only thing you do is take the positions relative to each of the sequences, and thus you initialize a matrix of pairs, as you can see it here directly, where you compare what is residue number 1 against residue number 1, number 2, number 3, number 4, etc., up to n numbers that your particular proteins have. And as a third part, what is done is a search for these templates in databases, using alignment tools at the sequence level to search, for example, in the Protein Data Bank database, or even using structural similarity searches with algorithms such as FULTSIC to find homologs. Then you already have precisely what alignment is. You already have your pairwise representation, or this initial comparison. And now you also have the information of which structures would be homologous at the sequence level with respect to your protein that you want to represent. So now we go through what is known as the embedding layer. This phase is what is responsible for creating precisely these representations of embeddings that we previously investigated to generate what is the MSA and the paid representation. Later we moved on to what would be the first transformer within the AlphaFold architecture, which is called Evoformer. It is a 48-layer transformer that is made up of 90 million parameters, and that will be in charge of distilling the evolutionary and structural information to enrich both the pairwise representation and the MSA representation, as we can see here, which are its main outputs in this sense. And later, now updating these two representations. What is obtained is precisely a representation that is now much richer in terms of evolutionary and structural information, which in this case will be used by a second transformer which we call the structural module. A transformer that is made up of 8 layers, and that in this case only has 2 million parameters. So as you can see, we could actually consider AlphaFold's architecture to be relatively small compared to giant models like GPT-4 or GPT-5 that have billions of parameters, and in this case, we reach approximately 100 million parameters. However, what happens here in the structural module is that what is the representation by pairs of residues is taken, but only the first row of our MSA representation will be taken because this row is the one that corresponds to what would be our protein in question that we intend to model. As you can see, different homologs are found encoded in this embedding, and what would be the first row will be precisely the protein that we are interested in modeling. Finally, what this structural module will give us as output is precisely the prediction of the atomic coordinates, where AlphaFold also gives us a confidence metric regarding the credibility of the atomic positions of each of our proteins, which is called PLDDT. And that in this case, we could see regions colored in blue as regions with high confidence, and regions colored in orange or red as low confidence. Finally, this three-dimensional structure is generated, which can be used again during several recycling phases of the entire algorithm, where, as you can see, the resulting information, which would be the atomic coordinates, now goes on to update what would be the representation of pairs of residues, as well as what is the representation of the MCA, to run the 48 layers of the Evoformer again, the 8 layers of what is the structural module, and you can repeat this cycle approximately 3 to 5 times. It is most recommended, although it can be extended even more in some specific cases. Something important to recognize in this architecture is that it is known to be differentiable from term to term. What does this mean? Well, the parameters related to the transformer of the Evoformer or the transformer of the structural module will be mutually updated in such a way that, as they are connected from term to term, the optimizations that are required to precisely compute the backpropagation will be considered by the structural module, as well as by what the Evoformer is, even by all the neural networks that allow connection between modules. So it is said that it is from term to term because precisely, although we have modules that are separate and that each one is in charge of a specific activity. In this case, the Evoformer is in charge of creating representations rich in information, and the structural module takes these representations rich in information to make its structural prediction. Both are interconnected in terms of the backpropagation of the gradients, so that they can precisely configure their weights in order to precisely optimize the prediction. So this is what is known as term to term. And something important to recognize is that when we try to know how the AlphaFold architecture works, it can really be quite complex. Understanding how it actually works is a scientific problem in itself, because AlphaFold is a product of computational engineering. So something that we usually find in disclosure practices or criticism of this type of algorithms is precisely that they function as black boxes. But the fact is that the interpretability of the algorithm did not really lie in its conception, but rather the purpose was precisely to make the prediction itself. Understanding how it works is obviously a job that even requires a good theoretical framework, as well as paving a way to find the most correct way to even perform perturbations, how to objectively carry out evaluations that allow us to isolate each of these modules from the architecture, and understand what their contribution is to the algorithm as a whole. So it is not an easy task in this sense, and we must recognize what is precisely a research problem in itself. So with that said, we move on to explore in more detail how this first layer works in general, which is the embedding layer. To generate precisely what is the representation of the MSA and the pairwise representation of residues. Well, as you can see here, the embedding layer is monstrous. It is horrible, don't be scared. It is a little easier to understand when you start to observe what the colors of each of the inputs are. In particular, in the input layer or embedding layer for inputs, in this particular case, up to 19 derived input filters are computed, both what the sequence is and the alignment. So in this case we have two main ones, which, as I mentioned previously, would actually be what the relative position is, or which we can see as a kind of positional encoding as in the transformer, which would be precisely the residue 0, 1, 2, 3, 4, 5, 6, which precisely gives us information on the positionality of each of the residues in our sequence. And the other would be precisely to encode what our sequence is, in the form of a categorical form that is like one hot encoded that will eventually serve to tokenize it, and then later take it to the embedding level. Here what you can see is that even with what these representations of our particular sequence are used, as you can see here in this series of connections in blue, all the information passes through to be able to be precisely injected into what the pairwise representation is and precisely initialize it. As you can see, these arrows indicate what the flow of information is, and between each of these arrows something like linear, 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 linear is indicated, which in all these cases are precisely multi-layer perceptrons that do not necessarily include a nonlinear activation function, but that precisely what they help us is to modify the dimensionality of our embeddings that we are transforming so that they can be coupled to the dimensionality of the next input, and then in general to the network as a whole. So, although we are talking about two main networks of the transformer type, in reality, there are many neural networks in this sense to be able to couple the dimensionality between each of the AlphaBooth modules. As you can see here, there are at least 1, 2, 3, 4, 5, 6, 7, up to even 8 linear networks to be able to precisely deal with the dimensionality of the different information channels. Again then, this is the contribution that is obtained from what is the singular sequence that we intend to model in order to create the pairwise representation. As a second observation here, we have the inputs derived from the MSA, which in this case are tokenized not only as an alphabet of 20 tokens, but also 3 more are included, one for a token that in this case is not known in the alignment, another to indicate a gap, or even to signal what the mask is. Remember that the AlphaFold Evoformer in particular has a behavior similar to what BERT-type transformers would be, which aim to mask to learn information-rich representations. That is, it is not generative, so that is precisely why a masking token is used in this sense. And in this case, what we see again is that two main representations are generated derived from the multiple alignment of sequences. The sequence that we intend to model in this case, which is a human protein, will always be the one found in this representation known as the MCA fit, which is the main one. And in this case, due to memory limitations, what would be the sequences of our alignment are usually shown randomly, up to a maximum of 512 sequences. Then this MCA fit is generated, where precisely it will be occupied, along with what is the information of our specific sequence. To now generate the representation of the MSA, as you can see here. However, if our alignment perhaps has, I don't know, 2,000 sequences, 3,000 or even 10,000 sequences, because of memory limits precisely, we cannot use all of them. So what the DeepMind team developed is another second representation called extra MCA fit, which now, well, it has the remaining proteins that we do not use for this main representation, and which in this case can be even larger, up to a maximum of 5,120 proteins, where a less intensive computation is done, but it is also used precisely to enrich the information of the representations. In this case, this representation called the extra MCA fit is used. It is passed again through a series of linear layers. And then we generate a representation of the MCA, but alternate or extra however you want to say it, which will be processed by a small version of the Evoformer, which in this case has about 4 layers in relation to the 40 or so that the main transformer had. However, this small version of the Evoformer evidently uses heuristics, including its tension modules, to avoid a large computational cost. And what it intends is precisely to refine the pairwise representation with sequences that have not been selected as representative within what would be this first main representation of what the MSA representation is. So as you can see, the two main inputs have already been created, which are precisely the pairwise representation, such as the MSA representation, which in this case even the representation of pairs of residues even goes through this mini Evoformer to be enriched in informational terms, and now it is beginning to be used by what the Evoformer is. And what is the representation of the MSA passes almost directly to the Evoformer, as you can see. And here is precisely what I was telling you, that the information derived from the structural templates is not necessarily required to be able to initialize this type of inputs. As you can see here, in order to create them, at no time is information explicitly derived from protein structures used. But it can be added, indicated by these plus or concatenation symbols, that in this case, for example, derived from the three-dimensional structures of our proteins, information is computed such as the IV angles or information on what would be the pairs of residues at the level of their distances between beta carbons. Again, each one is passed through different neural networks to be able to generate the embeddings and couple them to the required dimensions. And in this case, what is the pairwise representation is added, and they are even added to what is the MSA representation in order to be able to inject this structural information into our main two representations. And as you can see precisely, if we eliminate what this information is, since it does not disturb what the two main representations are because its computation is independent of the structures, and it has even been observed that when the sequence alignment that we have is rich in sequences and rich in sequence diversity, what AlphaFold does is tend to ignore the information that derives from what the three-dimensional structures are, since it mainly has a more information-oriented approach than MSA representation. In this case, in AlphaFold2, what we could adopt as the most important representation within the entire framework is precisely the representation of the MSA. So well, now you understand that with what is mainly the sequence that we want to model together with what is the alignment, the two main inputs are generated, the pairwise representation and the MSA representation, and they are not necessarily required in the template information, but they will be of great help to precisely improve the predictions. And here again, discussing a little what the importance of the MSA is for what the AlphaFold algorithm is precisely. As you can see, in order to create this type of alignment, we can start from our input sequence. It is searched in databases with algorithms such as Jackhammer or MMSX, and then we will have a series of homologs regarding what our query sequence would be. And this is precisely how we can observe this type of alignment. At the top, you will find the sequence that we intend to model, and in each of the positions on the x-axis, each of the residues within our protein, and on the y-axis, the number of homologous sequences that were found in the databases. What factors determine precisely how many counterparts we will find in the databases? Well, precisely, for example, the type of folding. Perhaps we're talking about a superfold, and then there are many homologous sequences deposited in sequence databases. Or perhaps we're talking about these folds that are relatively less abundant, that perhaps it is more difficult to find them in the databases. So being able to model a protein will also depend a lot on what the folding itself is. Rare folds are going to be more difficult to model, and relatively common folds like the 10-barrel are going to be much easier to model. And obviously, this will also depend on the database to which you are making the queries, because if you take a database like the one presented in the NCBI, which is a database called NT, in this case NT, it will have 500 million proteins. But perhaps if you take databases that are more curated, that integrate even more information from multiple metagenomic projects, then your database will go from 500 million proteins to perhaps 2 billion protein. So then you could find many more homologs that can precisely recapitulate the structural information of your particular protein. So look at other databases or even curated databases, for example, that were specifically for viruses, could be a good strategy in the event that their protein was more challenging to be predicted with AlphaFold in this sense. And what we also observe is a color gradient from blue to red. In this case, the proteins most similar in terms of sequence to our query protein are colored from blue to greenish to yellowish if they already have approximately 50% sequence similarity. And that there will even be homologs with very little sequence similarity, and even more fragmented to only specific regions of the proteins, which will precisely have a color ranging from orange to reddish in this case. For example, we can see these fragments present a certain level of homology to have been integrated into our alignment, but they only correspond to the regions between the residues approximately 50%. To approximately 80, and then we can see what the average coverage of all these sequences would be in such a way that we can notice that what would be the first 25 residues of this protein are not as well covered in relation to what the other positions would be, such as positions 100 to 200, which have up to 6,000 sequences, right? In this case, it is an ideal alignment. This alignment is super nice because it has, as you can see, up to 7,000 homologs, which is quite a bit of information, and we can observe this gradient from blue to red, and there is no predominance of sequences that are highly similar to what our query protein would be. So we can say that this is a good alignment, despite the fact that these first positions of our protein, let's say that there is not much information regarding this specific protein. There will be other proteins that may be even less represented, and we can find not 7,000, but only a few dozen homologs in the databases, which is why it will be relatively more difficult to be modeled by AlphaFold. And when we inspect precisely what the importance of alignment is in AlphaFold's prediction capabilities, we see this type of graph, where what we have is on the x-axis a measure of the number of sequences is called the number of effective alignment sequences, which is a parameter that measures both the quantity and the diversity of the sequence, and that we can see is counted within what would be 1, 2, 3 until reaching 10, 100, 1,000 and 10,000 sequences. And in this case, we see on the y-axis that this structure is also being predicted using the predictions of the structure similarity metric called LDDT. Therefore, values usually close to 100 suggest that the protein is being predicted better, and PLDDT values of approximately 50 are suggestive of poor prediction. And so now we see two sets of proteins, one in orange and one in blue. The set in blue represents proteins that have precisely these structural templates deposited in the PDB, for which information can be found, of less than 30% of the entire structure. That is, there is not as much structural information in the databases. And the orange set represents proteins that have precisely these structural templates, but in this case, they cover up to more than 60% of the structure of our protein. So we can say that they are then relatively well represented in databases such as the PDB, and tentatively also in databases at the sequence level. What we observe is that precisely because the orange set is more abundant in information, its predictions, even with a few sequences, like even a few dozen up to 100, are relatively good, right? They start from almost 80% PLDDT until reaching 90-odd% PLDDT when there are up to 10,000 sequences. And that for proteins that have less structural and sequence information. Well, it's a little more challenging. However, we see that at a behavior of approximately 95 sequences, both protein classifications are being modeled quite well. So we could think that if our alignment has diverse sequences, and these are approximately 100 to 120, we could say that it is a good alignment with which we could think that AlphaFold will be able to make good predictions at the level of its atomic structure. This is a pattern that has been seen in multiple independent investigations. For example, here what we are observing is the proteome of a family of viruses called Flaviridae, and in this case, what we specifically observe are approximately 32,000 or almost 33,000 structural predictions for the proteome of this family. Again, in this case again, we mark here with a line of 100, which would be the optimal one to be able to find good predictions. And although here the authors decided to color black and not much information is offered regarding what the density or abundance of each of the proteins would be, we could assume or approximate that precisely this region that is marked in red would be the densest, the most populated by the type of AlphaFold predictions, which precisely correspond to predictions from approximately 75 to 100% PLDDT. So then again, if you have an alignment of up to 100 diverse sequences as well, you could think that in this case it is a good alignment and that otherwise, if you had a very poor alignment of just a few, about 20 or even 30 proteins, it would be a good strategy to consult other databases perhaps. Other interesting behaviors regarding what would be the use of the number of homologs we can carry out from an algorithm similar to Evoformer, which in this case is called MSA Pairformer, and in this case only the behavior of the MSA Pairformer is being evaluated, not coupled to a structural module, but only using the embeddings of the MSA Pairformer to be able to perform different modeling tasks, which in this case could be precisely the modeling of long-distance contacts, and in this case what we observe is that when we have, for example, alignments of approximately 32 sequences, these long-distance contacts are not really predicted so well, but when we have an element of approximately 128, then we can say that if it is predicting relatively well what the contacts would be. Long distance in several proteins, in particular 100 proteins in total, taken from this training dataset of an algorithm called TRNOZ. And we see that there is even a behavior similar to a plateau, where it seems that 256 or even 512 sequences no longer represent an explicit improvement in this sense. So again, the suggestion here is that if you have an alignment of about 100 to 120 homologous proteins, which are also diverse in terms of sequence, well, it may be a good alignment in this sense, as you can see from these predictions at the contact level. And this is a behavior that is also observed in terms of predicting interactions between proteins, which in this case, again, we observe on the y-axis, where these interactions between pairs of proteins are also being predicted, and and in this case up to 256, there is a slight improvement, but not so marked in that sense. And again, recommend that these types of predictions only derive from the embeddings of what the MSA performer is, not in the case, for example, of the AlphaFold case, where what is the Evoformer is coupled together with the structural module and other series of algorithms to be able to make the structural predictions. So even here, what we are explicitly evaluating is the behavior of an Evoformer-like module in order to understand the effect of the depth of our alignment. In particular, we can also recognize different ways of how we can run the AlphaFold algorithm. The default and perhaps the most recommended way is precisely to give our sequence as input, which will be a necessary input in all cases. We can also give it what is alignment and inject explicit information from the templates in the event that homologs are found in databases such as the Protein Data Bank. Another second way of running the algorithm is by eliminating the use of templates, which, as you can see, is not required in the sense to be able to initialize the MSA representations or the representations between pairs of residues. But we are missing precisely this type of explicit information derived from the structures. Another way to run it is without the alignment, which is not so recommended, but to run it with the information from the templates. These applications are not really used that much. They are used more for research into how the interpretability and explainability of AlphaFold operates, but it would not really be recommended. And there is another option that is also widely used in the case of de novo protein designs, which in this case is known as single sequence. It is also usually shared in a relatively similar way by other algorithms that also protect structures, such as the SM-fold or omega-fold algorithm, which in this case do not use alignments, nor do they use information from the templates in, that only explicitly take what the input protein is. So when you use this, what you do is evaluate the AlphaFold capabilities in a more strict way to see if this protein could really be a protein that can fold three-dimensionally and look at what its confidence prediction values would be in each of the residues, using this metric known as PLDDT. So usually when working with proteins again, these types of runs are also evaluated with AlphaFold to precisely assess whether it could be an interesting protein to be explored in terms of experiments in the molecular biology laboratory, and not only bioinformatics. Even training diversity can also be used to model different states within what would be this conformational heterogeneity that represents the assembly of our protein. Well, remembering, proteins are actually conformational assemblies, and so it has been seen that if we make different modifications to what the alignment is, for example, dividing it into a specific number of proteins, we only take the upper top 100 or the medium top or the lower top, masking what would be some residues that may be key or not so key within what the alignment is. Or even clustering the isolation, generate modifications of the type of information that is being injected into the AlphaFold algorithm, so that then, by modifying precisely the information that is contained within the MSA, it can now be related to what would be the prediction of the conformation of the protein from a state 1 to state 2, right? Remembering what these states would be like, for example, as open or closed, which we previously explored in the protein structure and protein function class, that in the case of being in an open state, we could find precisely what the most exposed active site is, and then you can more easily identify that perhaps some specific ligand is docking there. So this has been something that is constantly being researched, and there is a lot of discussion as to whether, for example, AlphaFold can really be useful in being able to model conformational assemblies of proteins. However, it is not a reason for discussion here. However, I present to you what this idea is, and that you can even explore it in case you are interested in precisely modeling conformational assemblies using AlphaFold, which, at least from my personal opinion, is quite useful in that sense. We also found behaviors related to MSA that are quite particular. For example, in this quite relevant post that we will discuss even later, they take on the task of cataloging what would be the 214 million structures predicted by AlphaFold that are available in their database. And when we looked at these 214 million structures, it was found that 39 million of them are actually sequence duplicates. Therefore, if they are sequence duplicates, one might expect their structural predictions to be quite similar. Be careful, not identical. Quite similar because obviously there is randomness in what the AlphaFold algorithm is, and so you will not necessarily obtain the same output explicitly every time you run it, but it will be quite consistent. However, it turned out that, although we are talking about duplicate sequences, it turns out that their structural predictions are completely different. And in this case, they found divergences at the level of structural values, such as the MRCD, which are already reflected in the structural predictions, in modifications, in what is the packing of domains, which would be the conformation of the folds, or other metrics more related to the fact that the dimensionality of these proteins is also coupled, in such a way that here we can observe two proteins that are duplicated, that have an RMSD of 64 Armstrongs, and as you can see, one is relatively blue in color, even redder blue. And here below it is predicted with lower reliability in several regions, and in addition, its three-dimensional configuration is completely different, which is why it yields these types of very different values. And so the authors suggest that perhaps one of the possible reasons why AlphaFold thus modeled two proteins that are duplicates is precisely due to the difference in the information contained in the multiple sequence alignment. And due to this publication, along with others, the AlphaFold database now integrates within the files that can be downloaded to be analyzed, also the alignment information, so that you consider it when you are reviewing. And in this case, it even indicates the version of the prediction that is being offered within the AlphaFold database, which in this case is version 4, but AlphaFold constantly. When users report that there are problems, then the task of make new predictions to see if they improved using more information, detailing more aspects, and then this version number is updated from 4, until it even seems to me that right now we are at version 6 in general. And well, now we move on to explore what the Evoformer is in particular, remembering that this is a transformer which is made up of 48 layers in total, and which represents approximately 90 million parameters. Evoformer, as you can see, is this module where what is done is to use the representation of the MSA together with the representation by pairs of residues in order to precisely move on to an update that is now much richer in information of both representations, as you can see. Now they go through two main channels, one which is the channel of what is the representation of the MSA and the channel of the representation of pairs of residues. Something relevant that happens inside Evoformer is that there is an intercommunication between both types of representations, such that the representation of the MSA influences the behavior of the representation of pairs of residues, and the representation of pairs of residues also influences what the representation of the MSA is. The information is injected so that both they are in constant communication and feed each other, which turned out to be a pretty good idea to be able to precisely distill the evolutionary, geometric, and spatial information of the proteins. In this case, what we observe in the case of the MSA channel is that the pairwise representation is injected, and then what is called row-level stress and column-level stress occurs. Then you go through a simple neural network of the two-layer perceptron type, and in this case, what it does again is precisely couple the dimensionality of what the output is in order to be able to give it to what is now the channel of the pairwise representation. And finally reach what is the representation of the MSA, and something relevant is that here, as you can see, These blue arrows are the escape connections that precisely what they help is to preserve the original information, and that the signal of the base input from which it is based is not lost. Here again, we also see other skip connections, which precisely prevent this information from disappearing, but also update it constantly throughout the algorithm. And then, in what is the channel of the pairwise representation of residues, what we observe is that two tension mechanisms also occur. In this case, what happens here is a tension that is triangular, and then here's something else that is an equal triangular but multiplicative update that we are going to explore later. But here right now, as we are going to focus on what the representation of the MSA is, it is relevant to know what type of tension occurs in these two modules. The purpose again is that we arrive at these two representations of residue pairs and the MSA. And in this case, we are going to take precisely what is the first row that corresponds to our CO2 protein that we want to model in order to make the structural prediction. But precisely what happens here are these two attention mechanisms, a tension at the row level and a tension at the column level. And in this case again, we can think precisely of the MSA as a way of defining a probability distribution that precisely what it does is recapitulate all the covariance information between pairs of residues as well, but now seen from the alignment with information from several homologs, which allows us to identify, for example, which amino acids are precisely the most important to be able to make structural predictions through the calculation of these two tension variants. This is exactly how the tension is computed at the column level, which in this case, as you can guess, by computing the tension between pairs of residues of the same protein, what is helping us is to identify those residues that may be more important in terms of their locality, as well as contacts or relationships at longer scales that are distal in the sequence, are perhaps being quite neighboring or in fairly close contact within the protein, three-dimensionally. So here again what we use is what the MCA representation is. And again, we find these linear layers to be able to adjust what would be the dimensionality of the embeddings. And in this module we find precisely what the classic attention value is, which in this case again we find what the queries, the keys are, also here the values, and precisely the classic submax function of any transformer is applied to be able to generate the attention values. And in this case we also see information, a derived channel, where the representation of the MSA is also taken for each of the rows of our alignment, which in this case is simply passed through a linear layer along with a sigmoid activation in order to model other types of nonlinear information that are also enriched within this type representations. And the result is precisely passed through another linear layer to precisely update the embedding state of the particular row in question. The thing here is that precisely we can use the information from the pairwise representation and inject it into what would be the tension computation, as you can see. The representation is taken in pairs. Again, neural networks are applied to be able to handle the dimensionality and make it compatible. And then you can inject precisely this information, and thus enrich it even more now with the information pertaining to what would be the structure of the proteins. Not only at the level of homology, but now at a precisely structural level. And this is precisely the part where there is an intercommunication between what would be the representation of the MSA together with what is the representation of the pairs of residues. The other variant of attention is precisely attention at the column level, which in this case, because by investigating what the positions of each of the residues of our alignment would be, what allows us to identify the coevolutionary information right. For example, see if there is an amino acid that is more conserved in the entire alignment because perhaps, for example, it is related to its catalytic activity, some binding site, or, for example, it is part of the hydrophobic core of the protein so that it can fold. And in this case, we see that there is not such rich feedback. In this case, there is simply the representation of the MSA where, well, again the query, the keys, the values are computed, and again, the same technique is used to model nonlinearly together with a sigmoid activation and inject it into a final representation, and now update the position of each of the residues of our alignment. So we could think that one of the most relevant parts could be precisely this, the computation of the tension by rows, because we can precisely inject the structural information. Although we also do not have to design what the convolutional information that we can find would be, whether, for example, in foods with 7,000 sequences, which could be a lot of information, or foods with a few dozen proteins, which in this case perhaps would not be much information, which can be distilled by computing the tension. In this case, it is a specific one. And here it is important to know that precisely the composition of the MSA is crucial, precisely to be able to make good predictions, because what is alignment must contain sufficient information, but it must not be precisely biased towards any specific point. It should not be over or over-represented in what would be different branches at a phylogenetic level. And this is precisely what we see on this map. In particular, here we are talking again about the favorite proteins of this course, beta-lactamases, which are proteins, quite represented in what are the databases. And then we find that when doing this type of sequence homology searches, we find alignments of up to thousands of sequences, in this case 7,000 sequences, even little more. And if we make a phylogenetic tree with these proteins, we will find patterns like this, where they can be observed as different clades within this phylogenetic tree. It turns out that when we see how it is reflected at the level of its structures, we find some variations that could be relatively relevant. In this case, all these sequences marked in blue would correspond to what we call class A beta-lactamases, but which now also belong to a subclass called subclass A1. And as you can see, here is the active oil. Several regions of alpha helices are the same. Here there are some folded betas and then other alpha helices. So we could think that it is a sandwich-type folding, and that in this case the class, a beta-lactamases but of the A2 subclass, represent some structural variations that are subtle when we compare them with what is the A1 subclass. As you can see here in orange, there is the presence of some folded betas, which in this case are not present in the structures of the beta-lactamases of subclass A1. And in the same way, we find a different arrangement of the loops that are around the active site, which is quite relevant because we now know that the loops can serve as a type of gate with their flexibility to access the active site. So it is then relevant to recognize precisely their contribution to what would be the catalytic or structural activity of each of the enzymes. And when seen with different configurations, we could hypothesize that they could have different behaviors at the substrate levels, catalytic efficiency, among other aspects related to the behavior of these enzymes. And we see precisely that this entire clade that is on this other side would correspond precisely to beta-lactamases of subclass A2. So if you are predicting, for example, a structure of subclass A1 beta-lactamases, well, it would be desirable to have an alignment like this, which is balanced, as you can see. There is not really a big bias towards pure blue sequences, or pure yellow sequences, or pure red sequences. And it's pretty uniform. We see a gradient of information that suggests to us that it is information-rich in general terms. However, for other proteins, this may not be the case, and we have uglier alignments like these. For example, in this case, when searching for this particular protein in the databases, it turned out that there are not that many homologs. There are only about 150 to 200 homologs, and most of them turn out to be relatively similar to our query sequence, as you can see in this gradient going from greenish to blue, and that there is a section of approximately 100 proteins, which in this case present a low sequence similarity. So we can think that this would be an element that, although informative, is more difficult to model because here it is biased by mainly two coloration patterns that we can observe, and this would also be another example of an element that is now not only complicated, but I would say perhaps of poor quality because here, as we can see, the average coverage behavior is relatively well represented, and here in this case drops quite a bit, to even only about 10 sequences for all these regions, from residues 300 to residue 600. And we observe that there is only this strip that is relatively well covered with approximately 70 homologs, which we now know is a lower cutoff point than what would be the recommendation of an element of 100 sequences, and that it turns out that the vast majority of all of these share a sequence similarity of less than 30% sequence identity. Therefore, we could think that the prediction derived from this type of training will be of poor quality, among other aspects. So now you can also recognize precisely the quality of the training that you are building and precisely be able to modify this type of information in the event that you are trying to model more specific behaviors related to a protein of interest to you. Well then, we already reviewed what the computation of stress is at the row level. We also reviewed the computation of stress at the column level and how precisely the information is explicitly injected by pairs of residues. And now we proceed to see how this part of the triangular tension calculation operates. Something that could be relatively complicated to understand, but is actually a bit simple in this sense. What the triangular tension actually does is precisely update the positions of each of the cells, but turning to see the relationships not only with another pair of residues, but a third pair is used, that is, a triplet in general, to be able to update the position of a residue in particular. And in this case, use is made of this triangular tension, because here is a way that we can inject spatial and geometric reasoning. Using a trigonometric mathematical property called the inequality of the triangle, which in this case is a mathematical approach, which suggests to us that the sum of two angles of the triangle, the sum of two sides of the triangle, cannot be greater than the other side of our triangle. In this case, what we observe is that being able to use this inequality of the triangle, it is the simplest way that we can inject into our algorithms a capacity of, let's call it that, logic, spatiality, geometric, because, as you recognize, triangles are the simplest polygon that we have, and that even on this type of polygons, terribly complex areas of mathematics are built, terribly useful in practical terms, which would be trigonometry, and thanks to triangles we can practically model large number of polyhedra. And in that sense, by using triplets of residuals, we can inject this type of information related to what would be the inequality of the triangle. That in particular, as we can see, by updating what would be the relationships between pairs of residues and considering a third pair, then we now see ourselves a little more limited with respect to the properties that can precisely be predicted in that sense, in such a way that if we know, let's say, the distance between I to j, which in this case would be side, which is a relatively relevant factor within what would be providing AlphaFold with spatial and geometric reasoning to better understand the structures. This can be seen in a mathematical way, as we just saw here, which is not so intuitive, so we can already see it explicitly in our protein. How literal it would be to think that, in our protein, we can draw precisely these triangles between pairs of residues. But in this case they are no longer pairs of residues, but rather triplets, so that to update the information of a specific residue, all possible triplets of residues must be computed, which can help us precisely to ensure a certain consistency at the geometric level of the predictions that are made at the level of the distances of predicted atoms between each one. And this really represents a huge computational cost, because it scales in a cubic way, such that even if we, for example, have a protein with 100 residues in general, this implies that we would need to make a calculation of approximately 1 million, well precisely, triplets, which is when comparing all the possible combinations of triplets with respect to a residue, as well as with the other pair. So then the computation of the triangular tension really represents a high computational cost within the AlphaFold algorithm, but it is also relatively relevant in its behavior. And as I suggested, what we can understand in a more intuitive way is precisely to look at our protein structure and draw these types of triangles along all the possible triplets of residues to update each of their positions. And by using this type of triangles together with the inequality of the triangle, precisely what we do is provide the algorithm with a certain geometric and spatial logic to be able to update the information of each of the positions of the residues. Specifically, as you may have observed, there were two modules of triangular attention, which in this case we calculated precisely from the pairwise representation. But in practical terms, the DeepMind team decided to solve this type of problem with a perspective more of graphs, of networks, that in this case we can see these positions within our pairwise representation equivalent to what this specific graph would be. And then the algorithm in this case was solved precisely with the use of graphs. In particular, they encounter two types of inequalities of the triangle, which in this case was a module related to what the calculation of tension itself is at a triangular level, and another module that what it does is simply a multiplication of matrices that does not necessarily imply this calculation of tension, which makes it much more economical to compute. But it helps us precisely to distill the most relevant information so that it can be passed to what would be the triangular attention module. So that then we have, even within these two triangular attention approaches, two ways of computing what each of these approaches would be precisely, either the multiplicative approach or the attention approach itself explicitly, which in this case, the simple way to understand it is, update the positions using the information in the rows of our pairwise representation, or using the information in the columns of our pairwise representation. Something that is also repeated when the calculation of the tension is done explicitly, only now it would be put in a way equivalent to what its representation with the graph would be. So that in the graph what we have are precisely nodes, such as this node I, the node J, the node K. And in this case, we have precisely relations that arise from this node, as you can see, the relation from I to J or the relation from I to K. In such a way that even when we see precisely the residue I, there are also relations that now enter our node, such as the relation from K to I or the relation from J to I. Something that is evidently also reflected in what would practically be the computation either at the row level in this case, or at the column level in this case, when you can observe in a little more detail how precisely the pairwise representation matrix behaves. This is precisely what happens during this calculation of the triangular tension, and as you can now recognize, it precisely provides the algorithm with a geometric logic that helps us better position the three-dimensionality of each of the residues with respect to the other in what would be the prediction of our structure. What this commutation of triangular tension is has also been explored by other types of algorithms similar to AlphaFold, in this case a variant called OmegaFold, which in this case makes use of several heuristics, including also a relatively simplified version of what triangular tension is, which in this case we see evaluated with respect to its importance. In this case, what we observe is on the x-axis. In this case, an Evoformer is not used. In this case, the authors called their Evoformer geoformer because it has other variants, but they precisely study what the attribution or importance of triangular tension is. So what we observe here is a value that means that contacts at various distances are also being predicted, as could be contacts at a close distance, at a medium distance, and at a far distance, each represented by different colors as we can see, along with what would be precisely violations of this inequality of the triangle, which in this case is represented by this red line. And what we see is precisely the behavior that is observed throughout the different layers of this geoformer, which also has approximately 50 layers, as would be the Evoformer. And in this case we see that as the number of violations of the triangle inequality is reduced, The predictions of the contacts at different ranges of distance are precisely improved, so it is then suggested that the algorithm is being able to distill this information from the geometric logic behind the proteins, something that, when we observe the contact maps derived from each of the layers, that we can observe from what would be the analysis of their tension maps. We observe precisely a better definition of what would be the regions of secondary structures or even increasingly complex contacts in such a way that, for example, here we practically notice a fairly simple pattern of some contiguous residues that even improves to what would be a pattern, a little more related to what would be a folding pattern, since it is a little more understandable, structured, let's call it that. Another team has also taken on the task of investigating the contribution of this type of triangle inequality mechanism, so that when computing this triangular tension, what is done is to inject precisely geometric information, so that it is then a kind of explicit inductive bias implemented within the AlphaFold architecture to improve its predictions. And in this case, what the authors do is, well, modify the triangular attention mechanism in such a way that instead of computing the triangular attention between all the possible pairs of residues, as we can see by these yellow lines, now they are only computed for a much smaller subset. So better information is no longer being extracted from the geometric logic behind the structure of the protein, but now it is a slightly more fragmented version. We are going to call it that, and it turns out that when doing this type of analysis, it can be said that the contribution of triangular tension is what helps us to identify which positions of residues can be in a direct contact relationship with respect to those that are in an indirect way, represented here with this scheme, where we can say that the residue I and K are in a direct form of contact because they are three-dimensionally contiguous in space, but the direct relationships, such as the residue J. In this case, I and J are not directly related in structural terms since there is no explicit contact between both residues, but since J is related to K, there is a contact. So precisely what triangular attention helps us is to identify these types of direct and indirect interactions, and obviously identify which pairs of residues are most relevant to be able to make structure predictions. Well, then now we move on to investigate what the Evoformer would be and its two main tension components, which would be the tension by rows and columns, as well as the triangular tension. So now in this case, we proceed to investigate how the structural module of AlphaFuzz operates. First, we are going to start from a simplified and reductionist scenario of how to carry out the structural prediction of proteins. In this case, we recognize that each of the amino acids that make up the protein, it is actually composed of multiple atoms, and each of these has three-dimensional coordinates on the XYZ axis. So why don't we just take these coordinates and predict them without needing to generate representations based on multiple sequence alignments, as we saw just now with the Evoformer in the case of AlphaFold2? Well, it turns out that when we work with proteins or molecules in general, one of their properties is that they do not precisely have a single orientation in space, but rather their three-dimensional behavior precisely makes them quite characteristic with respect to the effect they can have according to said three-dimensional configuration. So in order to deal with this problem, what the structural module does is use something called equivalence, and this property called equivalence, it is implemented within the second transformer that constitutes the AlphaFold2 architecture. In order to understand what equivalence is in a little more detail, we must first understand what Euclidean symmetries are, or transformations that a rigid body can have in a three-dimensional space. In this case, we have here a molecule represented by this hand, which in this case has at least three types of transformations. In this case, the rotation is quite clear. We are simply rotating in some plane with a specific angle. What would be translation? Moving it from one position to another. And then when we talk about reflection, we have two characteristic behaviors. One is what type of image is projected when the image is precisely inverted when viewed on another plane. And the second reflective behavior is to assess what the mirror image is, as we previously reviewed, related to what the L or D enantiomers are, and the general virality of the proteins. So we then, starting from what these Euclidean symmetries are, we can form what are known as symmetry groups, which are characterized by what type of these transformations remain invariant in the system. In the case there is a first group that is known as Euclidean. In this case the three properties are left invariant. But for us to be able to model the behavior of proteins, it turns out that there is another second group that is called Euclidean group 3 special, which in this case serves us because it does not leave invariant what are the reflection properties, and that precisely due to the chiral behavior that can occur in amino acids. Well, we can make use of precisely this type of symmetry group to be able to train structural prediction algorithms that consider this type of transformations in space, except for what would be reflections. So now we can understand a little better what equivalence would be, which itself is really have some consistency or determinism in the way that if I modify an input, then the output has to respond in the same way. In this case, equivalence as such is, properly speaking, if I apply an input, a transformation to the input, then the output must correspond in a consistent way to this transformation. And then we move on to another property, which is invariance, which really in plain terms is also a special case of equivalence, where in this case there is no change. But as we might think, this would ultimately be a consistent or deterministic response because there is no change. So for example, when we seek to integrate this type of forms of modeling within the architecture of neural networks, we are interested in the fact that precisely by applying transformations the behavior is equivalent. But in other cases, we are also interested in the fact that some properties are not invariant. For example, it is quite clear that proteins do not really change their identity or their thermostability in space, simply by having rotated or moved from one position within the cell to another in general terms. So we can say that what their biochemical properties are, because they are invariant. But also when we are working with transformers in particular, and obviously, we want to analyze this type of structures with this type of architecture. We are interested in the attention values that are being calculated between the residues to be invariant in three-dimensional terms, and that not because we have rotated the protein approximately 27 degrees, 48 degrees, 95 degrees. Now the tension values between two residues are modified only by this transformation. So here are two examples of how you can apply what is either equivalence or invariance, which remember is a special case of what is equivalence itself. And the last behavior that it can have is precisely that a response is non-equivalent, which in this case what it is when some input is applied, in this case well. It is not going to behave in a deterministic way, but it can be even more random, which could be really chaotic for our purposes. So here right now, we are going to explore what is another case, so that the perspective that we have to understand to be clear about what equivalence is in general is even clearer. So for example, in this case what we are observing is precisely a protein. We're first to be able to characterize both the structure and function in three-dimensional terms. We can observe what are some structural motifs, which in this case are marked as blue spheres. An analogy to this would be like identifying in this caricature of a cat what would be the regions, or the proportion that we could associate with what would be the cat's mouth, which in this case would reflect what is a structural motif. By analyzing what this structure would be, we are able to identify that three-dimensional arrangement that we can associate with some catalytic capacity or structural property of the protein. And for this, we have to be able to identify this configuration regardless of its position or orientation in the protein. That is, for us to use algorithms, these types of algorithms must precisely be able to deal with the rotation and translation of proteins, for example in chimerics. To be able to identify some catalytic triad, because perhaps we are working with an enzyme, Or we are perhaps analyzing what would be some hydrogen bonds that stabilize the structure of the protein quite well. So, first it is clear that we have to be able to identify what our structural motifs would be. However, it turns out that this is not enough. Well, now, in order to analyze it in more detail, it is evident that we have to give a somewhat more relativistic perspective, where now there is not only one structural reason involved, but there can be multiple structural reasons, or even what would be its immediate neighborhood in this sense. So now we have to analyze the relative information of both the orientation and the position of more than one structural motif. In this case, using what would be the analogy of the cat, well, we could think about now identifying what the region of its mouth is, such as the region associated with what would be the nose, or even expand our frame of reference even further and contemplate what would be all the attributes of the cat's face, such as the ears, eyes, whiskers, nose, among many other things that we can think of, which in this case we are now making it more relativistic by considering more than one structural group. However, as they will be able to think about equivalence because we also have to think about it within less than global invariance because, for example, when we make a prediction of a model of its three-dimensional structure, in this case, what matters to us is that the quality with which it is predicted is really invariant to whether we are rotating the protein in such a way that if then our protein has a high level of confidence, well, this is not modified because we move or rotate our protein, which would not be sensible to expect. So here we have several examples of how we can think of both equivalence and invariance in terms of what the structures of proteins would be and their consequent behaviors in both structure and function due to different structural motifs. And well, this is clear that it was intended to be integrated into the transformer architecture. However, when we look at the architecture, it turns out that we cannot really observe that there are some calculations, operations, that are really injecting us with these types of geometric inductive biases so that we can understand these types of three-dimensional aspects of these types of bodies. As you can see, really the basic architecture of the transformer, or original, it is simply from what the embeddings are to calculate what the query keys values matrices and their corresponding vectors are, and apply the classic tension operation by making the submax between what the queries and the keys are and then make the scalar product together with what the vector of the values is. So at that time, it was also very famous and very popular among many research community. What is known as a variant of the transformer that precisely integrates a particular type of Euclidean transformations within all of its operations. Something that is generally known as geometric deep learning, which precisely what it does is explicitly incorporate aspects related to the structure or symmetry of the bodies that are intended to be modeled. That in the case that we are working with molecules, well, it could be quite sensible to include aspects, as we can see in this network, that, for example, we could think is representing some type of small molecule in this sense, or even perhaps a small region of a protein where the residues are in contact. And then, in addition to providing each of these residues with some type of importance or centrality, for example, here the node with variable 1 would be larger because perhaps it has greater importance because perhaps it is a catalytic residue with respect to what would be the node called variable 5. We can provide each node precisely with some degree of centrality or importance arbitrarily. We can also encode what the relationships between each node would be. For example, in what direction, how long the bonds are, what type of bond it is, if for example it is covalent, non-covalent hydrogen bond. And we can include practically any type of interactions that exist between residues or between atoms with this type of coding, using a type of special functions called spherical harmonics, which precisely take more caution when dealing with three-dimensional structures, whether they are molecules or proteins as is our case. And precisely by decoding this type of information, then we can now create the corresponding matrices, which we can add to this entire mechanism of computing tension in such a way that when we know that they are calculating the scalar product between what are the Queerleys and Quies matrices, we can also add to another more scalar products that are precisely encoding these notions of ranges of distances in terms of Armstrongs between pairs of residues or the type of chemical bond. So that precisely this type of three-dimensional behavior is explicitly modeled. At that time, many were betting that AlphaFold was using precisely this variant of the transformer so that it could model the structure of proteins. However, as we will review in later slides, it was not as explicit as that. However, they did incorporate many geometric, physical, and evolutionary bases to be able to precisely take into account the three-dimensional patterns of the proteins, their angles, their physicochemistry, among other aspects that allowed this degree of sensitivity on the part of the architecture. For this in particular, a key element was precisely the use of what are known as local frames or residue frames in English, which in this case are precisely Euclidean transformations that aim to represent what would be the backbone of the protein in a local way, and take it to a global form with subsequent transformations that we will review below. So as you can see, here we are looking at the classic backbone of the protein, where we have what is the alpha carbon. Here we have the nitrogen and the carboxyl, the N or C terminus, whatever you want to say. And in this case, what the local AlphaVolt frames do is precisely look at what this configuration of alpha, amino and carboxyl carbon would be and represent them with. Let's call them that, little triangles, although in reality they are mathematical entities. So we then turn to look at precisely what alpha carbon, nitrogen, and carbon are to create this type of local frameworks. And what's done? We have built or defined the local frames that again represent the backbone of this protein. So now we eliminate what are the peptide bonds, so that the protein now no longer has this sequential or linear nature. But then the algorithm is given a certain freedom, so that it learns precisely to identify this sequence of amino acids. And in addition to each of these local frames, a precisely local reference frame is now added, where each of these little triangles in three-dimensional space will be able to both rotate and move to precisely be associated with what would be the prediction of the atomic coordinates of each of their respective atoms or residues in this case. So in this way, what we do is, by providing this local behavior, and through what would be the computation of multiple characteristics between each of these frames, the aim will be to take from a local frame to a global frame that precisely represents the three-dimensional structure of the protein, and that during this computation from local to global, precisely the interactions that are long distance and that are not necessarily contiguous at the sequence level, but can be distal in such a way that we could say some example that what would be residue 25 is in contact with residue 377 in the three-dimensional structure, but evidently at the sequence level since they are quite distal. Obviously, it is also clear that attention will be paid to the adjacent residues because they are close in three-dimensional space. But precisely what interests us, and thanks to the transformer, is that we can also now, in parallel, precisely integrate the long-distance interactions. And precisely each of these local reference frames is represented by what would be a rotation matrix, which in this case, when you review the AlphaFold article, what they do is use a specific type of transformation called quaternion. It is a type of data structure widely used in the world of three-dimensional modeling, which is much more computationally efficient. So when you recognize this term, so that you are not scared by this, it is the way in which the rotation matrices are represented, and also each of these frames or local frames are represented together with another translation vector, and it is precisely this creation of these local frames that allows, as you can see, the preservation of the angular information in what are the connections present in the backbone, and by being able to preserve this type of information through this representation. Precisely, the system is equipped with the ability to model aspects such as the chirality of the residues, something that obviously interests us a lot in order to be able to describe relatively faithfully what the behavior of each of the proteins would be. So once we understand what these local frameworks are, we can briefly explain what the structural module that is actually published in the publication would be. It appears illustrated in the AlphaFold publication. So, as you can see, what we do is start from the representation of the MSA that corresponds to our protein that we intend to model. And then we are going to go through a first module where a very particular tension is going to be computed, which is called the invariant point attention, or perhaps translated into Spanish, it would be a tension by pairs of points. If we can translate it like this, there really is no direct or faithful translation to what Spanish would be from my perspective, so we can leave it as invariant point attention. And in this case, when computing precisely this variant of attention, now what we are going to do is obtain what would be an update of this representation that corresponds to our protein. But as you can see in this part of the invariant point attention module, it is also fed back by what is the information from the pairwise representation of receipts, along with what would be the coordinates that have been learned from each of the representations of these local frames. That in this case, when you start training AlphaFold, each of these local frames is initialized in the same specific reference margin, as if it were precisely the singularity of a black hole, and something which the authors called black hole initialization. So once all the local frames that represent the backbone of our protein have been initialized under the same coordinate axis, we can then think precisely of this collection of local frames as if they were a gas in a three-dimensional space, where they are going to interact in such a way that they manage to resemble what would be the three-dimensional coordinates of the backbone of the protein, something that during this process of let's say or think of it so that each of the particles of these local frames are colliding, the authors again decided to give a quite conspicuous name that is known as the gas of residues or gas of local frames, because they are precisely in a three-dimensional space, and therefore they are creating what would be or reflecting the three-dimensional structure of the backbone of the protein. As you can see precisely here, all the backbones are precisely initialized in coordinates, just as it would be in the singularity of a black hole, and eventually, since they adopt what would be their three-dimensional configuration associated with the backbone of the protein. And as we can see, precisely this information on the coordinates of the local frames is also used to calculate this invariant point of tension module. Again, then now with the product of the invariant point of tension module, we can now update the coordinates of each of our local frames, as well as updating what the representation of our particular protein would be. And along with these two types of information, we now do the prediction of the x, y, z coordinates of each of our atoms. It is also done with the angles pi and psi, as well as the angles chi. And here, as you can see, precisely here, this three-dimensional arrangement of the local frames is now reflected here. We only now add the information of the chi angles that would correspond to each of the residues. That is, first what is being addressed is precisely the coordinates of the backbone, and subsequently the information on the chi angles is now being added, which, as you will recognize, are the angles related to the side chains of each of the specific residues. This is in general what happens in the AlphaFold structural module, and as you can notice here, this really happens during 8 layers of what this transformer represents, so that we could then see it as something cyclical, where then now these new coordinates of our local frames will be injected back into what is the invariant point attention module to further update what would be the representation of our protein. and again improve the predictions of the backbone coordinates, as well as the side chains. In addition to that, the calculation of what is known as the PLDDT also occurs, which is this metric that we previously reviewed, which is precisely a measure that helps us evaluate the confidence that we can have in the quality of the predictions for each specific residue, which in this case is precisely seen, represented by a coloration that the authors designed, where we can talk about 4 large categories. If our residue has a PLDT value less than 50, we can say that it has a very low prediction quality. So the algorithm is really not sure that perhaps this specific residue could be associated with some PLDT region. Alpha helix, beta folded or twist, does not really know the identity of what would be the locations of its 3-dimensional coordinates for each of the atoms of said residue. When a waste now has a PLDDT of between 70, from 50 to 70, we can consider it to be of low quality. When a waste has a PLDDT of 70 to 90, we can say that the quality is already meritorious. We can say that it is reliable. And when that PLDDT is greater than 90, we can say that the algorithm has good confidence that those are the coordinates to which precisely this residue could correspond. Something that we can see already obviously colored in what this protein would be, where you can see that. Not necessarily regions associated with secondary structures, whether folded cebetas or alpha helices, even turns, will have a high quality of prediction, and in this case only this small region, perhaps because it had more information, derived from what is homology, since it was able to predict with better quality what the coordinates of the atoms of this region of the 3-dimensional structure would be. So with this, it really represented a very useful way that biologists can now precisely evaluate the quality with the structural models that we are working on thanks to this implementation carried out by AlphaFold. And in addition to this confidence metric, which would be one of the main ones, the PLDDT, 2 other metrics are also calculated, which is called the GM score prediction, which we also already reviewed in the protein structure biology class, and a new metric called the error of what would be the alignment between pairs of residues, which in this case we see represented by this type of matrices in light green, where they are precisely defined from 0 to 30 amps of distance, and what together these 2 quality metrics give us, also together with PLD, well, in this case only these 2, is a notion of how good is the global prediction in terms of the folding of the structure, or even when we are working with protein complexes. Well, the 3-dimensional configuration of said complex, which in this case, as you will remember, what the TM score does is give us a score when we align 2 proteins structurally, and they correspond to the same folding, also at higher values, since we can expect that the folding that we intended to model is being modeled. And in the case of this new metric called pi, which is precisely this alignment error, what it indicates or suggests to us is the degree of compaction or organization that we can find in our proteins. As you can see here, what would be the distance in Armstrong, if we go from this residue to this other, will be relatively short because it is a domain that is relatively compact. But when we compare the distances again in Armstrong of this residue with respect to this other region of the protein that even looks like another domain entirely, then it is quite sensible to note that in Armstrong terms this distance is much longer. Regarding these others that are contiguous, something that we precisely see reflected in what would be these maps, where this entire first region would correspond to this domain of the protein, and this entire second region would correspond to this other region where it turns out that it is represented by 2 alpha helices, and that this separation is precisely reflected in these light green frames that would reflect precisely what would be the loop regions that represent this protein in general. We are going to review these metrics in a little more detail, but now we are going to pay a little more attention to what this idea of black hole initialization represents, where precisely what we want is for AlphaFold to autonomously learn the sequential nature of the proteins, in such a way that, as we remove during this representation of local frames, what would be the peptide bonds? Well, then now we also inject a new way, or encourage a new way for AlphaFold to learn the sequential structure of proteins. And so this is where this concept of waste gas comes in, where all the frames are going to be initialized on the same coordinate axis, and then these frames are going to have this freedom to both move and rotate in this space, and that this rotation, well, dynamics will be precisely regulated or managed by what is the module called invariant point detention, which, thanks precisely to these two types of representations, is that the three-dimensional interactions between residues can be modeled, which in this case, as we are going to observe, are all initialized here, and we are going to see little by little how AlphaFold learns to represent both the secondary structure regions and the protein in general. As you can see, they all initialized at the same point very quickly, and there are really no clear enough regions that we can discern associated with some type of secondary structure. And it is little by little over the training time represented by these numbers at the bottom in bold that the algorithm precisely learns to decode what would be, which residue corresponds to which region of secondary structure based on its three-dimensional arrangements, own and the neighborhood. And that's how, in general, he was now able to learn to predict what the three-dimensional coordinates of each of the atoms of each of the residues of the protein would be. And now you may wonder, how does this module that seems to really be the heart of the structural module work? Here again the relevance of this module or this operation, invariant point attention, or IPA for its acronym in English. It is precisely what we intend is that the tension that is provided between two residues in this three-dimensional space is invariant in terms of rotations and translation of the protein, right? And this is achieved precisely with this representation of local frameworks, as we can see here. And this representation is really what AlphaFold allows is, well, precisely providing a capacity, let's call it that, although perhaps it is not so explicit, of geometric or spatial reasoning in three-dimensional terms. And this is achieved precisely by, within each of these reference frames, latent representations will be created in terms of query keys and values, which will precisely represent these local reference frames. So once we create these latent representations of queries, keys, and values, we can compute, as you can already guess, what it would be like to compute the tension. In this case, now we are going to compute precisely what the affinities would be, or perhaps in more familiar terms, what the Euclidean distances would be between local frames, as you can see here, between what would be the query vectors with respect to the keys vectors, for each of all possible combinations of local frames. And then, thanks to this type of measurements, we can associate some type of affinity that is actually reflected as the Euclidean distance in a three-dimensional space that would exist between each of the local frames that represent said amino acid in the backbone of the protein. And so this really is an operation that what it aims to do is once we identify these Euclidean affinities or distances between local frames, it precisely takes us through a series of transformations between what would be this local frame towards a global frame in general that is capable of representing the three-dimensional coordinates of the protein's backbone. Something that, as we can see here, is really explicitly how the tension between two pairs of residues is computed for each of the tension heads within what is the structural module. As you can see, in this type of tension there are several terms, and perhaps the most relevant thing is that in this first two pairs of terms it is information derived from the Evoformer where they will be used. The representations, or rather the attention calculated through the keys and query keys vectors, that really, since these derive from what is the one-dimensional representation of our protein, we can say that it can be information of one dimension, and in addition to this type of information, a bias is added that comes from what is the pairwise representation of proteins. That we could think about it again, as we covered from the beginning of this explanation of the architecture of AlphaFold, as the distances between pairs of residues of the protein. And in this case, when seen as a two-dimensional matrix, we can precisely think that now this is information in two dimensions. And this last part of what is the computation of tension in general is precisely what is known as the invariant point of tension, which we can think of as in terms of information that now derives from three dimensions, where again, what we have are precisely these local frames or residue frames in the form of each one, represented by both these contemporaneous as well as translation vectors together with what would be these new latent vectors that represent here each of these little points, because literally, it is really called invariant point of tension, because these types of latent points are projected where the queries are. The keys, and thanks to their computation together with what would be the calculation of the dot product together with the local frames that is taken from a local reference frame to a global reference frame hand in hand with other properties, such as this type of operation that is known as the L2 norm, a type of transformation widely used in machine learning in general. And that precisely can be interpreted in terms of Euclidean distances, and that is really what we could point out as what provides all this calculation, all this operation of invariance in this particular system. Also here there are some other parameters that help learning, and that are even parameters that have to be learned during AlphaFold training, which we are not going to discuss as much. But really here what I intend to communicate is the idea that in this particular module, the invariant point of tension, or IPA, what is generated are these little points that now represent the local frames, and that the tension is now computed. So that we can now think of it in terms of Euclidean distances between pairs of queries and keys that represent each of our local residual frames. As such, when we can see in more detail how the structural module works, we are going to see a horrible monster like this, where, thanks to the addition of colors, we can more easily identify what would be the information that derives from a dimension or the representation of the embedding of our protein that we want to model. The two-dimensional information that comes from what is the pairwise representation, which we already learned previously from the Evoformer, and what would be the IPA, or embedding point of tension, which we can see here in green. Where here you can see precisely how in this section the latent vectors associated with each of these points, queries, keys, and values are generated. As you can see here, we start from the representation of our particular protein and first calculate the affinities, or something similar to what the tension would be in this case, and this representation is biased or influenced. So it would be precisely a type of information that simply passes through a linear layer to be able to be dealt with what would be the dimensions of the respective embeddings, and integrated in a more harmonious way to become integrated. In this new module in particular, as you can see, it goes through this linear layer, and then this particular information is known as peer pathways, which is injected with what would be the representation derived from this first type of attention in general, derived from the representation of our protein specifically. And now here we turn to see how these local frameworks or waste gases work, where precisely these latent vectors are generated, and what would be the local affinities are computed between pairs of local frames, so that they learn to identify which ones are more contiguous in three-dimensional space and which ones are more distal. But when they are all seen precisely initialized in the same reference frame, they allow the identification precisely of these interactions that are long distance and short distance. And then again, this type of information is now used to update this first type of representation, where now you can see that we now move to a module that we can call integration, where you can notice quite well how it is calculated then now. What would be the tension of what the pairwise representation is? The tension derived from these generated latent vectors or query keys points of what would be our initial latent repression of our protein, and the information derived precisely from our representation of the particular protein to now generate an update of this state that actually includes the information of both 1 dimension and 2 dimensions, as well as now 3 dimensions, where now the coordinates of the atoms that correspond to each of the backbone residues are also considered precisely. This is not the only thing that happens in the structural module. Obviously, once this update of what would be our protein that we intend to model has been generated, it will be used together with another set of neural networks, such as this one called the backbone update, which is precisely responsible for predicting what the rotation and translation vectors would be. Then there is yet another series of systems that precisely derive the particular angles, pi, psi, omega, or chi, along with the new positions of each of the local frames. And finally, now compute in a more comprehensive way what the positions of each of the atoms in the system would be. So, as you can see, there really are many neural networks, and in fact, for each of these lines, as we previously discussed, it is also a type of neural network that does not have an activation function, but that helps us deal with dimensionality to precisely arrive at what the prediction of an atomic structure would be. As I mentioned, in addition to what is the prediction of atomic coordinates, they are also predicted 2 key confidence metrics, which is the PLDDT or the GM score. In this case, as we previously reviewed in what the protein structure class is, this metric known as the LDDT really represents the fraction of distances that have been preserved in a delimited range of interaction in Armstrong's terms. But that does not precisely require aligning the structures, which is computationally efficient for this particular case. As we can see here, in this case it would be this region delimited by 15 Armstrongs, but it could also be 0.5, 1, 2, or 4 Armstrongs. And we can identify that these distances indicated here in blue are also preserved with respect to what would now perhaps be our prediction, where now in this other region we will see if, for example, this distance between this residue I, with respect to this other residue J, well, it really is consistent with this other one, which is the real protein of the PDB. This is the first metric that is predicted, and the second is precisely the TM score, which, as we already reviewed, what this metric aims to do is give us an idea of whether our folding really corresponds to one of the type that we intend to model, with values of approximately 0.5 greater, suggesting that the same type of folding is being modeled. And this is precisely what then in the article they will be able to find when they compare on the. And what we observe is that when we note that they also correlate through this Pearson correlation coefficient, we can see that the predictions regarding the real values are quite good, with a coefficient of approximately 0.76 in the case of the PLDDT and another Pearson coefficient of 0.85 in the case of the PTM, for a total, in this particular case of this type of graphs, of 10,000 or almost 11,000 structural monomers. So we can say that at least in their predictive capabilities for these two types of metrics, they do have quite good quality in this sense. The other metric that I mentioned is precisely this alignment error, which helps us precisely to have an idea of how much confidence there is in the term of their relative positions between pairs of residues. That again, as we already saw, precisely those domains that are in a more compact way, because they are globular, will present a smaller range of distances, because precisely they are a domain where each of the structures or residues is very packed together with respect to other regions that could precisely be declared. When we explore this type of maps or heat maps, where we mark now with this mouse now this entire region, that would correspond to this other domain. And that precisely what it can give us is an idea of the general organization of each of the domains of the protein, although it will not necessarily correspond precisely to what a domain is in this case. As you can see here in this other protein, in this case at the top, the protein is colored in terms of PLDDT. And as you can see, really in general terms, it could be said that it is a normal protein with good quality because most of the waste is blue. And in fact, in order to be able to obtain, let's say, a global metric that helps us evaluate whether a protein is also predicted, it could be a good idea to take the PLDT values of each of the residues and take an average of all of them. And for example, if there were 100 residues, then we add them all and divide by the total number of residues, right? That represents then what the rating of the protein as a whole would be, right? Not from each of the waste. However, when we go to the AlphaFold database of structures, it turns out that in this case, when we color precisely by this metric called PAE, we can observe that this first region is assigned to what would be a particular domain. This other region also corresponds to the other domain. And this other region is actually a little more ambiguous, because only this other part is recognized, when in reality perhaps we could think that. All of this could represent a complete domain, if nothing more than the presence of this spin region. It's just that in general terms, then this metric known as PAE helps us to have an approximation of whether, for example, some region could be represented as a domain, but to be able to really evaluate this, we have to do analysis with other types of tools that precisely when comparing it with what would be the bases of the classifications such as the CAD, the scope, or other domain classification systems, since they precisely tell us that a certain region corresponds to a specific domain. This is obviously a little more useful when perhaps we talk about protein complexes, where precisely we are interested in whether two monomers that have now been predicted as a protein complex. Protein because they are as if in three-dimensional contact, and then we could expect a map that would be of a darker green color compared to a light green, depending on the type of interaction that exists between this type of monomers. Here we have what would be a summary of this type of metrics that in general terms what helps us is to evaluate, both in local and global terms, the confidence that exists precisely in our structural prediction, and that in the case, precisely that we are working with. There is also the equivalent of each of these metrics, where an I is usually added to indicate that we are talking about an interface, where for example there is the IPI or IPTM, or some even say IPLDT, although this does not sound so sensible to me, but I have seen it, so it is more reliable when they come to see as IPI or IPTM, which represents precisely in its perspective now as a protein complex. So as we can see, first we have the elementary, basic, or most representative confidence metric, which is the PLDDT, which usually takes, or rather is defined within a range from 0 to 100, where when we look at a residue that has a PLDDT of 90 or more, we can think that what are the atomic coordinates of its side chains would be correct in this sense. When the PLDT is greater than 70, we can think that what the backbone is well predicted, and that when the PLDT is less than 50, it is possibly not well predicted, or more than possibly, rather it is not well predicted what that specific region would be, or perhaps also in a quite curious way, it could be related to intrinsically disordered regions, as we previously reviewed in the protein structure class. Regarding the metric called pi, which is this alignment error, what it simply tells us is precisely the confidence that exists between the distance between two pairs of residues. And it takes precisely values between 0 to 3 angstroms, and can give us an idea of what the globular organization of each of the domains of the protein would be, but not necessarily each of the regions can be seen associated with a domain. In general, then we have again what it would be. The predicted PTM or TM score, and again this metric takes values like the original TM from 0 to 1. And in this case, we can say that if it is a value less than 2, we are talking about the prediction being quite bad because the folding that we intended to model is not really being predicted. when the PTM is greater than 0.5, perhaps if we are talking in global terms that it was predicted in a general way. Or global are folding relatively well. And well, values between 0.7 and 0.9, well, perhaps we now have greater confidence that our protein folding was predicted well in global terms. We can also use other types of analysis to, let's say, validate our predictions with AlphaFold. In this case, the idea of replicates is also usually recommended precisely. Be able to precisely evaluate whether the prediction is consistent. So we could actually have, when we run the AlphaFold algorithm using Google Cloud. In this case, the prediction of up to 5 structures for each one is implemented by default. And then precisely which is the best in terms of PLDDT is ranked, as rank number 1, rank number 2, rank number 3, with rank number 1 being the one with the best PLDDT, and rank number 5 being the one with the worst PLDDT. But if, for example, we observe that in general terms a region always appears very poorly predicted, and it turns out that this region is associated with an intrinsically disordered region. We could think that perhaps it is because it is already an intrinsically disordered region, or because some other type of inferences that resulted from our specific analysis. And another metric that we can also evaluate to validate the quality of our predictions is to look at whether our structural prediction is biochemically plausible. In terms of, for example, we carry our chimera, and then we see the patterns of hydrogen bonds. We see potentials like their electrostatics. If, for example, hydrophobic residues really are buried in the protein, or if it turns out in an implausible way that hydrophobic residues are now exposed, then perhaps in this case, we could think that the protein does not make sense in biochemical terms because this type of residue should be buried by that hydrogen, while what would be the hydrophilic ones should be precisely in the surface region of the protein. I also raised this once, at least on my Twitter profile, to see what some colleagues thought about whether we have to use another series of programs that validate, let's say, by third parties what the quality of our predictions would be. For example, I have seen that some use programs or scores such as Malpravity, AFRank, Ramachandran plots, even newer ones such as AF3 score, but at least in what I discussed with 2 colleagues, together perhaps with what I also think regarding AlphaFold's quality metrics, for me it would be more than enough to just make this type of assessments, using the quality metrics that AlphaFold precisely gives us, and that perhaps these quality metrics from third parties would be unnecessary when actually choosing a set of candidates to express within our protein, or perhaps use them for some other type of specific data selection filtering. This again will depend a lot on the experimental design they are doing as well as what they intend to discover or analyze. In some cases, the percentage of protein exposed to the solvent is used, other metrics derived, for example from Rosetta, and a further set of metrics that may help you rank which would be the best candidates for your proteins to express. But it is to validate what would be, well, its 3-dimensional structure. To me, it seems sufficient to do this type of replication and analysis to ensure that, for example, its hydrophobic residues are buried in the protein. Or they could even predict it using another type of system. For example, if you are modeling a protein with AlphaFold2, perhaps you could now predict it with, for example, the Evoformer, which we previously reviewed when we were explaining what the Evoformer and triangular tension are. Or even the new version of AlphaFold, such as AlphaFold3. So they might think that they now have the AlphaFold2 prediction, along with the AlphaFold3 prediction. And they see that the protein, the monomer, or even the complex that they are looking to model is being predicted well. Well, it would seem to be enough in terms of validations, depending again on the type of work they are looking to develop. to say that its structure was predicted with good quality or not. So good. We have already reviewed what this first layer is, which is the input features, then this second layer, which is the Evoformer, and again now we have finished explaining what the structural module that we just reviewed would be. So now we move on to what would be the next component of what the AlphaFold architecture is, which is the ability to recycle precisely all this information where we see precisely that we take the representation in pairs. And we also take the information from our updated representation, already using all the information that we saw with IPA and all this, to again be injected into our MSA representation and our initial pairwise representation, and again to be refined again through 48 layers of Evoformer and again 8 layers of what is the structural module. And it turns out that this behavior, where what they do is precisely, well, injecting this updated information into new cycles, what they have called precisely recycling, is sometimes quite productive to really improve the qualities of the structural predictions of some proteins. For example, here we see a protein in total, which in this case has two specific domains, domain 1 and domain 2, which in this case are marked in orange or blue, the same here in the three-dimensional structure. And what we observe, that is, on the yes-axis, is that it is also being predicted using the GDT metric with respect to the recycle cycles that are occurring by the Evoformer module, which in this case, as there were 48 layers, well, this would be a first recycle, these would be 96 layers or 2 recycles, and so on, 3 recycles up to 4 recycles. And so you can notice that it really is a fairly large number. 192 layers through which the information flows, so this is also obviously computationally expensive. So for example, in this case, the behavior of these two proteins is quite friendly in terms of the predictions, because only in what would be first run are predictions with a relatively good quality in this sense already achieved, and that when we increase the number of recycles of the architecture, it turns out that it remains relatively constant. There is no change really considerable in terms of the quality of the predictions, or even it would be minimal, that perhaps does not reflect a productivity in terms of the computation that is being invested. This is the behavior presented by this type of proteins, but there will also be other proteins that are therefore more difficult to model, as is precisely the one represented with this case of a protein, marked with this green line where what we observe is that the predictions really during the first AlphaFold runs are bad. They do not reach a GDT of less than 50, which is really quite bad because we are really looking for something greater than 70. And during what would be the second iteration, it improves a little more. The third iteration a little even more. And until the fourth iteration, it already improves even at a level of 80% of GDT, which would be quite good now. And it turns out that when we turn to see what type of proteins these are, for example, in this case, it is a protein from a gene product called ORF8, from what would be the SARS-CoV-2 virus. And when we look at the quality of its alignment, as you can see here, it really is a protein that cannot be generated with a high-quality alignment, as we discussed previously, where we actually found 3 large sections, where there are a few homologs that are quite similar to what our query protein is. There is a slightly larger number of approximately 10 homologs that turn out to be at an intermediate level of sequence similarity, and another 10 homologs that almost share very little sequence similarity. So we can think that during this distillation process learned during the Evoformer in structural and evolutionary terms, because it really cannot be associated precisely what the information derived from the alignment would be with what would be the three-dimensional coordinates that are learned during the structural module. That is, it is a protein that does not have evolutionary information from which the structure can be learned, and that thanks precisely to this recycle mechanism. The structural prediction can be precisely improved, despite the scarcity of counterparts, as we can see in their alignment. And here what we observe is precisely how the prediction of a protein can be observed, a protein complex of 863 amino acids, where during the first phases of recycling, different conformations of each of the regions and domains are explored, and then there will be some with formations that are quite implausible, but which turn out to reflect the way in which AlphaFold learned precisely to model this type of protein complexes. As you can see there are some three-dimensional configurations that are quite implausible, as you can see there with that alpha helix region that is quite promiscuous in three-dimensional terms, and that is not until you reach precisely what would be a third recycling phase, which can already be said to adopt a relatively sensible three-dimensional configuration compared to those we saw that were explored throughout this recycling process. So this was also quite useful from the AlphaFold authors as a mechanism to improve predictions in case you don't find enough homology information in the alignment, or that they are quite large macromolecular complexes, as this could be the case of up to almost 900 residues in total, which really with other types of algorithms that preceded AlphaFold was actually quite difficult to achieve here, as you can see in general terms, and more than anything for what would be proteins that are a little common within the range of what is considered common for a protein of approximately 300 residues in general. It turns out that even with no recycles or up to 3 recycles, quite good predictions are actually achieved, as you can see here from these box and whisker diagrams, which in this case what they are doing is evaluating precisely the TM score on the yes-axis with respect to the number of recycles for a set of proteins de novo. That is, they were designed and therefore do not have much homology information derived from the alignments. And in this case it turns out that there was a bug in the first versions of AlphaFold, which has already been corrected, and that in this case, even with just one recycle, you already have a general or average TM score of above 0.8, and that is being refined little by little as we add a number of recycles. And that, for example, when you are using the AlphaFold platform, by default, it might be a good idea to select a total number of 3 recycles for your proteins. In case you do not know in advance how represented your sequences are in the databases to be able to actually derive those homologs from said databases. This is the behavior of some proteins again, but perhaps not of all proteins again. In fact, it has also been seen that there are some proteins that are actually much more challenging in terms of achieving a prediction with relatively good confidence, in such a way that up to 15 recycles are required in total, as we can see here. In the first few cycles, there are really no regions of observed secondary structure. It really seems quite disordered in this sense. And it is not until, for example, what would be a recycle from an 11th iteration to the 15th iteration, where we can really better discern the patterns of associated alpha helices to these proteins again, which are transmembrane, and as you might guess, since they are transmembrane, these types of proteins are not as represented as if they were proteins in the cytoplasm or extracellularly, and it is actually much more complicated for AlphaFold to learn to predict this type of protein, because its behavior is hydrophilic, or rather contrary to what this pattern of hydrophobic in the center and hydrophilic on the outside would be. Proteins that are precisely membrane-bound invert this pattern, and because there are not many types of these proteins deposited in the protein data bank, then AlphaFold is more challenging in this sense to predict their three-dimensional structure. And in this case, thanks to recycles, we can precisely achieve better predictions. An also quite interesting feature during the AlphaFold training is this idea that the authors called self-distillation, where it is actually a word that was used to refer to the fact that in the first instance the AlphaFold training precisely used the structures deposited in the protein data bank, which was for the cutoff date where the algorithms were trained, and the data were downloaded in order to precisely carry out the exploratory analysis of the data and the entire cleaning and analysis procedure, since there were approximately 175,000 structures present in the PDB. And with this precisely, they trained a first version of AlphaFold, using the modules that we already reviewed, such as the Evoformer and the structural module. Now, as we train this first version of AlphaFold, the authors decided to take precisely their model and now take sequences that have been clustered in terms of 30% sequence similarity from a database of protein sequences called UniProt, a database widely used in terms of protein research. And then, once clustered, what would be this set of sequences? They found a total of approximately 355,000 clusters or proteins at 30% sequence similarity, with which they now took their AlphaFold model and then used it to predict their three-dimensional structures, and thus enrich that first set of 175,000 proteins that they had. And now, by being able to make these types of predictions, they went to a total of approximately 530,000 structures with which this first version of AlphaFold was retrained, so that it could now adopt what would be the information present in this type of structures. Obviously, all those that were structures with poor qualities were discarded, and only those that had good qualities were preserved. And other types of mechanisms were also used to enrich the signal that we can derive from this type of proteins, right? Such as only taking certain fragments of the proteins, or only training a first phase to small monomeric proteins of no more than 300 residues, and that in later stages of the training, we now move on to model much larger proteins of 500 or even 1,000 residues, so that then during the training AlphaFold would gradually learn to distill the information of proteins that are increasingly more complicated in this sense. In fact, this is precisely something that is reflected when we look at its loss function, where we can see that there are two general terms. In fact, in this first one, rather, they used a special set of functions when it was this first training phase. And then they added two other terms in particular, here in this case marked LX resolved and in this case LVIL, during a fine-tuning phase, so that AlphaFold could precisely learn to predict the three-dimensional structures of increasingly complicated proteins. In this case, we're going to explore each of these terms to see what they mean in this spatial equation. What is called this is the frame-aligned point error, or FAPE loss. In this case, it is precisely taking each of our local waste frameworks and practically aligning them of what the prediction would be in green with respect to the original structure in gray. And then, for example, here we can see that what would be the distances in these local frames of what would be the original with respect to the predicted one, since there is an error in this case, in this specific prediction for this scheme. And this is precisely what penalizes this type of loss function in particular. The fade loss, which was actually also a new addition, an intellectual contribution and innovation in the type of algorithms used to predict three-dimensional structures by the Google DeepMate team. And then what we observe is that there is another variant of this FAPE loss, which in this case is called AUX, auxiliary, where what is done is to compute the FAPE loss, but for the intermediate structures calculated in the structural module, which, as you remember, are 8 layers in total. So then, by adding this term, what is promoted, incentivizes the algorithm, is that even from the first stages of training, in the first layers of the structural module, structures with good quality are already being predicted in this sense. Then we have a third term, which is dist, which in this case is a loss associated with what would be the distogram. As we review what the architecture of AlphaFold1 is, the histogram represents a set of distance distributions for each pair of residues. And this is such rich information that it was also decided to preserve in what is AlphaFold2, only that it was no longer given as much relevance in what was AlphaFold1. But in this case, it is also a signal that is also quite rich in structural terms of the proteins, and that was now incorporated as a parameter within the loss function. And that perhaps we could see it a little more reflected in what would be the structural injection, with what would be the pairwise representation of residues implemented in FF2. Then we observe what is a loss associated with what is precisely the behavior of the architecture of the Evoformer, which, as you remember, is a BERT-type transformer. So then during this entire procedure of generating updates of both the MSA representation and the pairwise representation of residues, also what is done is precisely to mask residues up to approximately 15% of the total residues of the alignment, in such a way that in some cases some residue of our protein in particular is masked at random or even complete regions or perhaps complete columns, and this more than anything will be reflected with respect to randomness, because it is. Masking of approximately 15% of the total tokens being operated in general is usually used. And then, thanks to this masking loss implemented in the Evoformer, what is precisely promoted to the algorithm is the idea that it learns to distill coevolutionary information from all the residues so that it precisely becomes able to say which residue would be most likely to go in that masked region. And so precisely learning more and more fine representations of the pairs of residues, because this is also being enriched in a feedback way, let's call it that, with what it would be the representation by pairs of residues. Then we have this other metric, this other loss term, where what is done is also injected into the algorithm. This ability to increasingly improve its predictions in terms of confidence, where precisely with a lower penalty of approximately 0.01. As you can see here in the case of the Evoformer, this is the main module in AlphaFold2 in terms of the loss functions that are implemented, because here it has a value of 2, not even what the fave loss would be. In this case, predicting your predictions with good quality. It has a lower weight within the network, but precisely the idea is also promoted that it becomes capable of precisely detecting regions that are well predicted compared to those that, despite having some three-dimensional structure associated with secondary structure patterns, are poorly predicted, as you can see in this particular region of folded betas and alpha helices. This, as you can see, constitute the first stage of AlphaFold training. But a second stage of fine-tuning was also precisely added, where what would be physical information is now injected that allows even further improvement of the predictions, as well as in terms of the physical-chemical limits present in the proteins. And this was done with another term in particular, where what was done was to indicate to the model if the predictions for which you are focusing on that specific task start from a structure with a high resolution, obviously to structures that have a poor resolution, seen as greater than 3 Armstrong's resolution in general, where we can observe that what would be the electronic densities would no longer be so capable of really reflecting the positions of the electrons of the side chains or the backbone of the protein. Well then, by indicating that these proteins, let's say, being less than 3 Armstrong in terms of resolution, they are well predicted in terms of their atomic coordinates, then focus on learning more information about this type of good quality structures present in the PDB. And also added one more term of violations in terms of the links and their lengths, as well as the angles, or even what would be the clashes between side chains, which, even as you can see here, also have a high penalty cost. So it is in this last stage, when AlphaFold is already capable of generally representing the protein quite well, where this new stage or phase is now enabled, where it must now be sensitive enough so that aspects such as how much is the maximum it can produce are not violated. Measuring a hydrogen bond between 2 specific side chains, or the fact precisely that there are no clashes in the side chains of the protein, because that is impossible physically, chemically, and well, not really. So, thanks to this whole set of penalties in the loss function in general, and through this strategy of first teaching you in general terms to represent proteins and then refining its understanding in physical terms, is that it was really possible to have a fairly fine system that what it does is model the coordinates of the backbone quite well. Then once the coordinates of the backbone have been modeled, the coordinates of what the side chains are constructed in such a way that they respect precisely the physical sense of their three-dimensional positioning in the protein. In fact, as a last step, also in case it has not been greatly improved with this type of penalties, violations of angles and collisions, what is known as a relaxation is also usually applied using the AMBER program, where these empirical functions that we previously reviewed are applied. Associated with what would be these models of molecular dynamics or docking, or in general in terms of energy such as Rosetta, Foldex among other tools where we find, for example, here that before what would be the relaxation with amber because there is precisely a collision between the side chains of this methionine with this tryptophan as you can see here which, physically, it is not plausible and that after the relaxation. With amber by minimizing the energy that also represents a computational cost within the algorithm because then these side chains are no longer colliding precisely and now we can see that they adopt a three-dimensional configuration. Really more sensible because there is no longer a sterile collision in that sense, where now the methionine is free in that sense, with its respective charge, and also here the tryptophan rings are not in complete collision with the methionine itself, which also helps us when even modeling a protein that is plausible in structural terms to be able to refine our analyses that, for example, a contact is being established, that, for example, is binding to a ligand, or that is capable of carrying out a specific catalytic mechanism. So again, depending on the type of analysis you are performing, it would also be a good idea to apply this type of relaxations with amber that are not necessarily activated by default when, for example, you use the AlphaFold platform. But for example, when you are wanting, say, develop a project where they intend to model the entire proteome of a specific organism, since we are talking about protomic scales, that is, approximately 5,000 to 10,000 proteins in the case of a bacteria, it would not really be computationally feasible to apply relaxation, because you would have to apply relaxation to 5,000 proteins, which will take you and cost much more time and money. So again, it will depend on the type of project they are carrying out to be able to assess whether or not it would be necessary to carry out this type of relaxations, which what they do is in final terms to further improve the arrangement of each of the atomic coordinates of their protein. And finally, we come to these ablation studies, or in other words, what each of the components that we previously reviewed contributes so much to the architecture of AlphaFold2 in general. And this is precisely what we observe. In terms of two confidence metrics, well, quality, such as the GDT or the LDDT, for two sets, which in this case is precisely the proteins derived from the CASB, which they provide to be able to precisely evaluate, as well as the test with, which the entire algorithm in general was trained. Or well, when all the data splitting was done. This set was the test that was designated, and the training, well, any other set that is completely, or in the best of cases, quite independent with respect to this training set. And what we observe is that, here we observe precisely as a first instance, is that the idea of incorporating this self-distillation mechanism that ended up representing approximately 350,000 more proteins of which it learned. Well, it did have an improvement in terms of the qualities that are predicted in both sets when we compare it with respect to a baseline of what would be a triplicate of the AlphaFold architecture in general terms, as we already reviewed. That is, by integrating more data, which in this case are not proteins that are not real, but are proteins that are deposited in the databases, and that after a curation process, selected the best ones from which to learn. Well, even more information could be distilled, despite the fact that these three-dimensional structures were not available. Then we found that by not incorporating the information from the structural templates, AlphaFold really does not seem to be so diminished in its predictive capabilities. Maybe a little yes in what would be the evaluation of the AlphaFold test set, But in general terms, with what would be the CAS evaluation set, it does not really look so deserving in this sense. Also, when we disable what is the head that computes the histograms of the three-dimensional structures, that is, these distributions between pairs of residues that condition the three-dimensional structure, we also do not observe a very marked decrease in general terms. A bit similar also occurs when, or rather not so similar, when what would be the creation of multiple sequence alignments and their consequent representation of the MSA in evolutionary and structural terms is disabled, where perhaps now in what would be the AlphaFold test, now it is seen a little more diminished in its predictive capabilities, and perhaps not so much in what would be its proteins from the set provided by the CASP. And they pass. Well, relatively similar behaviors when we observe whether the invarying point attention module is enabled, when the masking capabilities of the Evoformer are also disturbed, when what are the recycles are not used, when the triangular attention mechanisms or attention at the column level are not implemented within the Evoformer, or even when this option that the architecture is made is not enabled. Differentiable from term to term, which makes communication of the Evoformer with respect to the structural module impossible. Or even what does have a great havoc on the predictive capabilities is precisely inactivating what would be the invariant point attention together with what would be the recycle module. And there you can see that the predictive capabilities are quite affected. And in general, what the authors wanted to communicate with this is the idea that all these multiple mechanisms that they introduced into their architecture add little by little to the success that constitutes AlphaFold. Although you can disable some of these modules that we previously reviewed in detail, it is the sum of the set that makes their capabilities so good to be able to model the structure of proteins. It is true that you can precisely provide key mechanisms such as embedding point attention or recycling if they can help a lot more. But it is precisely the set that really constitutes its predictive capabilities. And this is something that they precisely wanted to highlight during the publication, where the authors really assigned central importance to what would be the Evoformer, which precisely by being able to integrate both the information from the pairwise representation and the MCA representation, allows both evolutionary and spatial reasoning of the proteins to be able to make their predictions. And that in this case, it is these representations that are key so that the structural module can operate in terms of its predictions. And so now one of the questions that we can ask ourselves is, well then, this would be a reflection of the famous title of the article on transformers is attention. All you really need transformers were well the only thing necessary to be able to solve the problem posed by the CASP approximately 50 years ago. Well, it turns out that no, tension is really not all we needed to be able to solve this type of problem. Here what you can see is a comparison of the architectures of other large models to really predict the structure of proteins, such as the ESM1B protein language model, which as we previously covered, is a model trained only with sequences. And in this case, it uses the architecture of the classic transformer. They really can't see more enriching or conspicuous modifications to an archetypal language model. Then we find its adapted version, but now to be able to be trained with multiple sequence alignments, where mechanisms such as attention at the row level and at the column level are incorporated, but it is still adapted. Well, a quite classic transformer architecture in this sense. And then, when we look back at the AlphaFold architecture, now you're also able to recognize what all these modules represent. Here is the initialization of the MSA representations, as well as the pairwise representation, the Pairformer module, the update of these two types of representation that will be used by the structural module to make its predictions and its consequent recycling of the entire architecture. And we see that within the Evoformer there is also precisely a transformer, but a transformer that has these mechanisms of attention at the column level, attention at the row level, and triangular attention, as we can also see. that even when we compare these architectures, for example, in terms of the number of layers, or perhaps even more striking, would be the number of parameters, we see that AlphaFold has only about 100 million parameters, and yet, in terms of the qualities of the structural predictions that we can derive from the MSA transformer, or SM1B, they do not reach the quality that AlphaFold2 does. By integrating precisely this type of tension mechanisms, along with a structural module that we also previously reviewed, since it integrates many physical, chemical, and evolutionary bases regarding how to model proteins. In fact, even the amount of information that was used to train AlphaFold is estimated to be approximately 100 times less than the amount of sequences used to train MCA transformer, or SM1B. So then it is also quite clear that the classic transformer alone is not capable of solving the challenge posed by the CAS more than 50 years ago. But it was precisely the innovation of all these attention mechanisms, representation of local frames, even distillation by AlphaFold, that precisely led to its success as an architecture to predict the structure of proteins, something that we have already represented in this new slide, where I also highly recommend what this discussion or perspective would be, where the reasons for AlphaBot's success are addressed in a little more detail, where we can clearly think that the main reasons are precisely the attention mechanisms implemented in its architecture. Attention by column, attention by rows, triangular attention, or attention by invariant points, since it precisely contributes precisely to what would be its total success with respect to the classic voltage computed by the original transformer. We also find precisely that the second reason for its success is its capacity to be able to distill coevolutionary information from the alignment, and this also reflects precisely that if this type of signals can be distilled, it is because the information is present in the first instance, which reflects precisely the importance that there were large amounts of data deposited in the databases, which also, both in quantity, since they were precisely abundant, as well as in diversity in terms of homology, right? So that AlphaFold could precisely apply this type of evolutionary algorithms and identify which residues would be most relevant at the level of the specific protein, but also at the level of the entire protein family or the set of homologs that were derived to really be able to address the problem of predicting your specific structure. As a third reason, we also clearly understand what equivalence is. This sensitivity of being able to reason in three-dimensional terms, in terms of rotations and translations, to be able to precisely model a good protein. And lastly, we have precisely its capacity to be differentiable from term to term, where the parameters learned in the Evoformer, they are fed back by the parameters learned in the structural module during backpropagation and vice versa. All the parameters implemented in the general high background architecture are fed back into themselves during training thanks to backpropagation, despite the fact that they are modules specialized in learning some specific aspect of the proteins. We can say that what Evoformer is precisely responsible for learning the biological and evolutionary bases of proteins, while the structural module, as its name explicitly says, is responsible for learning the three-dimensional spatial representations of said protein. And thanks precisely to backpropagation and its ability to be interconnected, we can ensure that during the gradient computation, the weights are updated in such a way that both consider each other to update themselves. And then it's pretty clear that really, it was not a single idea that led to the success of AlphaFold, but it was a whole set of ideas, an entire engineering process that led precisely to AlphaFold being this transformative system for all of science. And this is also quite clear, quite blunt, when we look back at the fact that really when we look for a problem to solve with this type of algorithms based on deep learning, well really, it must meet very particular characteristics, such that there are very few problems that are also posed within science to be solved, in quotes. In this case, we see that what was the approach carried out by the CASB more than 50 years ago, as it was quite key in terms of the development of databases, in terms of the scientific community. Incentivized to solve this type of problem, along with the resources provided by the institutions to address this type of problem, in such a way that the three pillars that would be minimally necessary to say that a problem can be precisely solved through deep learning algorithms is one, that if this problem is easily defined in quantitative terms, two, and perhaps in my most important perspective, that there is enough data, and that these data are of very good quality, and that they are obviously also diverse, as was the case of the protein data bank with approximately 200,000 three-dimensional structures created by thousands of scientists over more than 50 years since its existence, and even also databases such as Uniprot with approximately 200 million protein sequences that, well, really, they come from metagenomic experiments, massive sequencing campaigns at the level of human genomes or populations of other animals or other species, and each of these experiments also contributed to the success of AlphaFold, although they are not directly recognized. Each of the scientists who decided to upload their databases to that guy of infrastructure to be able to share it with other scientists within the entire community, since he contributed his grain of sand in that sense. And as a third pillar, we have precisely having a set of metrics that are both quantitative and humanly interpretable, which in terms of the CASB we see reflected with metrics such as the GDT, the LDDT, or the TM score, along with other forms that were developed precisely by the scientists in charge of organizing the CASB to evaluate the consistency of the structural predictions made by different groups, and that together these three pillars represent the minimum set necessary to be able to pose a problem that could be addressed using deep learning algorithms in this case. Something that really is not so shared in what would be the approach to other problems of a scientific nature. For example, when we find that someone suggests that we are going to cure cancer thanks to artificial intelligence. Well, these types of pillars are not really available in such a way that, for example, addressing the disease of cancer really involves multiple types of cancer, and yet each cancer is a specific context for both the patient who has it, as well as the pathological context in which it is developing. So in this case, the degree of complexity is much greater and obviously requires a much more detailed approach to be able to be addressed in terms of possible solutions that we could find through artificial intelligence algorithms. Not any problem can be solved in this sense using this type of algorithms. And well, now we move on to a new phase of this class, where we are now going to explore the main applications of what was DeepMind's algorithm, AlphaFold, and as well as its impact within science, something that from my perspective is quite well illustrated by this film, that I really like, that represents what is the great diversity of protein structures to which we now have access, and that precisely this was one of the greatest contributions by Google DeepMind, who decided to take, which is the protein sequence database most used by us, which is Uniprot, with approximately 250 million sequences, and then they decided to run AlphaFold to be able to now precisely have one structure for each of these 200 million structures. So now in its structural biology database, we now have predicted models for around 200 million sequences to be able to precisely access this structural diversity. As you can see here, this would be the diversity represented in terms of circles by different databases. One would be precisely the PDB, which counted less at the time this diagram was made, with approximately 190,000 structures. Later, in what was the first version of AlphaFold, they released about 1 million structures only associated with what would be the protomes of the species considered as research models, namely Escherichia coli, the fruit fly, among other species, since the structural diversity to which we now have access was increased. And finally, because it was quite a considerable number, it implied a great challenge to be able to deploy all this technology so that it was accessible to any user through a set of clicks, since all 200 million lost structures were released, and as you can see, it increased over time as we can see. And what is this timeline? In such a way that, when what would be the entire Uniprot database was reached, later even new analysis mechanisms were added, such as selecting through PLDDT. Visualize the domains using the PAE metric, or even when carrying out somewhat more massive downloads, know the domains, among many other analyses that we can now do with a few simple clicks, and that fortunately require a strong background or technical bioinformatics knowledge in this sense. That is, this type of bioinformatic analysis of proteins is now available with a few simple clicks, and not necessarily with large computing centers. And as you can see, just for what would be the comparison of what diversity is at the sequence level with respect to diversity at the structure level, we have this other diagram where we observe precisely a slightly more updated version of the PDB, in this case with 214,000 structures. And as you can see, this orange dot represents a speck of dust within what would be the diversity of sequences in other immensely larger databases. For example, it could be the PFAM with approximately 60 million sequences, the UNIREF90 with 193 million sequences, or even the Big Fantastic Database, or BFD for its acronym in English, which in this case is one of the largest sequence databases that we knew of up to that point, with approximately 2.5 billion sequences. This is not the largest database. There are currently other even larger sequence databases, which in this case we are going to address in what would be the last class of this course on protein data and biases, and the sequence level and the structure level. However, it serves us quite well to be able to represent the idea that when we see in the news that AlphaFold predicted the structure of all the proteins in existence, that is totally incorrect, because what AlphaFold did was take the database that is most famous within the protein scientific community, the Uniprot, and he predicted 200 million structures, and 200 million is by far the highest number we know of, as could 2.5 billion. However, it is quite evident what the structural contribution that this database provides would be, which was also reflected in the detection of possible new foldings, such as those represented under the CAD scheme. As we can see over several decades of research, and above all what would be evident, the democratization of sequencing technologies, as well as the lowering of the costs of, and that in this case, thanks to AlphaFold's 200 million structural predictions, we can now evaluate whether even this number of 1500 can really be expanded considerably or not. And this is something that has appeared in several publications independently, and that perhaps here I only put two of the most representative publications in this sense, who took precisely these 200 million structures and clustered them in structural terms to evaluate how many clusters could be found in structural terms within this set, in such a way that these two publications even suggest the existence of at least 1 million structural clusters, even up to 2.3 million structural clusters, and even a considerable part of them would have no structural or even functional similarity with the proteins that we already knew previously. For this reason, it is precisely pending to be able to characterize them both in structural and functional terms to assess whether they are really new foldings. And among the set of proteins that we can find in this type of two publications, we find some quite conspicuous or beautiful foldings, whatever you want to call it. How could this heart-shaped folding, represented by the alpha helices, be a second folding that is called a beta flower, precisely because it is composed of regions of betas folded in the shape of a flower? Or my personal favorite, which is this molecular horn, which is quite curious to find this type of shapes within what would be a protein. And precisely this type of much more detailed analysis to determine whether they are really new folds or not was carried out by this type of projects such as the Dominius Encyclopedia, who took these 200 million structures predicted by AlphaFold, and now they analyzed them under the CAF scale to find how many domains we would find within these predicted structures. So after a set of bioinformatic analyses, it was determined that there could be approximately 370 million domains in this set of protein structures made by AlphaFold. And as we can see, they're composed in a prototypical way of a protein, no longer than 300 amino acids in length. And that in general terms, the vast majority of them, or the equivalent of 54%, have high-quality predictions, that is, with a PLDDT greater than 90. Even a considerable high-quality set, with approximately 39% of the total, and only a fairly small subset of what would be low-quality and very low-quality predictions. So there would then be very interesting candidates to investigate if they could really. It can be represented as new foldings, or in terms of structural diversity, to a previously known family. When we look back at precisely the type of domains that were found in this analysis, we find two quite interesting groups. A group that is associated with what would be highly symmetrical proteins that we previously did not conceive could occur, or that at least we had not observed because the proteins with high symmetry that we were able to observe, at least until now, were approximately grade 6 to grade 9. And now, thanks to AlphaFold, we found that even grade 11 proteins could exist. That is, we take what would be a small segment, which is precisely repeated up to 11 times, as we can see in these two structures. And in total, these high-symmetry proteins represent approximately 6,400 clusters of the total. And even when we look again, after having discarded the worst predicted proteins, after having courted other databases to corroborate if there is no coincidence in terms of sequence or in structural terms, it turns out that there could be approximately 7,500 putative new domains or new folds that are pending to be evaluated by some other technique to corroborate that they are a new three-dimensional pattern of proteins. However, it is quite clear that what would be the structural diversity also went through the roof towards multiple foldings, as we can see here in what would be the top 100 superfamilies most represented in this database, where we even find an increase of up to 200% for some proteins, such as immunoglobulins or in particular macroglobulins, as well as other proteins that were not as represented as some associated with membrane regions among multiple other structures. So now when we perform a structural analysis of a specific fold, or some family or superfamily, it would be a pretty good idea to access the resources now available in this new domain database to do a better analysis in terms of both sequence diversity and protein structure diversity for said family. And so it was that we also reached the point of what some will consider as the end of the experiments, because when AlphaFold was launched, it really was a furor within the community, which is slightly reflected by some comments by members of the protein science community, such as Mohammed Al-Qureshi, who even in a tweet precisely said that it seems that DeepMind has solved the structure prediction problem. And be careful, it is quite clear when it comes to structure prediction, not folding, which, as we previously addressed, are completely different problems. And that even he thought, at least in terms of predicting structures, that he had already finished. This is evidently also reflected in a large number of memes that appeared on social networks, including publications such as journalistic notes in what would be the most renowned scientific publishers. They would even suggest that structural biologists would be out of a job because we can now crystallize proteins with just a few clicks and a computer, which is completely false and completely wrong to think that there really would be no more structural biology thanks to AlphaFold. Even, obviously, there were also people with dissenting opinions regarding the advances that we could achieve through deep learning algorithms, such as this colleague called Mike Thompson. who also believed that we could already reach model qualities of approximately 2 amps of resolution, using what would be modeling by homology of structures, and that in this case, they would only draw his attention when the resolution. Qualities of this type of models reached resolution as clean as 0.2 amps could be. A type of resolution that is usually required to be able to make characterizations at the enzymatic level of the catalytic mechanisms and superspecific positions of the positioning of each of the atoms of what would be the active site of the enzymes. So as you can see, there were like two sides of the coin. What would be a furor for the community in an exacerbated and even quite rude way regarding the work of many fellow structural biologists, and another, well, somewhat more pessimistic, or even reluctant to adopt this type of artificial intelligence algorithms as a new set of tools that open up a new window and perspective to be able to really analyze protein information. In that sense, it is quite clear that we have to recognize what the limitations of the AlphaFold2 algorithm are, since AlphaFold2 really predicts monomeric proteins, including protein complexes too, and their units within these protein complexes, in case they are absent in that sense. However, what it does deal with is being able to predict what the multiple conformations related to the assembly of the structure would be. As we previously reviewed, the structures are actually conformational assemblies, and the protein, although less physiological, is found in different substates that are quite similar to each other in what would be its native representation. What AlphaFold2 also does is have a great challenge when predicting the effects of point mutations, so that if, for example, when we mutate a residue from a tyrosine to a leonine, AlphaFold is not sensitive enough to detect whether that particular mutation could completely break the folding. And there are also obviously proteins that are much more difficult to model, such as proteins related to the world of antibodies, who because of the excessive diversity they have, since it is evidently quite challenging to be able to be modeled with what would be the statistical patterns that AlphaFold learned from the PDB and the sequence database. And obviously things that AlphaFold2 cannot do is precisely model more biomolecular type interactions, meaning protein interactions with nucleic acids, DNA or RNA, with ligands or metal ions, post-translational modifications or membrane proteins. Due precisely to these limitations, it was an issue to be addressed in the development of the AlphaFold3 algorithm to now be able to model this type of biomolecular interactions. Here we can see an example of the importance of the specific context of each of the structures, so that we then have a structure composed of folded veins, which adopt a quite characteristic configuration, similar to a double helix, such as DNA. However, this protein was not crystallized under the classic phosphate buffer, but in this case it was crystallized in a set of alcohols, as you can see here in the text. And due to the interaction of the amino acids with the solvent, this three-dimensional configuration, quite atypical in this sense, was established. So we could ask ourselves the question, what will happen if I take the sequence of this protein and predict it with AlphaFold? Will you be capable enough to give me this three-dimensional arrangement? It turns out that when I take the sequence and model it with AlphaFold, and obviously its 5 replications, we see that these 5 are all of poor quality, and that clearly when I try to align what this prediction is with respect to what the structure would be, as we can see by its red and blue coloring, there really is not a total alignment of this. That is to say, AlphaFold does not integrate information such as that of the solvent in this sense. And this is quite clear because it is not entered for it. It is even recognized that there are actually many proteins within the cell that actually see their behavior configured depending precisely on the environmental conditions in which they are found. There are some that even, for example, in this case of pH, change their three-dimensional configuration so that they are open or closed, depending on a greater increase or decrease in pH. And it is quite clear that tools like AlphaFold really in current terms are not capable of taking into account what those environmental factors such as temperature, solvent composition, or pH would be in their predictions, because they are Precisely structural prediction algorithms. A second example of the limitations of AlphaFold also comes from this other study, where they analyze a set of 250 families. That they would have spurious translations of what their proteins would be, which in this case is known as the ANTIFAM database. So, since they are spurious proteins, we would not think that their predictions with ANFAFOL had high confidence values of PLDDT. However, it turns out that when we look at precisely which sets of proteins have good PLDDT, we found a few dozen proteins that can be relatively predicted with a considerable level of confidence, between 70 and 90 in the PLDT value, which is evidently not consistent with the idea that these proteins are spurious. However, it is quite interesting that AlphaFold shows this behavior, particularly with proteins that are less than 100 receipts in total. So what is suggested, at least in this publication, is that we must be particularly careful when we are modeling relatively small or even small proteins with the AlphaFold algorithm, because AlphaFold is not so sensitive to be able to detect how plausible the structure of this type of shorter proteins would be, and that as they become larger, we can think. Each of the residues imposes a greater set of limitations in order to satisfy the structure, and therefore is more analytical in this sense. But since the number of residues is reduced precisely, it is not as reliable in this sense, which is precisely what we see in this diagram, where what is graphed is the sequence length with respect to what the PLDDT values would be. And precisely, the black line shows us what a cut of 100 amino acids would be, where we can see that the majority of these antifam proteins are predicted between 50 and 70 PLDDT values, and a few dozen between 70 and 90. And when we replicate this analysis now, using completely random proteins, we see that AlphaFold also predicts them in a range of between 50 and 70 PLVT, considering them as medium quality or almost low quality. But because they are random, or in this case they were spurious like the antifan, we would not expect that type of behavior from AlphaFold. We would then like it to be more sensitive to be able to detect spurious proteins, which in this case is not as likely when they are small proteins. A second paper also suggests to us that AlphaFold2, but not AlphaFold3, is capable of making high-confidence predictions for structural repeats, which we can see precisely in this diagram, where what they do is precisely take small segments of sequences, and then we repeat them to see what types of proteins result, which would be solenoids, highly symmetrical structures as we could see in this sense. And it turns out that when we observe what type of residues are found in the hydrophobic and hydrophilic part of the protein, it is quite implausible to find what are residues that we usually associate with what is the surface due to its hydrophilic behavior. In this case, we find lysines, aspartic acids or histidines, which are buried in the hydrophobic core, which does not happen because amino acids of a more hydrophobic nature constitute the core of the proteins. So then that would also be like a second example that even though there are proteins with good predictions in terms of PLDT, we can find implausible patterns in this case, and that this type of analysis is really necessary to be able to, well, explore or validate AlphaFold's structural predictions. Something that in that sense was also relatively atypical when taking only repetitions and duplicating them a greater number of times, and that perhaps does not reflect as much as happens in terms of a protein that we find naturally. But obviously this should not happen if AlphaFold understood the biochemistry of proteins completely well. A third example that also caused a lot of noise in the structural biology community is this other one, where the authors, a group of structural biologists, actually suggest that AlphaFold's predictions are quite valuable hypotheses, which even accelerate structural biology, but which are not capable of precisely replacing experiments by X, K, and M-ray crystallography, among other types of structural analysis. And so here what we observe are the electron density maps associated precisely with AlphaFold's predictions. As we can see here on the right, what electron density maps do is precisely position the location of each of the electrons, of each of the atoms of our side chains, or of the protein in general. And in this case, the better the resolution we have, obviously the positioning of those atoms will be more accurate. So then resolutions of approximately 1 Armstrong, well then they are quite good. As we can see, we could even determine the presence of what would be side chains in a fairly detailed way, and that at least in terms of 2 Armstrongs, which would be the resolution where AlphaFold's capabilities are found. Well, we did not find this level of detail, but we did find a very good level of detail that could well delimit what the side chains would be, right? At 3 Armstrong resolution, we obviously lose more detail, but it is still a good coarse-grain representation. And at 5 Armstrong resolution, we really lose a lot of information, and that in this case would perhaps be mainly useful when we are talking about large macromolecular complexes with hundreds of peptide chains, which in this case being such large proteins is quite difficult. It is sensible to expect that they cannot be resolved to levels as exquisite as an Armstrong resolution would be. And then precisely what they do in this publication is compare the electron density maps of what the real models would be with respect to their predictions with AlphaFold, and finding some behaviors as in the case that they do overlap in a relatively good way, with approximately a deviation in RMC of approximately 0.5 Armstrongs, where only a few side chains come out precisely from these electron density maps. We also find cases where there is a complete discordance between both types of configurations, so that when we assume the electron density maps, we observe that in the case of AlphaFold, precisely some chains are completely removed already, in a more continuous way. And a third behavior where, although there is not a complete coincidence between what would be 2 regions of electron density, it would be plausible in structural terms, because perhaps they would be alternative conformations that would be explored using the AlphaFold2 algorithm. So then, to generalize the conclusions of this research, what the authors suggest is that between 7 to 20% of the side chains for proteins that have a PLDT greater than 70%, they may be incompatible in terms of what would be the data obtained by cryo-ion or X-ray crystallography experiments. And in general terms, we found that for proteins with a PLDT greater than 90, what would be the error of their side chains in terms of electron density could be approximately 0.6 Armstrong resolution, finding approximately. In general terms, a deviation of 2 resolution ranges for a total of 10% of the residues that make up this type of protein. So then, when we are carrying out analyses at the level of side chains, because we are interested in characterizing much more information related to, for example, the binding site, the catalytic site, among other properties that require precisely the analysis of the side chains. It is therefore advisable to work with proteins that have a PLDT greater than 90%, desirable perhaps 95%, and perhaps when we are doing this type of analysis with proteins with a PLDT less than 90 or even less than 80, depending on the context, we should be much more careful regarding the observations that we can make with this type of models made by AlphaFold. So well, it's also good to really think. What is AlphaFold? How can we represent it in our heads? So with this, we address 2 opinions from colleagues who work precisely in the area of structural biology and also application with deep learning, such as the opinion of Frank Noy, who suggests to us, at least speaking regarding AlphaFold3, but also extends to what AlphaFold2 would be, that these types of algorithms are what they are structural prediction methods. They are not emulators of physics. And obviously when they are trained with data from the PDB as well as from UNIPRO, it is clear that they learn statistical patterns that could be correlated in physical, chemical, or in general biological terms, but that were not explicitly trained to emulate these physical conditions necessary for them to exist in reality. As a second opinion, we have this other one from Mohammed Al-Qureshi, which I really like a lot, because at least from my personal perspective, it helped me a lot to understand what an AlphaFold structure is. That in this case, both AlphaFold2 and any other method such as AlphaFold3, Rosetta Fold, bolts, among other algorithms, we can think precisely of this type of structural predictions not as a structural state associated with what would be the minimum energy under physiological conditions, but as a structural state that is more likely to have crystallized under specific experimental conditions. What does this mean? Well, AlphaFold was trained mainly with the PDB data, and the PDB data really has a bias towards proteins that were easily crystallizable, because all the proteins by all the groups that investigated that could be crystallized and that turned out to be failed attempts, since obviously that information is not deposited in the data link. So there would then be a bias in the PDB of proteins that are more likely to crystallize under a set of, for example, specific solvent conditions or specific expression systems, mainly associated with E. coli, among another set of information that could really be biasing the type of information present in the PDB, even though we are talking about quite diverse proteins. In fact, this is something else that we are going to address in detail in what is the last class of the course. Or in general, the conclusion is that a model is as good as the data with which it is trained. And although it is evident that the PDB data is of very good quality, really every database has its biases. And it is our job to reflect on these biases, to try to mitigate them as much as possible in this sense. And here are precisely observations that come from other colleagues, who even also recognize that precisely when this type of algorithms are trained, mainly with globular proteins, they have a greater tendency to overpredict structures that are structured. We even have another bias, for example illustrated here with this bar graph, where we find the type of techniques that were used to resolve the 250,000 structures deposited in the PDB, where it turns out that about 80% of the structures were determined using X-ray crystallography techniques. So perhaps we could think that AlphaFold2, and in general all the algorithms that use the PDB structures, would have a statistical representation of the patterns they learned, a little more enriched towards the type of structures that can be obtained with. Because these types of structures are overpopulated here, and after all they are signals that could perhaps be biasing the behavior of the algorithm. Despite these biases, the community has also adopted what AlphaFold's predictions are, to be able to incorporate them into their experiments, so that in specific cases where there are precisely no previous structures, we can now take AlphaFold's predicted models so that the work is reduced both in terms of time, effort, and money used to be able to precisely determine some macromolecular complex. In this case, both for X-ray crystallography and cryo-EM methods, what is done in general terms is to take models of a desirable quality to be able, in the case of X-ray crystallography, to enrich the data obtained from the diffraction of the data, or in the case of cryo-EM, in what is the refinement model in this phase of the electron density maps. In general terms, we could see it as if we have a huge macromolecular complex, and in that macromolecular complex, we find a certain little hole associated with a protein that we know. Well, we can predict precisely the protein that we know and see that it also fits in terms of the electron density in that little hole, and then see if the macromolecular complex then makes sense in its entirety when integrating this new prediction. However, it is quite clear that this cannot occur in all cases, but certain requirements must be met in terms of the quality of the models, so that, for example, in the case that we are working with large macromolecular complexes, it is not recommended to use this type of AlphaFold models when they are resolutions greater than 6 Armstrongs in general. And this has ended up in quite famous publications, such as the structural characterization of the nuclear pore of eukaryotes, which was a structure that we did not really know the details of previously, and that thanks to AlphaFold models together with cryo-EM techniques and other analyses, it was possible to determine its three-dimensional configuration, which really allows its characterization a little more detailed with respect to how it is, that several molecules can precisely access the nucleus of eukaryotes. Another aspect was that also during the launch of AlphaFold, when it really became open source to the community, obviously everyone decided to tinker with the algorithm and see what capabilities it would have. And in this sense, two researchers independently observed that if we added, in this case, a binding or linker sequence composed of simple residues such as glycines or even alanines, which probably did not represent a tendency to form secondary structures, or even by extending what would be this index residue at the time of tokenization of the AlphaFold representations, we could even model multimedical protein complexes. As you can see, simply by adding this linker, we can join what would be two proteins that should be a complex, and in this case it turned out that AlphaFold could predict protein complexes, even though it was really designed to predict monomers. And then, due to these observations, the DeepMind team decided to invest even more resources to retrain their version of AlphaFold, now specifically to improve its predictive capabilities for protein complexes, in such a way that new terms were integrated into the loss function. New terms in the architecture, as we already reviewed, not as drastic, but obviously more sensitive to be able to really capture the information of the protein complexes, so that now we have a second version of AlphaFold, which is called AlphaFold Multimer, which precisely specializes in the prediction of protein complexes, as we could see in this bobbin, which is on that left, which would be the structure obtained through experimental methods. And on the right, what would be the structure predicted with AlphaFold, colored by PLDDT, or in the center, the same structure predicted by AlphaFold but colored by chains. And what we observe is that, although it is a fairly large macromolecular complex, well, its predictions are actually quite good, and perhaps they would allow us to characterize complexes of another type that perhaps we did not know about, in that, thanks to this algorithm, we can now save both time and money to be able to characterize this type of macromolecular complexes. And just as a curious fact, when you check what this AlphaFold multimer preprint is, it turns out that the DeepMind team cited their tweets from the researchers we saw previously. Therefore, it is also highly recommended to have a certain presence on social networks to find out what is happening, and perhaps through some interaction with the community, your tweets may eventually be cited in the odd publication. Another series of analyses regarding how AlphaFold operates in its predictions falls on the part of this group, or consortium called OpenFold, who decided to retrain the entire AlphaFold architecture, using in this case PyTorch and other tools that help its optimization in terms of the energy cost to be able to execute the algorithm. So they then released a reproduction of AlphaFold2, which is also open source, and which really facilitates what its development would be in algorithmic terms. And it is expected to do the same for AlphaFold3, which has already happened, and even for future versions of AlphaFold such as AlphaFold4 or perhaps 5. And perhaps the most interesting thing about this publication that I highly recommend reading is the fact that by having greater freedom regarding precisely the training approach used to train these types of algorithms, well, we find information more related to what the patterns that are being learned would be. For example, what is easier to learn? Regions of beta folds or alpha helices? At what point do secondary structures begin to be learned? What would be the importance of templates in general for AlphaFold? So all that information can be found precisely in the publication, and I would think this is a must-read if you want to learn about AlphaFold. But maybe the two most relevant observations, at least from my perspective, are these. The main one is that 90% of the model's accuracy can be achieved, with only 3% of the training time, which allows us to train such good algorithms if we obviously do not have the computational resources that the Google demand team did have, which in this case is a massive computing center, and that obviously it is not as accessible to practically the entire scientific community. And as a second observation, we also have that AlphaFold in general has two learning phases. One that is approximately 5% of the total training time, where what you do is learn to derive evolutionary information from the alignment. And another second phase equivalent to approximately 95% of the time, where they acquire a certain physical understanding of the proteins that allows them to precisely rank them as the best hypotheses and obviously the best hypothesis. Give it to us as the structural prediction that corresponds to our sequence. And here what we observe is precisely a map that reflects this idea, where what we have is on the x-axis the training time, and on the y-axis the quality of the predictions evaluated using the LDDT. And then we have different regions, although in general there are two, precisely this first phase where it learns to derive coevolutionary patterns, and this second pink phase where precisely it acquires this physical understanding of proteins, and a last one where precisely the behavior of what would be, for example, the activation of violation losses, of characterization losses at 3 amperes resolution for very good quality proteins, among other aspects, is refined. And it turns out that when we zoom in to be able to evaluate what type of proteins they are predicting throughout these training phases, the type of patterns we observe is quite characteristic and important, because here as we observe in this first stage the increase is quite considerable. However, now here we find a plateau where perhaps it is not really being reflected in an improvement in structural terms, but it turns out that it is. Since it is precisely in this specific region, here we observe a window of 2,000 training steps up to 12,000, approximately equivalent to this region, where we now find here on the y-axis another metric related to which the structures are also being predicted. And we find that in this first plateau or this specific region, there is actually an increase in the predictions in terms of their quality until we reach precisely the training term where the predictions are practically being refined in their entirety. And this is thanks to the integration precisely of both the training and fine-tuning stages of AlphaFold2, where although it seems that this entire training range is monotonous, in reality, what AlphaFold is doing is refining the structural understanding of proteins in order to reach a final level here established by this box and whisker diagram, where we could find a correlation of approximately 0.9, considered predictions of very good quality in this sense. Research has also been done regarding the interpretability or explainability of the AlphaFold algorithm, so that even here what is suggested is that AlphaFold learned a kind of energy function that allows it to evaluate this agreement or coincidence between the sequences and structures. However, it requires alignment information or template information in order to access what would be the search for the global optimum. What does this mean? As you can see here in the diagram, really when we give a sequence to predict, AlphaFold must be able to identify what patterns we could associate to establish a structural hypothesis. That is, what structure said sequence could correspond to. So we can then pose this problem in terms of an energy landscape, where high-energy conformations, not thermodynamics, but energy seen as a statistical potential, since they are very unlikely structures, perhaps precisely because they are disordered, and only a very small set of proteins could be those that are folding correctly in three-dimensional space, perhaps associated with what would be their native state in physiological terms, but as we previously reviewed, not necessarily. And in this case, the contribution of the MSA, or rather of the information provided by this entire set of sequences, is really to serve as a kind of seed in this entire process of searching for a specific pattern. So now we can state what the structure prediction problem is in terms of a search for a global minimum in this energy landscape. And precisely what the information provided by the alignment does is tell us, hey, focus only on the structural hypotheses that belong to this region, because we use precisely the MCA as a seed during this global search. And then it is the structural module which through these 8-layer cycles, as well as the recycles that you can use, since it is now responsible for determining locally what the best structural hypothesis would be, which is reflected in terms of what are precisely the confidence metrics that AlphaFold provides in this sense. So, the most probable structure in terms of what the structural module found are those that precisely reflect a higher degree of certainty in terms of their confidence metrics. And this has two implications. The first is that we can then use precisely these confidence metrics, which the authors here refer to as this learned function, to be able to precisely evaluate structures without the presence of a sequence module alignment, because we then really developed an understanding of what the energetic conditions would be to be able to precisely select the best candidates. And the second implication is that precisely what alignment and structural templates do is eliminate false negatives by precisely providing a seed, a starting point during this process of optimizing structural hypotheses. So we are going to explore in a little more detail what they do in this article, but obviously for more details, I still recommend consulting it to know all the information. What is relevant here is to really know what we give as input to AlphaFold when we give it a template, a structural template. Three main inputs are obtained when we give a template. The first is what would be its amino acid sequence, represented by a one-hot type vector. The second input is a distance matrix from its beta carbons. And the third input is the pi-psi angles, as well as the chi-psi, which are precisely related to what would be the backbone and the side chains. So here we are going to address what the authors did to reach these conclusions. In total, what they did was analyze a set of structures called the Rosetta dataset, composed of approximately 133 structures, each of which has several hundred alternative conformations which we call decoys. And then the procedure to follow within this framework was point number 1, to take precisely the sequence that we want to predict and give it a decoy as a template as alternate information. Then we disable the use of what is the MSA, so that AlphaFold does not have this information as a seed, and now the decoy information falls precisely as a seed for the exploration process in the global search for candidates. However, then we must even modify what would be the information provided by this decoy, because we already saw that there are three main inputs that are derived from the templates, in such a way that now we replace the amino acid chain with a series of gaps, and we also hide all the information of the side chains, except for what the beta carbons are, since what are the beta carbons are what AlphaFold uses to be able to calculate things like histograms. And we hide as much information as possible so that, let's say in simple terms, we only have the minimum necessary information related to the decoy, or in general to the backbone of the decoy, so that it serves as a seed in the global search for what would be a structural hypothesis. We then ran the AlphaFold algorithm, then calculated precisely these confidence metrics, which in this case was a product of the PLDDT, the PTM, and the TM score, to aggregate precisely the main trust metrics generated by AlphaFold, and then we use precisely these trust metrics to rank the best candidates, which is precisely what we observe here in terms of the TM score and in terms of the generalized trust of AlphaFold, and that we observe precisely that what would be the Spearman correlation of this type of strategies is established at approximately 0.95, and that when we compare it with other approaches, well, if they are quite diminished in their abilities, what could be this other neural network called deep a CNET with approximately 0.83, or even the energy calculated by Rosetta that we previously investigated, which finds many statistical and physical patterns of the proteins, it ranks with a correlation of approximately 0.76. That is, AlphaVolts really has abilities to be able to discriminate between structural hypotheses, so that we can see here how these ones to which it assigns less confidence, and which are also reflected with a lower number of team score, are really predictions that have a low PLDDT, and that the best candidates, such as this other one at the end of the graph, really have good PLDDT characteristics, which are reflected both in the generalized confidence and in the GM score of the protein. So then, tentatively, AlphaFold did indeed learn to discriminate precisely between structural hypotheses through this conceptualization of a statistical energy. Be careful, non-thermodynamic statistical energy, which is quite important to understand in this sense. And obviously using what would be these abilities of AlphaFold has also been applied to its use in what would be in the design of proteins, or inverting the architecture, so that it was now of a more generative nature similar to ChatGPT. As we observe, this would be a representation of what the architecture of AlphaFold2 is. In this case, we see that we have to create the alignment from our query sequence, and then we can also use templates, the MSA. And then we go through this first module, which would be the Evoformer, which also gives us a histogram as input. And then all this information is passed as input to the structural module, which gives us the atomic coordinates of each of the atoms of our proteins. What it also gives us is confidence both at the level of each of the residues and between pairs of residues. And then researchers started asking, can we then invert this architecture so that it then serves as a generative model? In this case, what is done is to take a structure and then compute what its histogram would be, and then if AlphaFold gives us a histogram, then now we can condition it to reproduce the histogram that we are giving it as input. And then thanks to what the gradient backflow is, we can generate a feedback loop where AlphaFold is now capable of giving us a sequence that becomes capable of representing or approximating the histogram patterns that we are giving it as input. And this is how the main protein design approaches were developed with a more tinge of hallucinations or generative nature based on AlphaFold, to generate both new sequences and new structures capable of precisely satisfying a previous structure as a seed. And these approaches were explored at least since approximately 2023, 2022, with quite notable advances in the field of protein generation and binding to other proteins that are known as binders, where we found precisely protocols that used this type of generative or hallucination approaches with the AlphaFold architecture to design new structures capable of binding to other proteins. And obviously, they serve as therapeutic targets for a multitude of diseases. And something quite relevant or interesting was really, in experimental terms, the success rate that you find with these types of approaches. So that when we design binders marked here in green, using de-learning algorithms, the experimental success rate in multiple cases, as you can see in each of these, it increases considerably compared to the generative methods that we would have with other algorithms that are more physics-based, such as Rosetta, which really showed the community that it would be even more worthwhile to now focus the research on what it would be like to inject information, biological, physico-chemical, evolutionary, biochemistry in general, to this type of deep learning algorithms so that we could now use them in a generative way, because their success rate is higher than the previously developed methods based explicitly on physics. And evidently this developed and reached one of its maximums, in my opinion. Obviously, it is no longer the best in current terms, but it is a widely used tool, which is the Bingrath algorithm, which was precisely used by AlphaFold, because one of the observations made during the training of AlphaFold multimer is that, perhaps during the training, since this algorithm learned precisely which residues have a greater propensity to form precisely interactions between pairs of proteins. And exploiting this observation was that they then decided to use it because if it detected this type of residues to precisely establish interactions between proteins, Then now we can exploit this to generate binders in this sense. And so this is precisely what they did. In this case, the algorithm is relatively simple. What you do is take the architecture of AlphaFold to multimer, and then give it precisely the indication to generate a sequence capable of binding to a target protein. And then you run the AlphaFold algorithm using these gradient backbending approaches along with another type of built-in loss function to make it more sensitive in terms of the secondary structures generated by these binders. And then the structural hypothesis that Beamcraft gives us. It is refined by another algorithm that maximizes its solubility, an algorithm called protein MPNN, in a solvent way, and then optimizes a sequence for us, which obviously corresponds to a structure that we can still predict. In this case, AlphaFold2 is used in its monomer form to validate this structural hypothesis, and it turns out that these proteins generated through this framework called Minecraft, they turn out to be quite good in experimental terms. How good? Well, here is what we see precisely. With the success rate we have using Beamcraft, we have rates between 10% and 100%, with an overall success rate of 46%. And this is quite shocking when we look at the success rates we had with physics-based methods, such as Rosetta. Because the rates of those types of algorithms were around 0.1%. So going from 0.1% to a minimum of 10% is really quite shocking. So we can even see here in this representation of what the generation of a binder would be in the original. What would be the protein already crystallized in pink, and the binder predicted by AlphaFold in gray color, and we see that the structures of both the prediction and the already crystallized one really share an RMSD of 1.7 Armstrongs. So we can say that it is of very good quality because it is in the range of approximately 2, and that the prediction made by Minecraft actually resembles quite well what the determined crystal would be. So it is not only something that was left in bioinformatic algorithms, but that the authors even took on the task of demonstrating it with multiple crystals to precisely corroborate that Beamcraft's design capabilities for generating binders are really quite good. And well, you will also wonder then, what happened in the next version of the CASP? Because AlphaFold2 was launched in what is CASP 14. However, years passed, and evidently CASP 15 was also released. And something that we can see is something quite funny in these two reports, where due to the success that AlphaFold represented in terms of solving the problem of monomers, in terms of predicting their structures, now the CASP opened new facets in what this competition would be, in such a way that now new areas of prediction are included, such as the interaction of biomolecular complexes, such as interaction with RNA structures, interaction with ligands, of oligomeric complexes, or even what would be conformational assemblies of proteins. And this was promoted thanks to the success of AlphaFold, where we are now advancing more and more in what would be the tasks addressed through the CASP. And this is something that is precisely represented in another very funny way in this type of graphics, where we find the best competitors in this CASP 15 competition, where it turns out that practically the majority of the groups used variants of AlphaFold, either enriching the alignment, disturbing it, or making slight modifications, but all absolutely using the AlphaFold algorithm. And perhaps only here, as almost at the end of the queue of the best competitors, do we find, for example, algorithms based on protein language models, such as the protein language models of the ESM family, which are found in this other region. And only the best, as you can see, used AlphaFold, AlphaFold, and AlphaFold. So then, when we make an analogy with another experiment, we find precisely another experiment that was quite famous at the time, which was the AlexNet moment, a competition that in this case was equally similar to the CASP, which in this case were researchers within the world of image classification, who developed a competition to precisely propose different deep learning algorithms, or not necessarily deep learning, but in this case it was Alex Krishkibensky, Ilya Sutskever and Geoffrey Hinton, who developed the idea that we could adopt convolutional neural networks to be able to model images. And it turns out that when they entered the competition, together with their algorithm that was called AlexNet, they managed to demonstrate that the use of neural networks was a way forward with quite interesting results, because the error rate found in terms of image classification using AlexNet was considerably reduced, as you can see, from about 0.3 to 1.5 overall. So we could say that an analog of what the AlphaFold effect would be is really what AlexNet is. So that then AlexNet, as it had a great impact in terms of artificial intelligence in general, as it demonstrated that this type of algorithms based on deep neural networks is actually a very useful tool. It made the entire community focus on research into this type of algorithms. And then, when we evaluated the context of AlphaFold, it really generated the same thing. It made a large part of the biological research community now focus on the research of algorithms based on deep learning, to be able to model their biological phenomena, something that both AlexNet and AlphaFold did, since they really caused great havoc in the community in terms of the perspectives it has in terms of research. And that is how we then arrive at AlphaFold3 with new perspectives regarding what these types of algorithms based on deep learning can do. And in particular, in the case of AlphaFold3, we can think of the proteins as a set of atomic trajectories that in this case are represented by this small diagram where the original atomic coordinates have to be, let's say, corrupted and returned to their original state. And during this process, the neural network is the one that learns precisely to generate the atomic coordinates that constitute this particular structure. And in that sense, what is intended is precisely to extend the capabilities of AlphaFold, not only to be able to predict what protein complexes are, as was done with AlphaFold2, as well as some other adaptations that sought to transplant what would be the ligands to protein structures. But now AlphaFold3 aims to model biomolecular interactions, that is, protein interactions with nucleic acids, with small ligands, as well as with metals, and in general, what can be said of any molecular entity. However, here AlphaFold3 also represented a turning point in the history of protein science, because in this case, the models are no longer accessible to the entire scientific community, but are now under a restrictive commercial license, which is why their use is not widely facilitated to be able to adapt to multiple scientific analyses. But this time there is already a commercial interest, and this has unfortunately been reproduced by several organizations that have trained their own versions of AlphaFold3, and that they precisely seek to obtain some benefit from their trained algorithms. So let's see what happens in the AlphaFold3 universe. And in particular, we first have to recognize what AlphaFold failed to model. In this case, it is quite clear, as we reviewed previously, that AlphaFold does not model interactions with nucleotides in general, nor interactions with ions and oligons, post-translational modifications, RNA complexes, with complicated proteins such as antibodies or membrane proteins. So, in this sense, AlphaFold was a victim of its own success, because in order to generalize this type of interactions, they had to practically get rid of the great architecture that they built in AlphaFold2. And in particular, one of the most relevant properties, which is precisely equivalence. So this is why the title of equivalence is dead, long live equivalence. Because it is even properly cited in the FAFL-III article, they found that this property, in terms of rotations and translations, does not seem to be required to predict biomolecular complexes, and that even the great weight that was the evolutionary representation provided by the multiple alignment of sequences, since it seems that then in AlphaFold3 it was completely left aside, because the great weight that now falls on this new algorithm is in the representation of pairs of residues, and not so much in the representation of the MSA as happened with AlphaFold2. So we are going to explore in more detail in this sense how the architecture had to be modified for AlphaFold3, and generalize precisely to biomolecular interactions. In this case, it is evident that both the structural module and the leviform module of AlphaFold2 had to be reinvented to precisely extend its capabilities to new forms of biomolecular interactions. In particular, let's see what AlphaFold is capable of doing. Well, it is quite clear that it can predict the structure of proteins in the form of monomers, which is relatively trivial, fortunately for recent times, and that even when we see reports related to the results of the CASB, or at least the latest version of the CASB, there is even talk then of what the prediction would be at the monomer level, or a single domain for a protein. It could be considered as a task that is almost solved, except for some particular proteins, precisely such as antibodies, some viral proteins, or those proteins that precisely do not have a sufficient number of homologs deposited in the databases and that are more like the only representatives of their protein families. These cases are precisely where algorithms similar to AlphaFold3 fail to estimate the structures. But where they can extend their capabilities is precisely to model interactions with nucleic acids such as DNA molecules. They can also directly predict the structure of RNA molecules. They would also be predictions of proteins with small ligands, whether, for example, ATP molecules or even quite large groups such as the tetrapyryl group of what are chlorophyll molecules. You can also modify interactions with small ions, perform post-translational modifications such as phosphorylation, which, as we already saw, this type of modification can really have a great impact on the three-dimensional configuration of the protein. Additions of sugars can also be made due to the glycosylation of the protein, including changes in the chemical bonds that may exist in the protein, forming different arrangements that can also be covalent or non-covalent in nature. So we can see that thanks to sacrificing what would be the equivalence provided by what is the structural module and the Evoformer, we can now extend the capabilities of AlphaFold3 to practically a great diversity of biomolecular interactions. And fortunately, in this sense, Google DeepMind also decided to enable what is a web server so that, again, if we do not have great computer skills, we can simply access what this server is and upload our sequences, our ligands, or practically any type of molecular interaction and send it with a few simple clicks so that the Google DeepMind server can make the prediction and have it in just a few minutes. Here as a reference, they have some proteins or biomolecules in general regarding how long they can take and how many tokens it costs precisely to model it. For example, an archetypal protein of approximately 300 residues considering it is a monomer may take between 2 to 3 minutes or even less, because the number of tokens is approximately 500. But obviously when we try to model a much larger macromolecular complex, the prediction can take between 8 to 12 minutes, costing up to approximately 3,000 tokens, because on this occasion it is tokenized not only at the residue level but also obviously the entities that are not properly proteins will have to be tokenized at the atom level, an aspect that we will review in more detail below. However, another much more relevant thing when AlphaFold3 appeared was that in this case it is no longer a completely open source model, where there is both academic and commercial freedom to be able to precisely develop the algorithm both in research and in extending its capabilities. In this case, it is quite important that you consider what type of research you are doing and what would be the ultimate goals that you would aim to achieve, because when you are modeling, well, your proteins with AlphaFold2 are clearly not going to have any problems because the license allows it. Analysis can be carried out practically scientific or of a more private industrial nature. So in general, it can be said that the AlphaFold2 algorithm can be applied to any activity, even commercial ones. However, when you're talking about AlphaFold3, things change. Here, the license is already more restricted, and even the authors themselves suggest that it only be used for what is fundamental academic research. However, if eventually your research results in perhaps a patent, and throughout the characterization process, you use the algorithms related to AlphaFold3, then there may be a little more complications regarding the assignment of said patent and its subsequent attempt in commercial applications. So this is also a new idea that they have to keep in mind regarding what type of algorithms they have to use based on their interests, whether academic or even commercial applications of their discoveries, characterizations and findings, etc. And well, first, we also have to look at what was happening in the research environment around AlphaFold3. During these times in particular, image generation models such as Midjourney, Flux, Dolly, or Stable Diffusion were having a huge boom, and that even spread thanks to what was OpenAI's Sora model, which was already possible to extend not only the ability to generate images, but now generate videos for a few seconds, which, as you can see here on the right, there really are quite interesting results, such as in this interpretation of a Dayhoff photograph, only in this sense with a minimalist watercolor style and with a bioinformatics background. So we can see that this type of algorithms based on diffusion models had great success, at least at this time. In this time window where AlphaFold was developed within the Google Bitmain laboratory. And these types of diffusion models, they are based on what is known as the heat equation or diffusion equation, which practically tells us how a system evolves over time. In this case, the diffusion of heat obviously applies to temperature, but it can really be extended to practically any type of system. It can even be applied, let's say, to what it is, systems like proteins, systems like music, systems such as images, among many other aspects, which in summary what they do is suggest to us how we start from an initial configuration with respect to the elements that said system contains, and how these elements are dispersed and blurred over time to reach a new configuration that would represent a second state, a third state, etc. This is the basis that diffusion algorithms are used to be able to precisely model the dynamics of any type of systems. And here a small disclaimer is the fact that, due to my limits of understanding, we are not going to cover in detail how diffusion models work, because they are terribly complicated, as you can see in this meme, because, well, a scientific article related to whatever a language model is, which, as we already explained with what the transformers and large language models class is relatively simpler to understand than when you delve into the formal mathematics of any scientific article that deals with a diffusion model. Yes, it is true, it is really complex mathematics. They are usually always handled with systems of stochastic differential equations, and you can even see it reflected a little in what would be the supplementary material of these two articles, which in the case of AlphaFold2 we are talking about 62 pages in total, and in the case of precisely a diffusion model such as one called Chroma that we are going to investigate later, we are talking about 123 pages with quite dense mathematics. Here I make the disclaimer of responsibility, that in this case we are not going to address it in such detail, but rather we are simply going to address it in a way that is easy to communicate. In this sense, if you are looking to understand in more detail how we can interpret and understand this type of diffusion models, I highly recommend this series of 3 videos that you can find for free on YouTube and thus go deeper into precisely the architecture of this type of models, which can even become a little more complex because they are not only diffusion models in their most simplistic form, but now diffusion models are combined with the transformer architecture or even with graph neural networks, so it can even become a little more complicated if they do not previously know precisely how this type of architecture works. So let's see in a very summary way how precisely these types of diffusion models work. The idea is to try to model a data distribution that represents the object that we want to model. In this case, we're talking about cats, so we are precisely trying to approximate a distribution of cats. So we can take this brown cat as an example here. And then the idea of this type of diffusion models is that precisely what is known as Gaussian noise is added, in such a way that in each of the noise addition steps a completely noisy image is generated that could be interpreted as a sample taken from a Gaussian or normal distribution in this sense. And precisely what these types of language models aim to learn is to eliminate with each of these steps the noise that has been previously added or that has been conditioned, which in this case is added through a normal or Gaussian distribution. So once we start from what an image, let's say, is completely corrupted, the model will have to be able to extract this noise, eliminate it to recapitulate an image, which does not necessarily have to be the same as the original image from which it was based, but it does have to correspond to the distribution, in this case of cats, which in this case we can see reflected in a cat that is now orange and now has an eye patch. So this is, let's say, the general idea of how these types of algorithms work. However, this is precisely with images, so that it can be more easily understood. But the idea is that we can also extrapolate this to practically any type of systems, such as the world of proteins. Again, here the idea is to start precisely from the atomic coordinates of a protein to a bimolecular complex, add noise, and eventually arrive obviously at what would be a normal distribution that, let's say, disturbs or it corrupts what would practically be the original coordinates of each of the atoms of the positions of the residues of our macromolecular complex. And again, the idea is to try to eliminate this noise little by little to recapitulate the distribution of the proteins. The distribution of only alpha or only beta proteins, or of proteins that interact with ATP. In general, let's say, many types of distributions that represent some specific protein or some specific type of images, if you want to interpret it that way. And the most relevant thing about this type of diffusion models is that they are very easily conditionable to multiple types of data here as we can. C is then the most typical form of training of what this type of algorithms are, where we start again from completely noisy coordinates. These coordinates are usually precisely to arrive at what would be the representation of a biomolecular complex, in this case, a protein ligand. And here, as you can see, precisely what is injected is, or rather what Gaussian noise is shown. But within this type of distributions, multiple modalities can also be conditioned, so that that type of information is injected into the algorithm based on an attempt to reproduce the type of statistical patterns that represent said modalities. As they could be precisely graphs of ligands, information derived from sequence, histograms of beta carbons, therefore of a protein structure, learned representations, as could be precisely the representation of the MSA of Alphafold2, even well as precisely what would be physical-chemical bases, as could precisely be the verification of equivalence or correction, precisely of chirality or steric clashes. So this is one of the benefits that this type of diffusion models offer, and why many people interested in this type of algorithms began to develop them more and more, which is precisely reflected in what would be the AlphaFold3 algorithm. Even one of the advantages that this type of diffusion models offer us is that when we have quite limited data, and not as extremely diverse as text-related data might be, as training an algorithm like ChatGPT might be. As would be the case in biology, we usually have very little data, because this data comes from experiments and is usually not as diverse as we would like. Well, it turns out that in these data regimes, or these particularly precarious data conditions, it turns out that diffusion models are relatively good in terms of what the validation loss would be. And here the only limit that limits us from being able to precisely apply this diffusion model is having sufficient computing capacity to be able to develop them. So let's review what we're seeing here in these two particular graphs. We are seeing what a model that was trained is in each of these points. To approximate what its validation loss would be with respect to what would be the number of parameters of the model, as well as the number of epochs with which it was trained on the y-axis, it is again the parameters, and on the x-axis, it would again be the epochs. But in this case, we are talking about diffusion models. And the idea of this preprint is precisely to try to investigate which of these two types of algorithms can serve us best when we have limited data, as would usually be the case in biology. So they then took a data budget, a dataset at most represented by 100 million tokens, and they trained different models with different numbers of parameters for different numbers of epochs to precisely try to approximate a mathematical model, such as this. One, isocyclines, which allows us to approximate what the validation results would have at each of these possible points that have been shown to us, to precisely know what type of algorithm could serve us best in matters of little data. And it turns out that when we train a diffusion model, or an autoregressive large language model with a single epoch, what is the validation loss is relatively lower in what is precisely the autoregressive approaches about what is a diffusion model approach. That is, here the validation is approximately 7.07, and in the case of diffusion models it is 10.65, so with little training. It would be more effective to choose what is precisely an autoregressive language model. But when we have more computation, and we can precisely train our algorithm for longer, and with more parameters, it turns out that diffusion models are more sensitive in capturing statistical patterns in data that are repeated as well as in those that are derived from limited datasets, such as those of biology. So here what we can observe is that with more epochs, the validation loss is usually lower in diffusion models compared to what would be the autoregressive models. In this case, you can see that here in the minimum loss found for the diffusion models, we have a loss of 3.55, compared to the minimum loss found in autoregressive models, which is 3.71. So in this case, it would be a good strategy to further explore how to apply this type of diffusion models in biological data that usually meet this condition, that they are not so large or so diverse. However, the limitation now is having enough computing power to be able to have this freedom to train much larger models, because here we are talking about an autoregressive model with approximately 150 million parameters. But in this case, we are talking about one with 420 million parameters for a considerable number of more epochs, 500, compared to what would be only 50 epochs of the autoregressive model. You could even apply this to your own research, considering both your computational limits and the database resources you have available, to try to model some dynamics that you are interested in. And again, I briefly explore that the application of diffusion models to model proteins, was not really an innovation provided by the Google DeadMate team with AlphaFold3, but was an idea that was previously explored by multiple other groups starting around 2002. In this case, what you have here is precisely a timeline of how the development of these diffusion models has been applied to protein science, where, for example, we recognize one of the most relevant preprints within the world of artificial intelligence, which is precisely the development of what was the diffusion models for a generative approach and then what would be its first applications to the world of proteins such as algorithms such as div-AP, folding div, among others, which first what they did was precisely model the coordinates only of the backbone, and then try to extend not only to the coordinates of the backbone, but to all the atoms that would correspond to said biomolecular complex. We are not going to address all of them in this particular class. We are only going to focus on perhaps the most relevant ones from my point of view, such as the Rosetta full diffusion algorithm, Chroma and Rosetta full diffusion all-atom. In this case, precisely what they intend is to increasingly extend their capabilities to now be able to generate biomolecular complexes of any kind, and not only biomolecular complexes of the protein-protein type. In this sense, what we observe here is precisely how Rosetta full diffusion works. And in this case, what you can see is precisely how all the coordinates are initialized within what would be, let's say, this noise and arrive at what would be, well, in this case, a number. Numerous protein structures that in this case were generated in a non-conditional way. That is, the algorithm is only told to run with this time, and the algorithm can suggest a great diversity of protein foldings, but the most relevant thing in the case of Rosetta full diffusion, which was one of the first diffusion model algorithms applied to proteins, is that it can precisely be conditioned to multiple specific aspects of the protein, such as, for example, conditioning it to comply with a specific symmetry, conditioning it to binding to a specific protein, conditioning it to the presence of a specific structural motif, or conditioning it again to some symmetry, such as degree 11 symmetry, degree 10 symmetry, etc. And then the diffusion model must be able to find precisely an atomic trajectory that is coincident with said condition to be able to generate, in this case, an interaction of the protein bundle type, or an interaction that includes precisely the structural motif. These conditioning ideas were later extended by what was this second article, where the Chroma algorithm is presented, which in this case was no longer only conditioned on these structural conditions, but now even conditions such as volumetric or even semantic from text were included. Obviously, they also included conditioning at the level of structural motifs or at the level of symmetry, but in this case conditioning was taken to the next level, even as I mentioned, including what is an indication by text. That is, for example, say, I want a protein composed of pure folded veins similar to team barrel, and the algorithm is able to understand exactly what characteristics are associated with this text with respect to a protein so that it can be generated. And we are not only talking about what was left in a bioinformatic test, but even that it has been led to experimental corroboration, as we could see in the case of these proteins or macromolecular complexes made in the form of letters of the alphabet, which would understand what volumetric conditioning is, in this case, as in the shape of a bunny, only in this case at the level of letters of the alphabet. And as you can see, what is the structure predicted by an algorithm, such as ESD unfold, has a shape similar to a D, a shape similar to an N or an A, and when we see and express precisely these proteins inside the cell to see if they are being expressed, it turns out that at my level of microscopy, we do observe these types of morphologies of the proteins, so we could say that they do exist. And that even this Chroma algorithm extended the protein generation capabilities to immensely large proteins of about 1,500 residues, which is extremely large. It is unimaginable that an algorithm is capable of generating so many residues, and that these residues are expressed due to the previous algorithms based on physics, since we are talking about small peptides of between 50 to 100 amine acids. And now with diffusion models, we can extend the generative capacities to around 1500 waste, which is really impressive in this sense. And even more shocking was one of the innovations provided by what is the Rosetta-Fold diffusion algorithm, but in its full atoms version, or all atoms however you want to translate it. Where in this case, they combined representations not only at the level of tokenizing the residues, but now the atoms were also tokenized. To try precisely to extend the biomolecular interactions, and that they were no longer only between proteins, but practically to any biomolecular entity, to understand what would be nucleic acids, metal ions, small ligands or covalent modifications, and that in this case, precisely Rosetta-Fold diffusion to LATOM was one of the first algorithms that demonstrated that it was possible precisely to generate this type of biomolecular complexes using the diffusion modeling paradigm. And that was precisely how we arrived at AlphaFold3, which temporarily, let's say, this would be the last article presented within this timeline, and that we can see, it arrived on May 8, 2024, and on this occasion we can see a great team of people behind what the AlphaFold3 algorithm is compared to what AlphaFold2 was with a team of approximately 31 authors. Here there are many more, and because it is obviously more complicated to be able to model this type of biomolecular interactions, they precisely needed many more people to be able to address this problem. However, this was not the only news coming from Google DeepMind. They also came with a new startup, which in this case is called Isomorphic Labs, which precisely seeks biotechnological developments at the level of medicines, oncogenic therapies, among many others. And here we can see precisely one of its first announcements in terms of raising capital to precisely start what is research with this new startup, where the person who is now, let's say, the scientific director within this startup is Max Genderberg. And what would be Demis Gazavis and John Jumper, since they continue to lead the coarse-grained projects within what is Google DeepMind in general, applied precisely in terms of artificial intelligence research applied to biology in general. And a second aspect that is also relevant to the arrival of AlphaFold3 is that, as you can see, the launch of the article occurred on May 8, 2024. However, in this case, it was not like in AlphaFold2, where the scientific article was released on the same day as the code on GitHub. In this case, it took approximately 7 months, including pressure from the community, for the AlphaFold3 model to be uploaded to this type of repository so that it would be more accessible to the scientific community's research, and thus precisely carry out their investigation to corroborate that these findings will be reproducible. And in this case, as I mentioned previously, the license is no longer open source, but is precisely limited to commercial uses, so only scientific or more academic research can be carried out, but its application. It is completely reserved to Google DeepMind to be able to extend it to patents or some other type of discovery of a similar nature. And during this period of time, there were also several groups, several people who tried to precisely reproduce the capabilities of AlphaFold3 by training their own algorithms based on what is the supplementary material of AlphaFold2. Sorry, from AlphaFold3. As you may remember, there were 7 months where we only had this information, and because Google DeepMind did not provide us with a code base with which we could start, multiple groups had to go through trial and error to find precisely a particular configuration that would allow them to train an algorithm similar to AlphaFold3. In this case, we can see a group called Legal Biosciences, who precisely by attempting to reproduce an architecture similar to AlphaFold3, found many discrepancies in the pseudocode present in the supplementary material with respect to what was known from the deep learning literature. something that was even found by another group, such as ByteDance, who may not know ByteDance at all, but they do know TikTok, which is precisely one of the companies that they are part of the ByteDance catalog, which is quite interesting how this type of business conglomerates also have interests within the application of biomedicine or technology in general, who released their own version of AlphaFold3, called ProteinX, and that again, through what is this search of trying to reproduce AlphaFold3, they found a series of errors during the training that when they executed their own versions, well, precisely they found that the model did not converge, that it was quite difficult to train it because perhaps it exploded or vanished from the gradient, or that there were simply errors, right? And then they even present a table with corrections regarding their particular implementation, compared to the supplementary material provided by Google DeepMind with their version of AlphaFold3. Even the scientific community, well, it was quite upset. Regarding these practices where the code is not being released, and we are talking about a scientific report published in Nature, something that in principle should be required of any research group to fully publish its code in order to evaluate whether the conclusions that are being made in the article are reproducible. In this case, by not providing the code, there is no access to this reproducibility, and it cannot be compared if what is said really by the publication, is it true or not? And this displeasure on the part of the community was reflected in a letter that was signed by approximately 650 scientists from around the world, where they precisely said that Google Bitmain should deliberate its models and its code to precisely know the replicability of this type of systems. And in this letter, it is precisely what is recognized. In this case, it was sent precisely to the editors of Nature, because it was the magazine where the AlphaFold3 algorithm was published, and where it is precisely recognized that AlphaFold2 was a milestone for the scientific community because it was an open source model that allowed, as we previously investigated in the community, to develop applications on this algorithm. For example, in extending its capabilities to be able to model protein-protein complexes, and that ideas were even shared, for example, on Twitter, which was reflected even in quotes, as we saw previously, in what was the preprint of AlphaFold2 multimeter. However, in this case, the community, again represented by approximately 650 scientists, to emphasize this large number of scientists, was quite disappointed by the absence of the code. Because this is a bad practice in terms of what scientific culture is, and that literally what it is doing is not being aligned with scientific principles, and blocking what would be scientific progress, which in general then we can see represented by this meme that, as we can see precisely, what it is an offer of what DeepMind gave us with respect to what DeepMind received from the scientific community, which in this case, Google DeepMind. Precisely, it received curated databases with values of billions of dollars, such as the PDB or the Uniprot. And in its first version of the AlphaFold3 web server, only up to 10 proteins could be modeled per day, regardless of the tokens they cost. You could send only 10 jobs to be able to model some biomolecular complex. And currently it seems to me that this limitation is only between 30 proteins or 50 proteins, if I remember correctly. If you are looking for more memes, not just gossip, you can even go to this thread on Twitter, where I also made a series of memes that I think reflected the feeling of the community quite well, because it was, well, a great impression regarding the capabilities that this type of algorithms can obtain, but a great disappointment to see these bad scientific practices, even boasted by many of us in the community, when in reality it is really bad scientific practice not to upload your code to be able to reproduce your findings. And it is with all this context that we arrive at this diagram that represents the architecture of AlphaFold3 in a simplified way. And here a key distinction with respect to AlphaFold2 is the fact that we do not operate with these local residue frames, but this time we operate directly on the three-dimensional coordinates of each of the protein atoms, that is, the x, y, and z coordinates of the three-dimensional plane. So in order to deal with that, it is clear that we have to make modifications at the architecture level. In this case, we have a first module. What it does is take all our inputs, sequences, structures, let's say, and represent them in embeddings. These embeddings will be given as input values to what would be the equivalent of the Evoformer in AlphaFold3. Only this time it is not called Evoformer, but rather Pairformer, because the calculation of tension is really more specialized than the pairwise representation, not the representation of the MSA as occurred in AlphaFold2. Furthermore, these initial embeddings will have to be enriched with two main modules, which is precisely the templates module and the alignments module, which respectively use the structural information as well as the alignment information to enrich the information contained in said embeddings. And both the raw embeddings, together with these enriched versions, they are given as input to what the Pairformer is, a total of 48 layers in total, which what they do is precisely distill all the information to create even richer representations in informational terms, and that in the end up giving us new representations at the embedding level that will have to be adopted by the diffusion module, which is in charge of generating these structural hypotheses that finally crystallize in the prediction of the macromolecular complex that we are trying to model. And again, this idea of recycling the architecture is adopted. That is, the outputs and representations learned during several cycles, so that the predictions are improved over time. And so we can now deal with a modeling of biomolecular complexes in such a way that we now extend the capabilities of AlphaFold2 to model only interactions between proteins, to model interactions with other types of biomolecules. And in this sense, we have some measures of the success of this new architecture, in this case using a benchmark called Post-Busters, which in particular specializes in the interaction of proteins with ligands. And specifically, we have on the y-axis a success rate as a percentage, and on the x-axis, a comparison of different algorithms capable of making these predictions of proteins with ligands in structural terms. We have AlphaFold3, Autodoc Bina, and Rosetta Fold Elatum, which is precisely this other version of diffusion that we previously reviewed, and we observe precisely that AlphaFold surpasses these two algorithms to be able to model protein ligand complexes. We also find complexes of the protein type with nucleic acid, which would be, for example, RNA protein, DNA protein, and the set of RNA provided by what is the CASP competition. And the comparison with two other algorithms, such as Rosetta Fold nucleic acids or an algorithm called alchemy. But in this case, it has an input that is from the human side, right? So it is expected to be relatively better than a completely autonomous one based on neural networks. And in this sense, we observe that in general the performance is not as good as we would like, because in general it is below 50 or 60% success rate. But in general terms, at least in this comparison, AlphaFold still turns out to be a good option when choosing to be able to model this type of complexes with nucleic acids. In another task, which in this case is being able to carry out covalent modifications, whether covalent unions with ligands, glycosylations, or modifications of residues at the level of protein, DNA, or RNA, since there is no algorithm with which we can compare equivalently with AlphaFold3, the results of this algorithm are simply presented, so the same. They perform average to maybe poor, with an approximately 60% overall success rate for covalent modifications. And finally, we have the set of predictions for proteins, which in this case, for example, in the case of monomer predictions, that is, a single domain which is considered a relatively solved task, as we saw in previous slides. Actually, the improvement is a bit marginal compared to what would be the latest version of AlphaFold, which would be AlphaFold2 Multimer in version 2.3, and in this case, there is no great advance. But where there is considerable progress is precisely in modeling what would be the interactions of protein-protein-protein complexes, or even protein complexes more of the type related to antibodies, which are relatively more difficult to model. So this is how we can observe its capabilities in general terms with respect to AlphaFold3, something that we are now going to investigate in detail which aspects of its architecture allowed these performances, which although they are not the best, serve as a starting point from where to improve for future algorithms that already model macromolecular complexes of all types of biomolecules. And before starting this, thank you again. The recommendation of other resources with which you can address this algorithm more, perhaps in a more algorithmic way, that is, explanation of each of the algorithms step by step, which is this idea of T-illustrated AlphaFold, which is created by these two characters, which is Alana and Jake, who really took on the task of creating diagrams to be able to represent the approximately 31 algorithms that constitute AlphaFold3. And even those who have greater experience in terms of deep learning even recognize that their architecture is quite complicated, which is perhaps why Miss Hazard and John Jumper won the Nobel Prize for their version of AlphaFold2. And let's hope that with AlphaFold3, 4 or 5, they will obviously be relatively complex architectures. And another resource that I also highly recommend would be this repository by Shenji that describes each of the algorithms step by step, not so much with diagrams but obviously with explanations of what each object represents, so it can help you understand more easily how the algorithms work and the representations that occur within each of them, because here at least we are going to address it in a simplified and easier to communicate way. And obviously not as detailed as if you directly reviewed the code deposited on GitHub, which is more complicated. So maybe after this class you can consult these two resources so that you can expand your knowledge regarding how AlphaFold3 works if you want to learn the algorithm behind it. This is the architecture in detail, and as you can see it is scary, because there are many modules that may not be so easy to identify. So we can help ourselves with little colors, which makes our lives more colorful and obviously easier. And in general terms, we can segment the architecture as a whole into 3 large parts. One that is dedicated to creating all the necessary inputs. Another that will take these inputs and generate representations or embeddings, rich in information, and another that is precisely takes these embeddings, and you can generate the structural hypotheses of the macromolecular complex that we are modeling. Specifically, we are starting precisely from the idea that we have to pre-process the data. To understand, we still have to do a homology search in databases at the sequence level, but obviously we also have to do a search for homologs now at the nucleotide level, because perhaps also now that we are modeling RNA or DNA, and also in the case that we are working with small ligands, we have to do what is known as the search for conformers, and a conformer, as you can imagine, any molecule, protein, ligand, or whatever has different conformational states, some associated with a higher energy value, and others at a lower energy value, where perhaps it is more easily associated with conditions where there are no collisions between its chains, among many other aspects, which make a reduced set of conformers become the reference or ideal ones to be able to start in a structure modeling task. Then we have to precisely execute a search for the most optimal structures of this type of ligands. These 3 types of information will go through a module that is precisely the input embedder, where all the tokenization and representation occurs at the embedding level, where what we have are 3 main pieces of information. As you can see, what is paired representation? We already know it, and now we have two more representations. One that is the single or unique representation. In this case, what it seeks to represent is the entirety of our macromolecular complex in the form of an embedding. In this case, if we are modeling a number, a protein of 100 residues, then we will have all the tokens related to all our residues. But in the case that we are modeling a protein-ligand complex, a complex that even includes metals or that includes DNA, RNA, well, we have to add all that information that is not a protein in a single embedding, so it then makes sense for its name to be unique or single. And also on this occasion, we are going to preserve all the inputs that we generate with this input embedder, because we are going to have to use them as a power source or seeds for other algorithms, and that they precisely adopt all this type of information represented at the embedding level, which can be related to the bonds of a ligand, the protein itself, the nucleic acids, among multiple other factors of your complex that you are trying to model, for a total of up to approximately 24 embeddings that represent this small diagram. And well, now we are going to move on to what the structural module is, which will precisely take what the pairwise representation is, along with what would be the information of the structural templates, to update the information contained in that type of representation and enrich it. Later, we will pass this new embedding to what is the MSA module, which in this case will obviously take the alignments of both proteins and nucleic acids to further enrich the information. And finally, this representation that, as you can see, first it goes through the template module and then through the MSA module. It will be given as an input value to the Pairformer, along with what is the unique representation of our macromolecular complex, in addition to the input coming from the embeddings of all our inputs of the molecular entities that we are trying to model. And this representation of the Pairformer precisely seeks to further enrich this type of vectors that are going to be precisely used as a seed for what is the diffusion module, where we are going to initialize the atomic coordinates completely in a noisy way. And then it is the diffusion module that is going to have to precisely learn to assign the structure in a conditional way to what are the embeddings coming from the Pairformer, but also what would be the embeddings inputs that we preserve. And finally, we have this structural hypothesis, which in this case we also have to use to be able to predict the confidence, the PLDT, the PAE, the PTM, using all these inputs, both from the performer, along with the input embeddings that we already had, and thus generate precisely these confidence predictions for each of the receipts, as well as the alignment error, among other metrics that we previously explored. So we are going to investigate in detail what happens in these first two modules, which is the preprocessing and tokenization phase of our inputs. In AlphaFold3, there is a particularity in the tokenization, in such a way that what the amino acids or nucleotides are represented by a single token, but in the case that these amino acids or nucleotides are modified, or even if we are talking about any other type of molecule that is not canonical or standard nucleotides or amino acids, we are going to use an atom-level representation of said molecular structure. What does this mean? Here we have the case of a canonical amino acid, which is serine, and as you can see, it is actually represented by 7 atoms that are heavy. That is, we are ignoring the hydrogens here since it is the simplest atom. So what serine really is represented by a total of 7 atoms. 1, 2, 3, 4, 5, 6, 7. Only in this case, because it's a canonical amino acid, it's actually going to be represented as a token. And this is to be able to optimize at a computational level what would be the representation of the entire protein. Here we also have a standard nucleotide, an adenosine or the letter A of DNA, which in this case is represented by 23 atoms, but since it is canonical, it is represented by a token. And now we have precisely the modifications. In this case, we have a phosphorine, which as you can see is a serine residue, but now attached to a phosphate group. So now there are a total of 11 atoms. So we are really talking about 11 tokens. And the same thing happens with a modification at the nucleotide level, which in this case is the addition of a methyl group. As you can see, this is an adenosine. Only in this case we have precisely an added methyl group. In total, we are talking about 24 atoms, so 24 tokens correspond. And likewise, what would be the representation of ligands? In this case, 24. An ibuprofen molecule that corresponds to 15 atoms, and therefore we are talking about 15 tokens. And among this type of representations, one of the most relevant things that also occurs is being able to measure the Euclidean distances between each of the atoms that make up each of these residues. So that in order to measure these Euclidean distances for each of the atoms that make up these residues, we have to take what is known as a central atom. In this case for proteins, it is quite sensible to take what is the alpha carbon of the protein. In the case of DNA and RNA molecules, we take carbon number 1 from each of the residues. And in the case of ligands, well, we are simply representing at the level of atoms, so we would not really be taking a central atom in particular. And in addition to this, information is also taken from the entire macromolecular complex, including the interactions between proteins and ligands. For example, here we have some residues that are perhaps interacting with a particular ligand. And here then we would have to take information. For example, if these residues, they are from the same chain of amino acids, well, from the same protein, or perhaps it is a protein complex that actually provides these binding sites from different chains. So perhaps this residue corresponds to chain A and this to chain B. We also take the information of its three-dimensional position, which would be, for example, its charge in the case of, for example, oxygen atoms or carbon atoms that are more neutral, as well as precisely the element associated with each of the atoms of said interaction. From what you can see is that the representation at the atom level that occurs in this case is actually richer, precisely to be able to model this type of biomolecular interactions in a more general way, and not only to protein residues. And now a processing phase also occurs that is quite important, which in the case that we are working directly with ligands, we are going to use a representation called SMILES, which allows us to represent any relatively small chemical structure in the form of a string of letters, which are actually the elements that constitute said molecular entity. Only in this case, it allows us to represent it as a text string instead of three-dimensional coordinates, which are relatively easier to model and represent. And in this sense, both for this type of ligands and for any protein residue or residue at the nucleotide level, we have to do what is known as the search for conformers. And as you know, molecules are actually assemblies of conformations. So a molecule is really vibrating all the time in time-space, and then there will be some configurations that are more probable due to their energetic conditions and others that are less probable. And we precisely explore this in the class of protein function, and particularly with force fields such as ambers or even force fields that are a little more optimized, so that they run faster but are more implemented heuristically, such as Rosetta's algorithms. And in this case, we are precisely using a force field provided by an algorithm widely used in cheminformatics called RDKIT. And RDKit provides, among many other tools, different force fields. And in this case, we use this field called ETKDG version 3, which precisely takes this type of small molecules, whether protein residues, nucleotide residues, or ligands, and does a conformational search in such a way that it gives us what would be its three-dimensional configuration that is associated to an optimal or more stable energy in this sense. And once we have represented the reference conformer for each of our residues of all types of molecules, it will be given as input values to what is the atom transformer, which in this case its name is quite suggestive of what it does, which is precisely to take all the information at the atomic level and add it so that it now goes from an atomic level to a token level. So that, for example, if I take what would be the atoms of a methionine residue, I have to calculate embeddings for all its atoms and represent them at the level of the methionine residue I am talking about. And in particular, this happens in a more optimized way, in a heuristic way, because processing attention at the level of all methionine atoms of an entire macromolecular complex, it is really computationally impractical. So we then have to implement versions of care that are more economical. And in this case, they are known as implementations of sparse attention, or what would be dispersed or scarce attention, if you want to translate it that way. In summary, what it is telling us is that we are not going to compute all the attention between pairs of tokens, but we are only going to flip to see the nearest neighbors of each of the specific tokens that we try to calculate the tension. And specifically, at least in this implementation in AlphaFold3, what is done is taking what would be the 32 atoms that can only serve their 128 neighboring atoms in relation to the sequence. That is, if we stop at residue number 15. Well, we turn to look at the atoms of what would be residue 14, residue 13, residue 12, but also residue 16, 17, 18. So that when we graph what the tension values would be, we are really observing this pattern of stairs, where we only look at the immediate neighbors of each one, but not at the totality of all the tokens that would represent the molecule. Because again, that is impossible in computational terms. And once this representation was generated at the token level, along with other series of neural networks that also add the information, we then created what is known as our unique or single representation, which in this case, trying to compare with what would represent AlphaFold2, would precisely be taking our representation of the alignment to the MSA representation, and in this case take only the first row that is equivalent to the protein that we are trying to model. This would be its equivalent at the level of AlphaFold2, which in principle must contain information on the residues, the conformers, the MSA, the molecules, and practically all the tokens that constitute the macromolecular complex that we are trying to model. And in addition to this unique representation of our molecule, we also evidently have our pairwise representation, which is also now updated at the atomic level. We just already know it. And precisely the embeddings of all the inputs that we give to the architecture, which in this case, as I mentioned, were precisely 24 embeddings in total, which can be related to what is information at the level of protein or at the level of nucleic acid, and that is represented at the level of type of atom, which would be the bonds, the charge, etc. In fact, here you can see how in total there are inputs that are, for example, 7 embeddings that are at the token level, 6 embeddings at the atom level, 5 embeddings at the MSA level, another 5 more at the level of what are the structural templates, and 1 more at the level of the information of the atoms, and that in what do they represent? Well, you can consult it in more detail in what is supplementary table number 5 of what is the AlphaFold article. AlphaFold3 in particular. Well, now we move on to the structural templates and MSA module. In this specific case, what the templates module is, we are not going to explore it in much detail because it is really a smaller version of Pairformer, which we are going to explore in more detail. But in general, what this module does is take the information from the backbone of the protein, or in general from the macromolecular complex, the histogram, what is the type of residue, as well as the masks that are being used, and add it through a series of linear or even non-linear layers to update the pairwise representation. And as you can see, there are really only 2 layers, and in the case of the MSA module, there are 4 layers. That is a total of 6 layers, so the processing is actually relatively reduced compared to what we observed during the structural module, or the Evoformer module in AlphaFold2. So now let's explore what the MSA module is. And in particular, when we look at the architecture of the MSA module, really what it reminds us of is an equivalent of the extra MSA stack in AlphaFold2, or in short, a mini Evoformer, which is really what we are looking at here. As you can see again, we have a channel where the representation by pairs will go, and another channel where it is precisely from the MSA. And again, in this sense, since we are limited by the computation, we have to do this strategy of subsampling clusters of our alignment, and then we inject the information from the embedding inputs that we generate. So we then take our entire MSA representation as input for this MSA module. And in fact, this is the only time where we precisely use this representation in its entirety, which contrasts quite a bit with what would be how the MSA representation is used in AlphaFold2. And as you can see, there are really many skip connections here that again, what they do is preserve the information. Only this time, as you can see at least in the MSA channel, there is no more attention computation at the column level and at the row level, but there is simply an operation here called the pi-wefted averaging, as well as the outer product mean, along with the classic transition. Remember that this is a multi-layer perception of 2 layers in general to be able to precisely add information. And here in this sense, the only feedback that exists between these 2 channels is through these 2 operations, the outer mean product and the pair-weighted averaging, where we can really see how the MSA information flows through the channel of the pair representation and vice versa. The information from the pairwise representation flows through what is the MSA channel, but not in a very complex way, but simply biasing what would be the calculation of this average in general through a more simplified operation that is not the tension. And here, in what is the channel of representation in pairs, well again, we find what the triangular tension is, that is to say, well, first this more multiplicative attention, this more well basic computation of being able to calculate that, well, what it does is update our representations, and subsequently well carry out the attention in a formal way, or well necessary to precisely enrich the representations in pairs, but already using well-established attention mechanisms. Again, it goes through multi-layer perception of 2 layers, and to precisely update our representation in pairs. And here in this sense, it is quite shocking to observe that there is no longer attention at the level of columns or at the level of rows, but rather we simply have these 2 operations. That when we look at what they represent in simple terms, they are really a much more simplified equivalent that does not require the calculation of tension, but simply averages and then nothing more. In the case of the outer product main, for example, here we will simply take the values actually at the column level and take an average. And in the case of pi-wefted averaging, what we will actually do is, instead of doing an attention computation at the row level, we simply take the pairs of residuals and add the bias from the pairwise representation, and we simply average. This type of information in a more simplified way. And this is due to the fact that information from molecules such as metals or molecules such as ligands is also flowing here, where there is really no evolutionary information that we can distill through tension mechanisms. And what we also want is to optimize the architecture so that it runs faster in the face of much larger macromolecular complexes than just proteins. Therefore, it was then necessary to adopt this type of practices to be able to obtain simple averages instead of doing more intensive calculations by calculating the tension. However, notice here that there is a feedback, in this case only by 4 layers of this module, to what would be the pairwise representation. But what is the triangular tension? Both because of their multiplicative mechanisms and because of the mechanisms formally composed of tension, they continue to be preserved. And when we turn to see precisely what effects this has at the level of dependence on the MCA to be able to precisely model complexes, we see that AlphaFold3 is less dependent on this type of information compared to AlphaFold2. As we saw, this was the graph that showed us precisely how dependent AlphaFold2 was on how represented a protein is within the PDB. And this would be the equivalent of what this behavior is in AlphaFold3, where again we have this metric that is the NEF on the x-axis, and what the macromolecular complex is also being predicted on the y-axis. And now we find the comparison between AlphaFold3 in light blue and AlphaFold2 in darker blue. And we really observe that there is not a big difference, but at least during a phase that is represented by alignments with low coverage, such as by just a few dozen sequences in alignment. For example, from here, which would be up to 100 sequences. We see that in these early stages, that is, with a few dozen sequences in our alignments, AlphaFold3 is capable of modeling structures whose confidence values are relatively higher compared to AlphaFold2. But really, in layman's terms, when we look at all the other sequences when they have more homologs in the alignment, since they are relatively comparable in this sense, and it is only in this first stage, where we are talking about a few dozen proteins, where we really see a slight improvement in AlphaFold3 with respect to AlphaFold2 in terms of sensitivity to the number of homologs in the alignment. Once we have inspected what happens in the structural templates module, as well as in the MSA module, we move on to explore what happens precisely in the Pairformer, which, as we previously explored, is responsible for generating embeddings that serve precisely as a seed for the diffusion module and generate precise structural hypotheses. Here we have a comparison of the Evoformer of AlphaFold2 with respect to the Pairformer in AlphaFold3, where perhaps the main differences that we observe is precisely what the channel is at the level of the MSA representation. And how does it interface with peer representation? Because in this case, in the Pairformer representation, there are no more precisely this type of calculations, which would be the calculation at the column level or the calculation of the tension at the row level, as well as really a feedback between these two types of channels. But actually in the Pairformer, what is triangular tension simply occurs. First with these two more multiplicative modules, and then with these two modules that are actually the calculation of the tension together, with the corresponding transition, that is the multi-layer perceptron of 2 layers, and what is the unique representation of our macromolecular complex, really, because it is only influenced in this sense with what would be the injection of this type of information coming from the output of this transition module. Where it is here, where only a relatively smaller version of what this tension would be at the row level is computed, where we really only have one row and an alignment of multiple sequences, where we have to compute this tension per row for each of the rows in our alignment. In this case, it is only calculated in the unique representation of our macromolecular complex in order to precisely update this representation. Along with what would be all this flow of information coming from the pairwise representation, which is also injected into the information of our single representation, along with the actual updating of our pairwise representation. So here you can see more clearly how these more computationally expensive stress mechanisms at the column and row level were actually eliminated for practically a triangular stress representation, along with a reduced form of the tension at the row level in this case, only at the level of our unique representation of the macromolecular complex in the Pairformer. So really the central axis, well as its name suggests, is to update the pairwise representation and inject this information into our unique representation. So now we move on to what is the diffusion module, which will precisely take this pairwise representation, along with our unique representation, as well as the inputs and embeddings that we generate, to be able to generate these structural hypotheses using a diffusion transformer. And first we have to explore this type of architecture, but in a more simplified way, because obviously when talking about diffusion, we are talking about architectures that are considerably more complex compared to the transformer, at least as far as my opinion is concerned. In that sense, here we have the typical architecture that was used to train this type of diffusion algorithms, which is a unit, which in real terms what it does is use a series of convolutional neural networks, where attention modules are actually added along with many skip connections, so that, for example, here we have how this type of architecture is applied to what would be the generation of images from text, where we have to first encode an embedding that gives this notion of temporality during these noise removal steps during diffusion or noising, along with what would be the prompt at the text level that we are giving as input. And in this case, we start precisely from the noise that will aim to model the distribution of images that we are trying to approximate. For example, a distribution of cats. And as we observe, what would be the embeddings of the prompt along with what would be the embeddings of temporality or denoising, since they are being injected by these convolution operations coupled to attention modules. So that, for example, this first complex of convolutional neural networks together with the voltage passes through a skip connection to what would be the last module. And this is precisely what makes it take this U-shape and is therefore called units. The thing is how we can really couple these types of architectures. Now, speaking in terms of transformers, to be able to do it in a more massive way, as this type of transformer architecture usually operates. So then, if we have a child between a transformer and a unit, it is precisely what we have now speaking as a diffusion transformer. And a diffusion transformer really looks like this, which is a much more complex architecture than a simple transformer, as well as more complex than a simple unit, where concepts from both architectures are now unified to be able to generate diffusion using the transformer architecture. As we can see, here there is really an architecture that is really an application to what would be the generation of images. In this sense, throughout this first channel would be the representation of our atomic coordinates. Only here, when talking about images, we have to segment the images into patches or segments, and that is why these modules appear here in particular adapted to images. But we can reduce them to interpret them as the input of what would be our atomic coordinates with noise. And in this other module, we also have precisely the descriptions at the level of, for example, the text, which in this case would be that it was an image of an orange cat. Only in this case, the descriptions are precisely the embeddings that we can generate at the level of, for example, the performer's outputs as well as the input embeddings that we generate during the embedding layer and here it is quite important that we also have some temporality embeddings that precisely what help us is to model this entire denoising procedure, where the transformer must identify what temporality we are talking about, associated with how much noise is being removed in that particular step. And once these two main channels are understood, we can understand what happens in the diffusion transformer block, which, as you can see, today there is actually the classic calculation of what is known as the multi-head voltage, in the sense that we have multiple tension heads, where each one operates this self-tension calculation, followed by an aggregation of a multi-layer perceptron, usually 2 layers, with nonlinear activation functions and a series of layer normalizations, which are represented by these blocks in blue. Only in this case, as you can see. Well, in fact, when we look at the voltage modules again, there's really nothing that we can be surprised about, because we have already reviewed how the transformer architecture operates, where we again have our query keys values. Matrices, we obviously have to calculate the scalar products, pass them through activation functions, make the corresponding projections to arrive precisely at the computation and concatenation of the voltage modules of each of our voltage heads. And then, when we observe precisely this feed-forward network, we are really talking about a classic multi-layer perceptron. We have in this case an activation function of the GELU type, only that, as you observe here, a kind of conditioning has to occur in such a way that attention is fed back by this type of information, which in this case are the descriptions of the macromolecular complex that we intend to model in this case. These embeddings, along with temporality, are passed to a multi-layer perceptron, which is now also coupled with new parameters that help to condition precisely at the level of layer normalizations to regulate how much is being learned or what aspects are being learned throughout the training, in this case with parameters known as alphas, betas, and ranges, and that in specific cases are injected into particular layer normalization modules. And it is precisely this whole set of architectures that makes the diffusion transformer capable of identifying the different steps of denoising or noise removal between each of the capture steps of the structure in general, in such a way that characteristics are learned that are dependent on time throughout the denoising process. And finally, we have again what is another general normalization and really a modification of the general dimensions of the embedding to project them to what would be the final result, which in this case would correspond to the result of an image that would be the noise along with other aspects such as the variance of the image. But when we are talking about protein structures, we are really talking about the output of this type of diffusion transformers. They are actually the coordinates of the x, y, and z axis of each of the atoms of our macromolecular complex. This is actually what happens in diffusion transformers. And really, the diffusion transformer is what happens in the diffusion module, where what we are doing is operating precisely at the level of atoms. What we have here is precisely how it is being conditioned, using the information from the input embeddings of the pairwise representation and the single representation at the token level, but also at the atom level, as you can see. And this is precisely, although it is more preprocessing of the data, a calculation that what it does is update our representations to enrich their information in such a way that first, we add all the information that is at the token level and inject it into the atom representations. And in turn, these representations of both tokens and atoms will be given as input values to different modules, which in this case appear here again, which is the atom transformer. In this case, what would be the encoder, and here in the other case, what would be the decoder. Only now we are going to investigate them next. And here also comes a new layer of information, where we really start from the atomic coordinates of our macromolecular complex, initialized randomly. Only in this case, obviously as the predictions of the structures are made, the positions of each of the atoms of our proteins are updated precisely, represented by blue, yellow, red, green, etc. spheres, which represent the atoms of the protein, as I mentioned. And in this case, we apply what would be rotations and translations randomly, in such a way that applying this type of transformations, what it does is encourage the diffusion module to approximate something similar to what happened during the invariant point attention mechanism implemented in AlphaFold2, in such a way that these properties of equivalence and also invariance. Remember that invariance is actually a special case of equivalence, since it can be learned through these data augmentation techniques where we apply rotations and translations randomly. And although the fact of equivalence is not being explicitly encoded in this architecture, the neural network is being encouraged to learn it autonomously, so it will really depend on whether we also perform this type of training together with the type, quantity, and diversity of data that we are using during training, so that it can try to approximate something that we at AlphaFold2 encode directly, as was precisely the invariant point attention mechanism, through aspects such as this L2 Christian norm, among other aspects. So now what we're going to do is use these updated coordinates of our atoms, along with the representations of our input embeddings, which, as you can see, are enriched by the representations at the token level, to pass it on to what is the atom transformer. But this time, we are going to use it only at the atom transformer encoder level. And in general, here what we observe is that there are 3 layers of what the encoder is, subsequently about 24 layers of a diffusion transformer in general. And this diffusion transformer, its outputs will now be given to the decoder of the atom transformer, which are also 3 layers in total, which in general terms what they do is learn the atomic context of our macromolecular complex on a small scale from the encoder and decoder modules of the atom transformer, but also at the global level of the atom transformer, in this case using the diffusion transformer model, here represented by a green block. And as you can imagine, this is really done because we want to model these quite fine interactions at the level of atoms, but we cannot extend it to the level of the entire protein in its entirety, because there are too many atoms that may be involved. So then we have to first do an analysis at the level of small noisy steps that what they do is, well, learn patterns related more to the specific atomic context, such as stereochemistry, for example. And then more noise is added using the diffusion transformer, which in this case allows us to model larger amounts of noise, because we are already talking about representations at the token level and not at the atom level, where it allows us to really capture the macromolecular complex on a more global scale in this sense. Or when we use the diffusion transformer, as you can see here, we also have as input what are the token level representations of our macromolecular complex, but also because what is the output of the encoder is used, and in general now these new learned representations are going to be given as input values to this last module, which is the decoder of the atom transformer, which in the end what it does is give us precisely then the atomic coordinates of our macromolecular complex only in this sense. We are talking about the fact that the diffusion module really has a nature of more generative artificial intelligence, and not discriminative artificial intelligence, as was the structural module in AlphaFold2. So in reality, when we make a prediction, what we are really having is a distribution of possible responses that may correspond to different conformations of our particular protein. And it is precisely this that makes this specific architecture have a more generative type behavior on this occasion, which is really one of the key points that occurs in AlphaFold3. Now we move on to review what the loss function is, so that here again aspects related to equivalence are not explicitly codified, but rather the model is encouraged to learn this type of behavior and not force it through a set of parameters. This is the loss in general terms, where we can find several regulation parameters, which are precisely these alphas. And we have here these loss terms, which are precisely the loss associated with the confidence metrics, the loss associated with the diffusion mechanism itself, and a loss associated with the histogram of the macromolecular complex. And really, when we see it like this, it is easier to understand. But obviously, when we detail it, we can investigate that really when we investigate what this confidence loss represents. We are in turn talking about other losses more related to, for example, the one that is also predicting the PLDDT values. A new error metric that in this case is called predicted distance error. What it does is measure the absolute distances, or rather, the error of the absolute distances between pairs of atoms. Another one regarding if, for example, we take a protein, a macromolecular complex, which in this case, not necessarily all the structures deposited in the PDB have resolved residues. In this case, in some proteins there are cases where gaps are simply left, and it is pointed out that in that particular residue, it was not possible to collect sufficient X-ray diffraction information or these shadows with cryo-EM to allow an electron density to be resolved for said residue, and therefore, that position is not resolved. So to be able to deal with that, well, a loss is added which makes us classify in a binary way if a residue is really found, resolved or not in the original structure that one intends to model. And finally, we have the loss of the predicted aligned error that we previously investigated in AlphaFold2, which in this case is also regularized when it is in a training phase or a subsequent fine-tuning phase. When we investigate what happens in what is the diffusion loss, we find the same terms properly associated, which would be the fact that this amount of noise is also being removed throughout the denoising steps, along with other aspects more related to what is actually the atomic coordinates of our macromolecular complexes, which in this case we see represented by this specific loss of what is the mean square error, which, as you can see, what it really does is compare the atomic coordinates of the original protein with respect to the predicted coordinates, and then calculate its mean square error. Also depending on what would be, well, respecting some specific distances of what would be, well, the links between structures, only that in this case this is activated in particular when the fine-tuning of AlphaFold3 is being done because first one wants, well, that the model learns coarse-grained representations, and that later with this constructed knowledge, it increasingly refines its hypotheses regarding how to represent the proteins. In this case, you are encouraged to respect these properties as well as respect how much a hydrogen bond measures, among other aspects in general, along with what would be a relatively smoother version of what the LDT predictions are, which, like what these 12 links would be, what they do is emphasize an understanding of the local geometry of the interactions between residues of the macromolecular complex. And as you can see, we don't really have more ad hoc, more specialized losses like the frame aligning point error in AlphaFold2. We also do not have the Evoformer masking losses, as well as the explicit losses of chiral violations or steric clashes. That they actually explicitly injected this kind of understanding into AlphaFold2 so that it learned to respect these properties. In this case, in AlphaFold3, we have a more general representation that encourages the algorithm to learn autonomously and not be explicitly coded, as we did in AlphaFold2. However, something the authors noticed when training AlphaFold3 is that due to its generative nature based on diffusion models, AlphaFold3 introduces, or perform something that is known as hallucinations, or in relatively simpler words, errors of something that should not be here. We have, for example, how to model a protein that has many intrinsically disordered regions, because it does it relatively well. Since in fact it was found that the PLDDT prediction are relatively associated with regions of little structure in this case. However, when we model this protein using AlphaFold2, it does look good relatively to what we might expect from disordered regions. But when we used AlphaFold3, we observed that this algorithm actually tends to predict regions that are supposedly completely disordered structures. In this case with some alpha helical regions, that is, it is adding regions of secondary structure where there are none in principle. That is, it is committing hallucinations. And these are not the only variants of hallucinations that occur, but they can also be hallucinations at the level of chiral violations. For example, in this case, where the chiral center of this specific small ligand is not being differentiated well. Or even much more explicit cases such as the collision of atoms, which is obviously impossible because two atoms cannot occupy the particular shared space. It is impossible for there to be overlaps in space. Thus, in general terms, Alpha-3 has difficulty with proteins that have little coevolutionary information, such as antibodies. Also proteins that have a high conformational diversity, proteins that evidently become difficult to crystallize and therefore have not been deposited in large quantities in the PDB, such as membrane proteins, as well as intrinsically disordered proteins or proteins with a high content of disordered regions, as could be the case in this case. Or even regarding what the ligands are, it is still pending to see that, which is also able to generalize to new ligands that have not been so widely used during AlphaFold3 training. So in order to resolve this type of errors or hallucinations, what the authors implemented is a solution called cross-distillation, something similar to what would be the self-distillation used in AlphaFold3. 2, which in this case is still a set of datasets where fine-tuning is done so that it learns to simulate what the correct conditions would be, which in this case would be to represent these regions as intrinsically disordered, and that from these datasets that try to really approximate a better representation of the proteins. This representation is achieved that is relatively more faithful, and that does not commit this type of hallucinations. Here in particular, we have what this cross-distillation process really consists of. What it does is allow AlphaFold to mimic desired behaviors, or that we already know about precisely how proteins behave. We have a total of 7 datasets that the authors took on the task of curating, and in each of them, a greater or lesser weight is given throughout the training, and also each of them. It has a greater or lesser number of structures from which it can be learned. In fact, because there are not so many macromolecular complexes deposited in the PDB, it was necessary to be able to find datasets from which we could take that information and predict that type of complex with a first version of AlphaFold3, and then retrain on that type of complex, but now already counted at the level of millions. Something similar to what happened in the AlphaFold2 cell distillation, where remember we took the magnify proteins and went from approximately 170,000 structures to approximately 500,000 structures. In this case, in general terms, we have a first dataset that takes the structures of the PDBs and weigh or grade them based on how diverse the structures are, as well as how well they were resolved. For example, if a protein has a resolution of about 2 Armstrongs, we could say that it is relatively good, compared to one that has up to 6 Armstrongs resolution of its electron-dense regions of each of the positions of its atoms. Also, something that matters to us is that because there is a great research bias towards a specific group of proteins, throughout the investigation of proteins deposited in the PDB, we have to cluster this set of sequences in such a way that we have the most representative ones that are not redundant, and this is precisely what constitutes this first dataset. Then we have a second dataset that is precisely distinguishes by trying to approximate this behavior that there are intrinsically disordered regions where precisely there does not have to be the presence of secondary structures, and that in this case precisely by training with this set of proteins that are shown in this way, obviously AlphaFold3 learns to approximate this type of behavior in this specific case of these residues. Then we have this set of dataset 3 and 4, where in summary, we do a cell distillation properly, using the AlphaFold2 algorithm, but to be able to predict proteins that are monomers, with more than 200 residues in total, as well as other small proteins with less than 200 residues, but in this case it can be seen that they are found in millions, where we have approximately 13 million plus 41 million, that is, approximately 55 million structures, to learn from now using AlphaFold3, which would also help distinguish which regions of secondary structures could be associated with a specific combination of residues. Again, now we have a fifth dataset, which takes the information stored in an RNA database, which is called RFAM. And then, we predict again, the structure of these types of sequences that are curated. And once we have predicted what their atomic structures are, we can retrain on them, obviously using criteria of the best predicted ones, so that, AlphaFold3 becomes capable of being able to model this type of RNA structures that are not so abundant in the PDB. And finally, we have a last set of data sets that is really intended to be able to model interactions between proteins and nucleic acids, in particular what would be DNA, so that we still have approximately 16,000 structures that were not available with their structural predictions, and that we took from curated databases such as JASPA. We identify which are the best predicted in terms of quality with the first version of AlphaFold3, and we retrain on the best candidates after a first round of screening. And this is how this cross-distillation process really occurs, which really in its entirety allows AlphaFold to mimic this type of desired behavior, or that we already know previously about how proteins behave. And this type of datasets is also regularized throughout the training phases that constitute really the entire AlphaFold2 training, where mainly what these data augmentations would be. What they do is precisely approximate this equivalence mechanism, as we previously discussed. The idea of this is that if we have a distribution of what the macromolecular complexes would be through these different training stages, together with the cross-distillation datasets, what we do is, therefore, it means that these two distributions of predicted with respect to real ones are relatively more comparable to each other, and that they overlap in that sense. And here what you can observe is precisely the training phases of the AlphaFold3 algorithm, which consists of a first training phase, where let's say that in general terms what is done is to learn the global context of the structures, as well as relatively small protein complexes. In fact, we see this data regarding relatively small protein complexes reflected by the term of how many tokens it is allowed to model, at least during this first stage of training, where the algorithm is limited in its capacity to be able to process a maximum of 384 tokens, and subsequently this capacity is expanded to be able to model increasingly larger complexes throughout the different stages, from fine-tuning 1, fine-tuning 2, up to even 3 stages of fine-tuning. In the case of the first stage of fine-tuning, what is done is to modify different regularization factors, which in this case allow AlphaFold3 to gradually learn what it would be like to distinguish protein-ligand interactions. In this case, proteins coming from macromolecular complexes, this time larger, or even protein regions. Disorder, as it could be precisely intrinsically disordered regions in this case, because we also see different factors where also, for example, here it is shown that so much is being averaged or encouraged that AlphaFold learns these properties of disorder through what would be the regularization of this parameter, where, well, these parameters are played with throughout the different training stages, or they are enabled so that it can be trained on these cross-distillation datasets. As we previously investigated, with what would be the interactions of proteins with DNA molecules or proteins that are later disordered. And finally, we have two more stages of fine-tuning, which is fine-tuning 2 and fine-tuning 3, where really what you do is, once you have learned these structural bases of the minimum required to be able to model relatively larger macromolecular complexes, now it is enabled what would be the understanding of transcription factors and much larger multimedical complexes so that, for example, here we can see the maximum number of chains with which the model can be trained, first passing through a first training stage, approximately only 20 chains up to a maximum of 50 chains. So now we are talking about much larger macromolecular complexes that could not have been learned initially in this first stage of training, because in this case, the authors found that if it is initialized directly with a value of 50, it is much more complicated because to converge during training to a relatively acceptable loss, then they have found that it is necessary to first apply a training phase followed by 3 phases of fine-tuning, since it really was a process of trial and error to really enable modeling these types of increasingly larger complexes. And yet, we also find some dynamics where perhaps it does not reflect our great abilities to be able to model macromolecular complexes beyond interactions between proteins. So actually even the authors themselves recognize that AlphaFold3 is relatively limited, obviously due to what would be the availability of the data, and that even in some aspects it could be overfit for some type of specific interactions. Here what we observe is that some types of molecular interactions are also belonging throughout the training steps, as can be seen here in the x-axis. And what we observe is that what is the understanding of the atoms within the ligands? In fact, it is being modeled relatively well. But for example, when the interaction of these ligands is being modeled with respect to their interactions with proteins, PLDDT values of approximately 60 are found. Even this value remains relatively monotonous over time. And so we can see other types of interactions. For example, what would be DNA chains with protein residues, or the RNA chains with the protein residues. However, the PRDDT values in general terms are around approximately 60%. And again, this is due to the scarcity of this type of data deposited in APDB. And it was even necessary to apply these cross-distillation mechanisms to generate an increase in the data available for training so that AlphaFold could recapitulate these types of interactions. In fact, one of the least present interactions in this type of databases, such as the PDB, are precisely the interactions between proteins and RNAs. So then we can see that it is really reflected at the level of the predictions in terms of PLDT in this type of macromolecular complexes that are really predicted to be of poor quality in simple terms. And that's how the architecture of AlphaFold3 actually happens. We first went through a phase of approximately CASP10, CASP12, where many algorithms were already beginning to use deep learning algorithms, particularly convolutional neural networks, which what they did was derive that type of convolutional information from the alignments. This type of information has already been replaced by the great computing capabilities of DeepMind, with the first version of AlphaFold1, which as we saw is also based on convolutional neural networks. And then this architecture had to be discarded because it simply could not generalize to much more complicated proteins, something that was practically addressed by what would be the transformer architecture implemented in AlphaFold2. mainly with the structural module and the Evoformer, where we could say that this problem of predicting protein structures was solved, but at the level of monomers, but not at the level of biomolecular complexes. And then again, the DeepMind team had to practically get rid of their architecture, and now restart it with what would be the Pairformer and the Diffusion module. Here what we really have is a comparison of what the architecture of AlphaFold2 would be with respect to the architecture of AlphaFold3, where AlphaFold2 is of a more predictive or discriminative nature, and in the case of AlphaFold3 due to its diffusion module it is generative. In AlphaFold2, we are clearly talking about a resolution at the residue level, while in AlphaFold3, we are already talking at the level of atoms. In the case of AlphaFold2, we are talking about it operating on these local residue frames along with the x-angles, while AlphaFold3 operates directly on the atomic coordinates of the atoms in the. In the case of AlphaFold2, properties such as equivalence are explicitly coded in such a way that it is forced to comply with these types of properties, while in AlphaFold3 it is not explicitly coded, but instead injected through these mechanisms such as cross-distillation, as well as loss terms, so that it can learn it autonomously. But the one who can learn it autonomously compared to it being explicitly codified obviously has different behaviors at the end of the training. Regarding the attention mechanisms implemented, for example, in AlphaFold2 we had invarying point attention mechanisms, we had column-level attention mechanisms, row-level attention mechanisms, and triangular attention. And in this case, in AlphaFold3, we really preserve what would be the triangular attention information, along with these new local and global attention mechanisms, in what is the diffusion module. Regarding the China, well obviously we have more custom-made China compared to AlphaFold2, where we have aspects such as FAPE loss, this frame-aligned point error, which precisely injects information related to equivalence. And in the case of AlphaFold3, well, we have these losses as the mean square error, which would be the three-dimensional positions of each of the atoms. In the case of AlphaFold2, how the predictions are initialized, we're talking about this black hole initialization, the black hole initialization, where, as you remember, we initialize all local residue frames at the same origin, and subsequently these frames are free to rotate and move in space, while in AlphaFold3, we use this idea of starting from conformers. Oh, here you have precisely this structural comparison. And something that is also relatively interesting to think about or reflect on is this idea of the bitter lesson suggested by this Sutton, Richard Sutton, who, as you will remember in the large language models and transformers class, it really seems that what search and learning algorithms are those that do manage to take advantage of what is the increase in computing capabilities, and that in order to generalize precisely to biomolecular interactions of greater diversity, it was necessary getting rid of all these more tailored confections in favor of a much more general algorithm, capable of dealing with a greater diversity in the type of biomolecular interactions. So then perhaps we could ask the question of whether AlphaFold3 can be seen as an example of Richard Sutton's bitter choice. And I leave that to you as a task for reflection or discussion with your friends regarding whether it happens or does not happen or half happens, etc. Now we will briefly address the set of developments that occurred since the launch of the article, as well as the AlphaFold3 code, where perhaps one of the most relevant is precisely this race for trying to reproduce the AlphaFold3 algorithm in such a way that there are both academic and private consortia that took on the task of reproducing the algorithm and evidently training their respective models to be able to have these capabilities of being able to model biomolecular complexes. However, as we saw, at least during its early stages where AlphaFold2 was published, but the code was not made, Well, there really was a set of groups that arrived at their same implementations through trial and error, finding relative inconsistencies between what the pseudocode would be with respect to the theory of deep learning. So a lot of time and money could have really been saved in this regard. And in this sense, it is clear that we have what the AlphaFold3 algorithm is. But we also find equivalent algorithms, such as Chi1, Dragonfold, Helixfold, BOLS1, Neuroplexler, ProteinX, RosettaFold, Aladdin, LIGO, NEO1, and OpenFold3, where each of them is also associated with a specific use license, which you also have to take into account with respect to your academic or even academic interests of business when carrying out some research, because there may be problems if during some level within your entire framework this type of algorithms were actually used, and then perhaps the assignment of the patent becomes more complicated. Because using an algorithm with intellectual property such as AlphaFold3, which is not associated with commercial use, but is strictly only for academic research. And in that sense, perhaps unless they specifically seek the patent, it would be better to adopt variants that are truly open source, as could be the case of Boltz, which contains an MIT-type license, which is really permissive for both commercial and academic purposes. Or perhaps even the Apache 2 license is also quite permissive for these types of activities. Even Boltz alone is not the only algorithm that has been updated, but there are already several updates to all these sets of algorithms currently, so that, for example, there are already several versions of CAI, as well as, for example, right now we are in the BOLTS2 version, and there are even subversions of Boltz that are specifically designed to generate proteins of the binder type, among other aspects. Or you can consider the licenses. In terms of the developments that you intend to make, it is quite important for your specific analyses. Other interesting findings also come from these two publications, where in summary, what they do is illustrate these capabilities of being able to model biomolecular complexes. In this case, in what would be biomolecular complexes of the type of proteins related to their interaction with, which would be lipids, or in particular oleic acids. And here what we observe is that, in this research, in summary, what they do is the structural determination via cryo-EM mechanisms of a macromolecular complex known as the resistome, which we can see in different views, such as the top, the bottom, and the side. And we compare it with respect to what the AlphaFold3 predictions would be using oleic acids or without using this type of oleic acids. And what we observe is that by adding this type of lipids, there is really an improvement in terms of the PLDD confidence metrics of the macromolecular complex. And it turns out that when the type of contacts between residues is inspected, with respect to what the structure determined using cryo, M would be, it turns out that the interactions determined from AlphaFold3, they are mostly recapitulated with respect to what this other method would be using cryo-N, which suggests that on some occasions and under quite specific conditions, biomolecular interactions can be recapitulated, such as protein interactions with ligands, as this case illustrates. And here is another second example related to this, specifically to what lipids would be, something that is much more complicated for AlphaFold. Regarding the fact that this type of data is not present in the PDB, where although in this other investigation it was not determined by X-ray crystallography or CRIM or any other mechanism what the macromolecular complex would be, similar behavior is observed where the addition of lipids precisely modifies what the confidence ranges to the AlphaFold predictions both in terms of PLDDT and PAA, as can be seen in the case where the presence of these lipids is precisely added. Better predictions are obtained both in terms of these two metrics compared to when the lipids are not precisely added. which suggests that perhaps AlphaFold is generalizing in some specific cases, as could be in this other macromolecular complex, which is a specific hexagon. However, this is much more difficult for other types of biomolecules, especially what would be RNA, because it is not one of the most abundant molecules in the databases. For example, when we look at the sequence level, it is clear that we have protein databases numbering in the billions, such as the Big Fantastic database. However, when we turn to look at the level of RNA sequences, it turns out that these sequences are obviously much rarer compared to proteins, in such a way that we would barely have in this type of databases, such as central RNA, less than 100 million RNA sequences. And even worse, what would be certain three-dimensional structures deposited in the PDB, where we have approximately 6,600 RNA structures, compared to what would be the approximately 250,000 protein structures deposited in the PDB. So it is really much clearer that the information at the RNA level is much scarcer. But not only at the level of information deposited in the databases, but also in information that we can distill from this type of representations. For example, when we talk about RNA, it is clear that we only have 4 symbols to represent this type of structures, compared to the 20 symbols that represent protein chains. In this way, each of these 20 wastes also has more diverse behaviors, such as hydrophobicity, charge, size, volume, etc., with respect to actually the practically hydrophilic and negatively charged behavior that RNA molecules present. And so, then, multiple physicochemical properties that make RNA relatively much more complicated to model, compared to what protein interactions would be. And this is something that is recognized by independent research, where here we can see precisely 2 examples of this, where what they really suggest to us is that we can also predict this type of structures related to the world of RNA, since it will depend on how much they resemble the previously known structures with which the algorithm has been trained. And really, the performance of that type of algorithm similar to AlphaFold3 or AlphaFold3 itself will depend quite precisely on the similarity with the data set with which these types of algorithms are trained. So it is not so easy to find generalization, at least in cases of interactions with RNA. We also found somewhat similar data regarding the interaction with ligands in this research that I also highly recommend that you investigate, where precisely what they do is investigate that this type of protein-ligand interactions are also being modeled in such a way that they determine that at least the current scaffolding methods of this type of complexes, what they would really be doing is memorizing the poses of the ligands present in the training data, which we observe in this graph. That on the y-axis is how well these biomolecular complexes are being predicted, and on the x-axis is how similar they are in terms of sequence similarity in relation to the training set. And here we observe different sequence similarity cuts. For example, the most extreme would be these of approximately 0 to 20% sequence similarity, which is telling us that it is already below the twilight zone. Even here there are other examples of 20 to 30 or 30 to 40, that perhaps we can still consider it still within this zone of the twilight zone. And we see precisely the comparison of different biomolecular prediction algorithms, such as AlphaFold3, ProteinX, K1, and Boltz1. And specifically, what we observed is a very similar behavior between all of them, even though they were trained independently, right? All of them including AlphaFold, although it is the one that performs the best here. We observe that as the type of protein-ligand interaction becomes more similar, in this case represented by, for example, 80 to 100% sequence similarity. Structural predictions are actually obtained that are, well, with a success rate of 80%. But when the sequence similarity between at 30-40 are lower at these scales, as we can see throughout this region, there really isn't a considerable success rate because it drops to about below 20%. So it is suggestive that it is effectively memorizing the poses of the interactions between ligands and proteins, something that they investigate a little more in detail regarding this preprint, which is why I recommend you do a lot more research if you want to learn a little more, where precisely what they suggest in order to address this type of problem is to incorporate terms inspired by physics, chemistry, biology, to be able to model more and more precisely this type of interactions, proteins or ligands, right? Regarding the fact that, for example, in AlphaFold2 these types of interactions were, well, explicitly coded, while in AlphaFold3, well, they were only promoted through, well, this whole set of techniques such as terms of the as well as random rotations and translations during the diffusion process, which, well, promote the adoption of equivalence, but it is not explicitly modeled. Also then, it is clear that it's wise to think about what future versions of AlphaFold2 will be like. And it is quite evident that in order to train any type of algorithm based on neural networks, or in general any artificial intelligence, we need better data in terms of quantity and diversity. And in that sense, it is quite represented by this meme, where you can usually find quite a few suggestions from the computing community more related to what would be deep learning architectures or even optimizers. But very rarely do you really see suggestions to improve the quantity and diversity of data by carrying out experiments in such a way that then most of the allocated budget actually goes into training this type of algorithms, even though they do not meet the data. And this is a shame because instead of investing this entire budget in just training, it would perhaps be smarter to invest part of it in generating new data that allows future algorithms to really generalize to new types of interactions. And this has actually been recognized by multiple consortia, both academic and private, such as even DeepMind itself, and for this reason, this new consortium was actually created, which is called OpenBind, and that at least in the following years they will take on the task of crystallizing protein-ligand complexes for a total of approximately 500,000 complexes of this nature, which would represent about a 20-fold increase in the type of molecules that are already deposited in the PDB with respect to this type of interactions. So it will be quite interesting to pay attention regarding the results of this consortium, who at least tell us that their data will also be open source, so that we can also train future deep learning algorithms of this type of models. Also, another aspect that must be reflected on is the fact of how incredibly rare an algorithm like AlphaFold2 really is, because when we start to inspect the literature, how many algorithms have this equivalence property? It is actually quite difficult to find algorithms that have even more than 300 million parameters. As you may remember, AlphaFold2 has approximately 100 million parameters, and yet it is one of the rarest algorithms to find because it explicitly injects properties such as equivalence into this type of algorithms. It is terribly difficult, and it is not so common to really scale these types of properties, and we have not found the mathematics to be able to train deep learning models at the scale of billions of parameters. But when we are talking about equivalence, we are really a little limited to models that are a few hundred million parameters, and not as large on the scale of GPT-4 or GPT-5 or Gemini. So even this is somewhat represented by this meme, where there is some part of the community that strongly defends the idea of being able to explicitly include this type of desirable properties on the part of biomolecules, such as precisely the equivalence of the SC3 type, along with what would be the specific discrete ones that are promoting the understanding of this type of biomolecular interactions, and that they are not as learned autonomously by the algorithm in this case. I am more within this part of the distribution, and where perhaps some colleagues who are precisely behind the advancement of this type of algorithms adopt more other practices of autonomy by deep learning algorithms where it is enough to actually use diffusion mechanisms together with other data augmentation mechanisms that make the algorithm itself learn those types of biochemical properties. And this is also a matter of opinions between each member of the community. So at least you already know what mine is. I am on the side that these types of terms can be explicitly incorporated so that then these models, they are really learning biology, biochemistry, or biophysics explicitly, although this is perhaps at the cost of generalization. And this will be aspects that we will see in future models, and see how much these types of properties, such as equivalence, can be adopted in future models. Regarding what the community adoption would be, and their opinions regarding what AlphaFold3 is. Actually, it has been seen that in general terms we can say that AlphaFold3, although it is very skilled in being able to model biomolecular interactions, is not really understanding in physical terms the contacts and interactions that exist between this type of biomolecules, and that, even as we saw previously, such as in the case of interactions with ligand proteins with respect to their memorization, perhaps what they would be. The performances that we saw when compared with various algorithms may therefore be biased by what is known as data leakage, and therefore are overestimated in that sense. And we also see this type of observations reflected on social networks, where several colleagues upload their examples of cases that are practically impossible on a physical level, and yet AlphaFold3 points out as well, high-quality structures that, in this case, we see here in particular how there is a superposition of atoms, in this case of a folded beta, with respect to this disordered region, and despite this superposition that is physically impossible, AlphaFold3 points out that it is of high confidence, which does not make sense. So there are even other colleagues who say, I have quite a few examples of this type, that despite the fact that they have high confidence, they are physically impossible. Which makes us think that precisely because the models made with with AlphaFold3, can really be seen as garbage, and that really perhaps the models generated through AlphaFold2, because they have this explicit coding of properties such as equivalence, they are more reliable in this sense. This is not the only type of opinion. For example, there are other colleagues who do the same on social networks. Well, they have spoken about it. For example, here it is suggested that perhaps the predictions made using AlphaFold3 would be inferior compared to AlphaFold2 in terms of the quality of the predictions, because this type of physicochemical properties are not explicitly included. Others also point out what it is precisely because this ability to generalize is not as good as we saw in the case of ligands or in the case of RNA molecules. That in general terms what they do is precisely see AlphaFold3, the models generated by AlphaFold3, because perhaps in some cases as garbage that has quite a few physical-chemical violations that are not plausible in neither physical nor biological terms. And despite that, the algorithm actually casts it as a good prediction, even with high confidence. And although perhaps my Twitter account is not the best forum to discuss this, at least I once conducted a survey that I am also on the side of AlphaFold2 regarding the quality of its models, especially when we talk about monomers, which are much easier, even complex protein-based, but not so with biomolecular interactions, because there it is clearly limited compared to AlphaFold3, which is quite clear that includes these models. Errors elegantly called hallucinations. And in this case, well, I asked at least in my community, excuse me, well, what system would be preferred? Whether AlphaFold2, AlphaFold3 or some other algorithm, such as Boltz2. And at least in my survey, well, it didn't have that many votes, but approximately 60 people voted, and it is curious that, well, 36% of the total votes, equivalent to 21 votes, precisely prefer AlphaFold2 models over AlphaFold3 models. And when we go to this type of responses from other types of algorithms, AlphaFold3-like models appear precisely, such as Chi1, Chi2 or Volts1, Volts2, which are also similar to FOL3, but are precisely open source and hence their adoption. So then, at least as a personal suggestion, I would tell you that if you are modeling monomer-type proteins or protein-protein interactions, it would be better to adopt FOL2 and not FOL3, and that only in the case that you are modeling biomolecular complexes, such as, for example, they could be interactions with RNA, with DNA, with ligands or metals, because clearly we are limited there, and then we must select precisely AlphaFold3 or an open source variant to be able to model this type of interactions. In addition to these reproductions of AlphaFold3, its capabilities have also been extended to be generative to multiple types of interactions. In this specific case, there is a second algorithm, launched by Google DeepMind, called AlphaProteo, which takes AlphaFold3 as a base, and in this case uses it as a generator of binding proteins to other proteins known as binders, and then through a series of algorithms what it does is filter the best candidates that can precisely bind to our specific target protein, such as the interaction of antibodies, something that is quite sought after in the world of pharmaceuticals. I'm already losing my voice. We also have, for example, the case of Boltz Design 1, which in this case what it does is create the architecture of Boltz 1, which is an equivalent to AlphaFold3, so that it can generate biomolecular structures of any type conditioned on some specific interaction, such as precisely generating structures capable of binding to a specific ligand. From a specific nucleotide, chains of DNA or RNA are made, proteins that bind to a metal, also that are capable of satisfying interactions of covalent modifications, and clearly also of the protein-protein type, of the binder type. And this is done by coupling different algorithms such as BOLS1 itself, along with others such as LIGAT-MPNN, which is a generator of sequences from structures, and that we are going to review in the protein design class, along with what would be the conditioning of some target structure that can be seen in terms of PLDT, the PAI, and the PTM, which in general terms what it does is use this type of information to condition the generative capabilities of the algorithms and backpropagate the entire signal towards the structures from which they are hypothesized, and thus generate both sequences and structures that can precisely recapitulate this type of interactions that one desires. It's also quite sensible to wonder what AlphaFold4 is going to look like. And in this sense, we can look at precisely what is currently trending in deep learning research, which in this case is, as we already saw, transformers were really the adoption by AlphaFold2, and currently many huge models, such as Gemini or GPT-5, really operate under the expert mix architecture, which in summary terms is the same as a transformer architecture, but in what is the feed-forward network part it is replaced for even more feed-forward networks, where it is now a router, which is also another feed-forward network in charge of assigning an expert, that is, a particular network to learn specific patterns. And this is precisely how a greater optimization of the parameters that are activated when this variant of the transformer is executed is achieved. It is also quite clear that there is great interest in incorporating physicochemical and evolutionary bases by these algorithms, so that they learn to explicitly model interactions such as chirality, collisions that are physically impossible, or even train with molecular dynamics data, so that they also recapitulate the diversity of conformational assemblies over time. Or perhaps also currently what would be the current trends based on reinforcement learning, such as the different types of training regimes, such as the PPO or DPO algorithms, which in summary, what they do is allow the agents, which in this case would be AlphaFold3, or variants similar to AlphaFold3, taking different options and choosing which ones would be the best based on some quality metric, such as the PLDT, the PAE, or the PTM. And in order to explore this question in more detail, I highly recommend this podcast, which is quite interesting from a colleague who has his channel on YouTube called All Posting, who also even has his account on Twitter, or to a colleague that many of us appreciate, which is precisely Sergey Ovchinnikov, where they are more dedicated to discussing what the next version of AlphaFold would be like, as well as future developments in this regard. For example, there has already been some research on this, where, for example, we find this particular preprint, where what the authors do is suggest a mechanism called query by asset outer product. So when AlphaFold calculates a tension, it is not really looking at the most important sequences, the most relevant in terms of its specific protein family. It only does so at the level of homologs, but not so much at the level of evenly its protein family. And as you already know, the organization of protein sequences can occur at the level of family, superfamily, and even folding right. And then precisely here we have an element of that type, where there are sequences much more similar to what our query protein would be. There is a subset of proteins that are homologous, but that perhaps would no longer correspond to the family of our protein. We do not really know. We would have to see the structural details. And there is another subset represented in red, where perhaps only homology can be detected. But we are no longer so sure that they belong to that specific protein family, and perhaps they belong to the superfamily in general terms, or perhaps in terms of folding globally. And what does this mean? Well, when we align the structures of the homologs belonging to this alignment, we find a structural diversity such that, for example, there are proteins in orange, which are quite different from what would be the proteins in green, or even the protein in blue, which has this whole other region of folded alpha helix and beta structures as an extra addition, and which are nevertheless part of what would be this superfamily of proteins. So when we are calculating the tension, perhaps we should pay more attention to it, to homologs of the specific protein family that we are trying to model, and not to all the homologs that in this case are much more different in this sense, that there is a whole protein set here different from those others, and this is precisely what this type of operation called query by acid outer product does, where what we see is, for example, in the case of a protein called OMPR, because being able to detect, using some metrics, such as this one called the Hamming distance, which is practically an approximation of sequence similarity, see how much tension can be assigned to what would be all the homologs with respect to the specific homologs to that family related to OMPR. And what is observed is that, through this query by acid outer product mechanism, really, a tension represented here by red dots is much greater. For example, here we have a margin of, for example, an arbitrary measure, which in this case tells us that it is 2, that is, more attention is being given to this set of proteins that are homologous, but are also specific to the OMPR family, and that the attention to the other rest of homologs is not as much, however. It is still considered, which In terms of the representations learned by this type of modules, such as the Evoformer or the Pairformer, which use this type of query by acid ultraproduct, perhaps it could be smarter to be able to distill even more details at a structural level. We also find other implementations that explicitly incorporate properties, such as the chirality of the molecules. In this case, with what would be the algorithm AlphaFold3, where when you investigate also in this preprint more details of how they are implemented, since several parameters are made that in summary, what they do is explicitly integrate properties related to chirality in such a way that this is also optimized during what is precisely the gradient descent. And finally, we have other advances, such as those I mentioned about Boltz2, where in addition to being able to train with biomolecular interactions, it is also trained on molecular dynamics data from databases such as Atlas, MDCAT, or MISAT, so that here we really have what would be the canonical module of one. Would be the pairwise representation together with our single representation, together with what would really be the affinities provided by this type of molecular dynamics data, so that the learned representations are thus further enriched in terms of the different conformals that may exist in a protein over time. However, it is obviously much more complicated to be able to integrate this type of information into training, because it also destabilizes the gradients over time, so its incorporation is not really that easy. And finally, I would like to conclude with this note from a colleague named Eric Lindahl, which really suggests to us is that any significant advance that we have had in our abilities to predict structures, as well as being able to understand things in bioinformatics, has really come from the introduction of more biology to these types of algorithms, and not necessarily from their sophistication in terms of increasing the number of parameters or making increasingly complex architectures. At least I am very much in line with this opinion, where it would be necessary to explicitly incorporate what we know in biological terms of proteins, and that it is also necessary to go hand in hand with the scalability of this type of algorithm, but always having as a basis this type of properties such as equivalence precisely, so that the algorithms are really explicitly learning to respect these types of properties. So well, we come to the conclusions part. Well, as a first conclusion, we have that what is the success of AlphaFold, both in AlphaFold2 and AlphaFold3, is really built on the work of thousands of scientists who precisely deposited their structures and sequences in databases that have been curated by many other people, and that we can say are very good quality data. And this is quite rare in general in science, in such a way that very few problems can be really solved, as the problem of protein structure prediction was solved by the CASP in that sense. As a second conclusion, we have that AlphaFold2 is not magic provided by deep learning algorithms. Actually, as you observed, it is an engineering product where many ideas were erroneous, and only a few were those that really led to success, and that are really reflected in the final architecture that we see in the Nature publication, as well as in the code. But there really was a lot of trial and error when, for example, incorporating what equivalence would be, how we incorporated it, what mathematics to use, among other aspects. As a third conclusion, we have that AlphaFold3 is not really an emulator of physical conditions of the proteins within the cell, nor is it evidently a predictor of the lower energy structures within the cell, or that is to say under physiological conditions, but really the AlphaFold models, because we have to see them as what they are, that they are structural predictions, and in that sense when seen precisely trained with the data available in the PDB that have their respective biases. Well, precisely this type of algorithms will have a greater tendency to predict structural states that are structured like globular proteins and not intrinsically disordered. that is, reflecting the biases deposited in the databases. As a fourth conclusion, we have that AlphaFold2 really was a victim of its own success. That is, for example, its inductive biases, such as the explicit incorporation of equivalents, in such a way that in order to extend its capabilities in terms of greater and also more diverse biomolecular interactions, it was necessary to adopt a more generalist architecture in this sense. And as a fifth conclusion, we have that under some specific circumstances, some models, some predictions made with AlphaFold2 may be as good as structures determined by classical experimental methods, such as X-ray crystallography or cryo-EM methods. However, both strategies are clearly hypotheses of a reality that is much more dynamic inside the cell. So, with this, we come to the end of this class. In the next one, we will explore in more detail the advances that have been made in protein engineering and its adoption together with artificial intelligence algorithms. So this is all for now, and thank you very much for joining me. See you in the next class.